most importantly, this symposium is supported by independent educational grants from TG Therapeutics, Sandoz, and Sanofi. And this is the continuing education credit, blah, 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 but you get CME for this. These are the faculty disclosures. And did I miss scan this. This is the pretest and also the registration. You may have gotten this in advance, and hopefully you did. But if you didn't, scan that and take the pretest. And then I will tell you we have a very nice afternoon for you with a spectacular faculty that's going to start off this CMSC meeting with some educational material that I think you will like. But I'll give you like 30 seconds to answer these 11 questions. And while you're answering those, I'll just go back over these. These are the agendas. We've got 3 programs as part of this lunchtime series. And keep in mind, there is lunch out there and coffee in the back. And I already told you about that. And the disclosures. So when you're done with that, scan this QR code, because then you could submit questions, which will come to me on this miniature iPad. And I will try and get to them as time allows. But we also will be able to take questions from the floor here, and we encourage them. So our faculty loves to answer questions, and they'd be happy to do that. And so we're going to start out with Professor Benjamin Greenberg, Professor of Neurology at the University of Texas Southwestern, who's going to do a review of biosimilar therapeutics for patients with multiple sclerosis. So, Ben.
All right, hello everyone. Welcome to CMSC for 2026. I appreciate the invitation to join you today and talk about this topic that's going to be taking up a lot more space in our heads and our clinics over the next several years. Dr. Lublin said that the speakers are happy to take questions, except me. No questions at the end. You can submit things in writing. Just kidding. So we're going to review biosimilars in general and then relative to multiple sclerosis. The activity was supported by an independent educational grant from Sandoz. So what are the objectives for today? So first, I want to acknowledge the elephant in the room when talking about biosimilar medications, and that the role of these overwhelmingly is to improve cost and access around the globe. We're going to talk about the regulatory pathway. What is a biosimilar? How is it different than a generic? And we're going to talk about what are the both clinical trial evidence basis and the real world evidence basis to support their use. And then we're going to talk about it specifically relative to multiple sclerosis. But let's start with the elephant in the room, and that is cost. So there's a very small slide. I'll put a red box around the important part. Looking at the initial cost when a drug entered market, and there are several listed here, both oral and infusible drugs, and then as of 2021, what the costs were. And the average cost per year was at $92,000. I remember distinctly, I'm going to date myself, when I was training, and I found out that glatiramer acetate was $9,000 a year. I was morally offended. I just couldn't believe that the drug was $9,000 to $11,000 a year. How could we charge so much money for an important drug? I would give my right arm for $9,000 a year again. At $90,000, this is really tilting things. And when we look at the case for biosimilars and expanded access, the statistics are not just where we are in terms of cost right now, but the annual price increase that happens, which is hovering around 10% per year. If you look around the world when biosimilars are introduced, the discounted prices can be anywhere from 5 to 45%. And if you were to project this in the United States, it would equate to about a $54 billion savings. So one of the questions that comes up is, well, when a biosimilar comes out, does it actually do any good? If you look at glatiramer acetate, which is not a biosimilar, it's a generic, The prescriptions remained about the same in terms of all disease-modifying therapies, but the overall cost fell, meaning that there was replacement with lower-priced items. If you look at Germany, what was found there when disease-modifying therapies came out was actually relapse rates went down overall, and this was attributed to a larger part of the population getting access to drugs that they weren't necessarily able to. And so one of the things that's talked about globally is often a barrier to remission is not the drug, but access to the drug. And this is where anything that brings down costs is incredibly important. But I want to state my personal opinion for the record, and that is pursuing cost savings should not come with risks to patients. And so fundamentally, we have to understand whether or not these next generation of patented drugs provide the same safety and efficacy as their reference products. And this is where the semantics and the vocabulary and the categorizations become important. Not all follow-on medicines are created equally. And this is the largest area of confusion amongst our patients and amongst clinicians around the world. And that's the difference between a generic and a biosimilar. They are not the same. So what is a generic medication? So the FDA gives a definition. It's a drug created to be the same as an already marketed brand name drug in dosage, form, safety, strength, route of administration, quality, performance characteristics, and intended use. Generics are essentially small molecules. So when Tylenol lost its patent, you had generic acetaminophen. And this was essentially a chemically synthesized, low molecular weight compound, and you can characterize it in every way, shape, or form. You show that it is bioequivalent. So when looking down the new drug pathway for a follow-on generic for a small molecule, you have to show that the ingredients, the route, the strength, the bioavailability are all basically the same at a chemical basis. You do not require head-to-head trials when approving a generic medication. And this lack of trials is based on the fact that these drugs are considered interchangeable at the chemical level. And so the FDA uses the reference products approval to accept marketing of a generic medication for a given indication. How is this different than a biosimilar medicine? So the FDA definition is listed here. Note that it says a biologic product that is highly similar to an FDA biologic product known as the reference product with no clinically meaningful differences in terms of safety, purity, or potency. So right off the bat, there's a tension between generic and biosimilar. A generic has to be the same, a biosimilar has to be highly similar. Why the different definitions? And it has to do with the fact that this pathway for approval is really restricted to large biologics, complex proteins derived from living cells. So for everyone in the room who has prescribed a monoclonal antibody, in case you didn't know, the monoclonal antibody was not synthesized chemically by a reaction. It is grown in cells. Imagine huge vats like brewers use for brewing beer, full of cultured cells that are genetically programmed to make the antibody, and it pumps the antibody out into the media, and the company that produces it isolates the antibody from the media, cleans it up, puts it in a syringe, and we give it to our patients. Every cell line that makes antibodies has little differences to it in terms of what the final antibody looks like at a glycosylation pattern more than anything else. And so when individuals have a patent on a biologic, they don't just own the biologic, they own the cell lines. And so if you want to create a biosimilar to a reference product, you have to do it in your own cell line. So by definition, the two molecules are not going to be 100% identical. So the pathway for approving a biosimilar is not the same as a generic. It's referred to as a BLA pathway. So this requires a full analytic, structural, PK, PD, and comparative clinical data. And the FDA looks at the totality of evidence. This is a far more rigorous process than what is used to approve generics. And importantly, the definition of no clinically meaningful differences means you have to do a clinical trial. Generics do not require any clinical trials to move forward. Biosimilars do in order to get an FDA approval. And this leads to a lot of misunderstanding between generics and biosimilars. They are not the same, and the evidence used to approve a biosimilar is not the same as one to approve the generic. So what does the path look like relative to biosimilars? So on the left, in blue, are the stages of development for a reference product. Pick your favorite monoclonal antibody, insert here. At the bottom, it starts with analytical operations. That's the foundation. What's the amino acid sequence of the antibody? What are the cells that are going to be used to produce it? You then do preclinical work, animal studies, cell culture studies. Does the drug do what we think it's going to do? Then you move into phase 1 trials, phase 2 trials, phase 3 trials, and you get an FDA approval, and you're marketed and patented and protected for a period of time. Then your patent protection runs out, and companies can come to market with biosimilars. What do they have to do to get FDA approval? They have to do a lot of analytical characterization. In fact, more than the reference product, because they have to show in every way, shape, or form that they are highly similar to the reference product. Often they are not required to do preclinical studies, but most of them do. And then they do PK, PD, and ultimately a confirmatory clinical trial to show the clinical equivalence between the reference product and the biosimilar product. And when you line up the difference between generics and biosimilars, and I won't belabor this much further, you can see that there are distinct differences that are incredibly important for us to understand, because the number one misunderstanding in the clinic when I discuss a biosimilar with a patient is they use the term generic, and totally understandable until you spend hours reading through FDA guidelines, which I'm sure everybody does in their spare time. There's no reason for any of us to know the difference between a generic or biosimilar, but as you can tell, there are distinct differences that have meaningful implications for our practice and for patients. And this has become a focus in the world of medicine because of a lot of concerns that have been raised around generic medications. So Catherine Ebon wrote this book, Bottle of Lies, great title, looking at the real-world risks of generics. And it wasn't specific to multiple sclerosis. This was just an investigative journalist looking into the international community of generic manufacturers. And this made a bestseller list and got a lot of attention. And for anyone who's interested in hearing a lot about this, my colleague Darren Akuda will be presenting the John Whitaker Lecture later at CMSC, and his focus is going to be on generic medications. And I'm going to show you, I'll give you a little preview of some of his data. I'm going to steal a little bit of his thunder. Don't tell him I did that. But this raised a lot of concerns in the world of generic manufacturing, because what the author uncovered was there are lots of manufacturers around the world in the business of generics. They don't get inspected on a regular basis. Some of the individuals are unseemly, cutting corners, and not living up to the standards, and we don't have an ability to really enforce regulations on a global scale. So they may turn in paperwork certifying a certain product. Whether or not that product turns out to be exactly the same is a question. And the issue is there are so many manufacturers of generics in the world. There's tons of them. You cannot control which manufacturer is supplying your drug on January versus February versus March. And all you have to do is ask our patients on generics, and they'll tell you, well, last month it was a little white pill. This month it's pink. The next month it's yellow. So it's not as if when you get a generic that you are locking into one manufacturer. Your pharmacy can change it on a monthly basis. That's not what happens with biosimilars, but it's wreaked havoc in the world of our trust for non-name brand medications. And I'm going to show you some data, good, bad, and indifferent, in the world of multiple sclerosis relative to generics. And I'm going to start with glatiramer acetate. Does anyone here, I know some people know, what glatiramer acetate is? Is it a small molecule in the way that fingolimod or teriflunomide or the other drugs are? It's not. It's taking 4 amino acids at a certain ratio, putting them in a blender, and creating these polypeptides. And I'm simplifying a little bit, but I'm not too far off. It's like the margarita of proteins, and it turns out that at a certain ratio, it has properties that look like some myelin proteins, and was thought and has been shown to shift the immune system. So if you're a generic manufacturer, how do you show that your product is the same as name brand, what was marketed as Copaxone, the original glatiramer acetate? So they had to come up with a whole list of assays, and I'm going to wait while you read each and every word on this slide. The point of this slide is just to show the variety of assays that were done, and they just had to show off to the side that the proteins that came out of the blender at the end were the sizes that you would expect when compared. And this should also remind everyone that every time we get a batch of glatiramer acetate, it's not exactly the same. So just think for a moment, as we've been injecting patients now for over 20 years, and year to year they get different batches, it's never the exact same batch. It has the same properties, but it's not the same. So this created a lot of interesting challenges for generic manufacturers and regulatory authorities. And there are a variety of different manufacturers out there who have taken different assays, and you can see the red and the blue, and no two manufacturers are doing it exactly the same. When we've looked at different generic versions of glatiramer acetate that came in the market, and this is work published by a colleague, George Hutton, and colleagues of his, you could look at claims databases comparing reference product to generic glatiramer acetate and find that the rates of relapses or claims-based relapses or use of steroids was the same or even lower in the generic version than the reference product. So at least in post-marketing surveillance, not the best data in the world, but it is what we've got, there was no meaningful, obvious difference. But remember that this can hide individual patient experiences. And this is where I'll turn our attention to teriflutamide. And again, as I said, a little bit of a preview of what you may hear from Dr. Okuda in the coming days. So I give Darren a lot of credit. He first reported on this case example, a 50-year-old woman who was seen at our center for multiple sclerosis. Her history is laid out here. She was treated with teriflutamide, the name brand Aubagio, when it was still under patent. She had moved from glatiramer acetate to teriflutamide, and she was doing well. And then the generic came out, and her insurance said, you need to take generic teriflutamide, not name brand anymore. This is a common event in our practices. And not long after the switch, she had a documented relapse. Now, I'll be honest, I would have said, wow, that stinks and that's highly suspicious. I'm not sure I would have done anything other than switch her. Darren said, bring me your bottle from home. And he sent it to an independent lab for testing, where he found, instead of there being 14 milligrams of teriflutamide in each pill, there was just over 7 milligrams of teriflutamide in each pill. And so it showed that her breakthrough disease, after having years of stability, taking 14 milligrams a day, I could argue 7 milligrams is probably not as effective as 14, and that lowering of the dose probably represented an independent risk for her relative to her relapse. And so this sparked a lot of interest in our group, again, led by Darren, to look at other generics that were coming out. And so I'll turn our attention to fingolimod. And Darren took the exact same approach, and the whole center helped out in terms of recruiting patients who would bring in bottles. He also got bottles sent to him from elsewhere, from different manufacturers. And in order to protect the names of the innocent and the guilty, they are called generic A, B, C, and D. And you can see on the right a range of dose. For any given drug, including name brand, there is a range of acceptable milligrams. It's never always 100% of the dose that you expect. It can be anywhere from 99 to 101%. That's perfectly acceptable. But you can see some as low in the 70s. And when we looked at patients who had made the switch from name brand to generic, one of the things that was looked at was the absolute lymphocyte counts, which are shown here. You can see the numbers over time. After the switch, in general, there were groups that had an increase in their absolute lymphocyte count, suggesting that, assuming that they are being compliant with their medication, they're not getting the same level of restriction of lymphocyte trafficking after the switch. And many of these, if not all, were related to manufacturers that had significantly below 90 or 80% of the advertised dose in the bottle. And so some of these patients went on to have relapses, and some of them switched off drug, either back to name brand or to a different drug. Separate from this, colleagues looked at the notion of switching from name brand to generic and what impact would it have on disability progression. And in this publication from 2023, those who switched had a statistically significant increase in rates or prevalence of disease progression, even independent of relapses. And so there's a concern, again, if we can't control for the quality of the product that we're taking, and it can change month to month in terms of the manufacturer, we never fully know what our patients are taking. In the setting of fingolimod, you can follow absolute lymphocyte counts to get just a sense of what's happening, but with things like teriflunomide, we don't have a biomarker relative to drug to see if somebody's getting the full dose. So there are a lot of concerns. Darren will scare you even more. I highly encourage people listening. It's a great talk. But should these concerns relative to generics cross over to biosimilars? So this is hitting the waves. Our patients are concerned. We're concerned. Practitioners are concerned. Is it fair to ascribe this concern to biosimilars? It's a very different world in biosimilars. So if you look over the last 11 years of biosimilars have started coming out for all indications, this is just through 2021. The numbers expanded since then. There were over 30 approved biosimilars starting back in 2015, 2016. If you look at the EMA, they've approved 88 biosimilars. Many more have gone through the process there. And there have been a lot of studies around switching. And the question is, if you take somebody on a reference product who's doing well, our patient on teriflunomide, on Aubagio, and you switch them, are bad things going to happen or not? So we can look to our colleagues in other disease states to learn a lot, because we have very little data within neurology. But I could argue that if there's going to be disease activation, we'd see it from our colleagues in rheumatology and oncology. And we have to ask the question, are the biosimilars as safe and effective as the reference product? And in patients who switch from a reference product to a biosimilar, do they have adverse events or breakthrough disease? So lots of ways to design this study. I'm going to walk you through a few that have been done and published. So one of the most meaningful was called the NORSWITCH study. So this enrolled individuals with a variety of rheumatologic conditions: Crohn's disease, ulcerative colitis, psoriatic arthritis, rheumatoid arthritis, and these were individuals who were stable on infliximab. So they were doing fine, they had been on infliximab, they felt like they were getting a benefit from it, and these individuals were randomized in a one-to-one fashion, just under 500 patients, randomized one-to-one, blinded, to receive either a reference product or a biosimilar infliximab. Okay? So this is essentially what would happen in our clinics when a biosimilar comes out and you have that patient who's stable and the insurance calls and says, you now have to give this different product because it's cheaper. And we all say, for cost savings to drive this, are we putting our patients at risk or not? This study tried to emulate that. And it looked at clinical outcomes, and this was a non-inferiority trial. So are the reference product and the biosimilar behaving the same? And for each of the individual conditions, there were disease-specific outcome measures, and they just set a threshold of, did people worsen or not? So the outcomes are on the next slide, and I want to draw your attention to the first column. So the first column, which is titled INX, is reference product infliximab. So remember, these are people who are stable on the drug, they want to be on the drug, they're happy with the drug, they sign the consent form, they get blindly randomized into reference product versus biosimilar, and anywhere from 20 to 40%, depending on the disease state, those individuals experience worsening during the course of the study. So on average, about a quarter, 26% of the individuals who remained on reference product had changes in their clinical well-being over the course of the study. So just because somebody worsens after an event doesn't mean it was related to the event, because nothing changed for these individuals. It was the natural course of their condition. And if you compare it to the reference product, which was 29% worsening, this was statistically indistinguishable between the two. And in fact, in some diagnostic groups, people who switched had less worsening than people who didn't. So there's no way to predict or to draw an association, at least on this version of a switch study. And it showed that relative to the reference infliximab, the biosimilar was not inferior, and it suggested that patients could be safely switched. And if somebody does poorly, we just don't know if it's them or the drug. because we can't pull the bottle and say there was half the amount in the bottle. I mean, you could, but with biosimilars, the regulations are different. So the manufacturing is very restricted. There aren't as many manufacturers, and there's a lot of work to show the similarity. So if you look at the world of rheumatology, if you look at the world of oncology, if you look at the world of gastroenterology, in all of these different places, there have been switch studies to show that patients on a biosimilar behave similarly to those on reference product. And the impact of this is pretty significant. Now, I would argue for our patient population, keeping individuals in remission, absolutely critically important. We do not want to sacrifice efficacy for cost savings. That's not a fair trade-off. In the world of oncology, it's a really big deal because now we're talking about survival and progression, disease-free survival. If you look at one group in our neighborhood, Texas Oncology, that tracked when biosimilar rituximab came out, as well as a couple other monoclonal antibodies, they had a $4 million savings in 1 month when switching, and they could not track any higher rate of breakthrough disease after the switch. This is one conglomeration of practice. It's a fairly large network. Let's be clear, Texas Oncology is huge. But if you're talking about saving a system $50 million in a year, that can have very meaningful impacts to how we deliver care. I would love some extra staffing in clinic. I don't know about you guys. Maybe everyone's overstaffed and everybody takes long lunch breaks and leaves at 4 with all your My Charts answered and phone calls returned and everybody spending an hour and a half in the room counseling. But assuming you're not, I'd love to see cost savings turn into just better quality and expansive care, but we have to find savings somewhere. So this is rheumatology, oncology, et cetera. What about neurology? So we have limited data, but we don't have zero data. So what is the current landscape? So if you look at the different drugs, there are lots of generics that have come out, but the biosimilars relative to us really fall into 2 categories. There's a biosimilar for natalizumab that's received, FDA approval, and there is a biosimilar, as we know, for rituximab, or Rituxan, the reference product, that have been used in a variety of ways. And I'm going to start with the rituximab. Why are we never going to see equivalent studies for rituximab in neurology? It's because rituximab is used off-label in neurology. So there's no reference clinical trial to go up against. For all of us who are using rituximab either in neuromyelitis optica or autoimmune encephalitis or even in multiple sclerosis, and I know you're out there, there are some of you out there doing it, there was never a registrational trial. So we're not going to see a head-to-head trial for a non-approved indication. So the only thing we can do is look for head-to-head trials for other indications with rituximab. And indeed, there have been 11 head-to-head randomized controlled trials of biosimilars for rituximab. And essentially, it behaves exactly the same. Whether you're looking in the rheumatologic world with RA or non-Hodgkin's lymphoma, every study that has compared both the safety and the efficacy and the rates of adverse events for biosimilar rituximab, infusion reactions, all of those, cannot tell a difference between reference product and biosimilar rituximab. We're just not going to get this data in neurology. And so I can say from our center, when there was a large switch to a biosimilar with rituximab, we tracked internally for a year and didn't have an increased rate in number of phone calls from the infusion center, number of pages for infusion reactions, or anything that was obviously different. And the nice thing, what gave us all comfort about rituximab switching, what was it? You could check the B cell count. And we all kind of sat back and said, okay, I feel okay about this, because as Ben switched from name brand reference product to a biosimilar, and the next month my B cell count was still undetectable, we all felt good about that. You can't do that with natalizumab. There isn't an assay that you can get in a clinical basis to determine whether or not biosimilar natalizumab is behaving the same way. But we do have a lot of data now in multiple sclerosis relative to the FDA-approved biosimilar natalizumab. So I won't go through this whole list, but remember the foundation of the approval process is the analytical chemistry, and indeed the package included an extensive analytic and structural review of the biosimilar natalizumab compared to the reference product, and ultimately, even in functional assays, binding assays, cell assays, could find no meaningful difference between the reference product and the name brand. This formed the foundation for that required clinical trial. So remember in the NOR-switch study, what they did is they took individuals who were stable on infliximab and randomized them to reference product versus biosimilar. In the Antelope study, it was a little different. These were individuals with multiple sclerosis who had had a relapse. This was a traditional kind of phase 3-esque MS trial where you take relapse-remitting patients who are considering going on natalizumab, and they randomize to either reference product or biosimilar. So these were not previously stable natalizumab-treated patients switching. These were de novo patients starting the drug. And interestingly, you'll see on the bottom that orange line, which the EU stands for the European Union reference natalizumab, at about halfway through the follow-up, a portion of those patients, about 25% of those patients, switched to the biosimilar. So there was data to see what happens with a switch halfway through. So does anyone know why this lists EU reference natalizumab? Is that different than US reference natalizumab? And the answer is yes. So the EU, the EMA, and the FDA have different rules about the antibody preparation in terms of the excipients and whether or not what preservatives can be used in the saline. So the molecule, the antibody is exactly the same, but the preservatives are slightly different between the EU and the US. So in studies, they track the difference between EU versus US, but the antibody is the same. There's no difference in the product of the antibody. So what happened in the Antelope study? Not a lot. The two arms were essentially the same. So these are one of the few studies where you don't want p-values less than 0.05. You want p-values to basically say that there's no difference. You want the confidence interval to cross 0 as it did here. The primary endpoint for the study was cumulative new active lesions. So it was an MRI-based study, and indeed there was no difference. They looked at annualized relapse rate, EDSS, treatment emergent adverse events, and importantly immunogenicity, and could not find a difference between them. One of the biggest questions that comes up with biosimilars is around immunogenicity. Well, Ben, what if somebody's stable on a drug, they switch and they develop an anti-drug antibody? Because it is a different molecule. It has glycosylation differences. And the question is, would that be clinically relevant? So the Antelope study did a lot of work on this, looking at the percentage of patients with anti-drug antibodies. And one of the things I'll point out, if you look at the second to last line, percentage of patients with the neutralizing antibodies, look at those numbers across the board, 88, 90%, regardless of whether or not you're using reference product or not. We constantly make anti-drug antibodies to biologics. The question is, do they have a clinically meaningful impact on the patient experience? And in general, the answer here is they do not. And if you look over time at the rates of and the titers of anti-drug antibodies, each of these groups are making anti-drug antibodies. They're making them in the same titers. And importantly, and it's not depicted here, that group that switched from the reference product to the biosimilar 24 weeks in, none of them who were anti-drug antibody negative went to positive. So the switch did not all of a sudden induce a new anti-drug antibody in that switch population. And importantly, if you looked at annualized relapse rates between antibody drug negative and positive patients, there's essentially no difference, no impact between the reference product and the biosimilar product, and there wasn't a statistical difference between neutralizing antibody positive or negative patients, and you can see it at the bottom, the 0.14, 0.16, all the way at the bottom there. So whether or not you get antibodies didn't have a clinical impact, and the difference between the reference product and the biosimilar was negligible. What other data do we have relative to biosimilar natalizumab? There's two important studies. These are real-world switch studies. So in this one, they were looking at just natalizumab drug levels as you switch from reference product to biosimilar, and essentially they're staying exactly the same. Importantly, over a quarter of patients reported new side effects. That is extremely concerning to me because I don't hear about side effects in my natalizumab patients. Does anyone here get phone calls from their natalizumab patients? It's one of the reasons I love natalizumab. It's a low phone call drug. And so it really does a good job, but that 28% really stuck out. And so in a separate study, there was a look at the reported side effects in an Amsterdam cohort after switch. Here's the important part of this study. everybody's told they're going to switch. So you say, hey, listen, your insurance called, you can't get name brand natalizumab anymore, those jerks want to save some money, we're switching you, good luck. And then people start calling relative to side effects. When they looked at this in a prospective fashion, the individuals who are aware of the switch had the highest rate of phone calls. And this is something that is referred to as the nocebo effect. So we've all heard of the placebo effect. So if I tell you I'm going to treat you, there's a general improvement in your condition. The nocebo effect is the reverse. If I say I'm going to do something that has risk or caution of, ooh, they're switching you to biosimilar, and didn't you read that book about generics, and I don't know what you're getting, and this could be creepy, I have no control over it, sure enough, the rates of having events go up. So this concept, both on the placebo effect, how we position medications, and the nocebo effect tell us how we discuss these medications has a direct impact on the patient experience in a statistically significant way. And indeed, it turns out that both our patients and ourselves have a lot of perception gaps when it comes to biosimilars. If we survey folks, a lot of people have concerns relative to switches, and if you go further into this publication, you find that a lot of people misunderstand what biosimilars are. They use generics interchangeably, but when educated about biosimilars, this concern goes down significantly. So natalizumab is FDA approved now. Rituximab biosimilars are FDA approved now. We're about to get a lot more. So number one prescribed monoclonal antibody in the United States for multiple sclerosis is ocrelizumab. It's the 800-pound gorilla in the monoclonal antibody space. It loses its patent in the next couple of years, 2 to 3 years. And so the studies are already going on for biosimilar ocrelizumab, because if you were a biosimilar manufacturer, wouldn't you want to get in on that massive market? And so we all need to become extremely capable of having conversations about biosimilars with our patients and being able to answer questions, because this is going to be an everyday occurrence for us as we go over the next several years. And indeed, I just wanted to highlight one bit of data talking about the pivotal role of MS nurses relative to biosimilar education. Our nurses are the first, middle, and last source of information about everything, but also biosimilars in terms of how that communication is handled, either on the phone, in person, on MyChart, or whatever the electronic portal of choice is. And indeed, if you ask our patients, what our nurses say is extremely meaningful, and how they say it is extremely meaningful. So it's important for not just PAs, nurse practitioners, physicians, to become very fluent in this language. We need every member of the medical team to feel comfortable discussing what a biosimilar is or isn't, and whether or not there should or should not be concern with a switch. So just some practical thoughts relative to having this conversation, and this is just my opinion. Always lead with the science. A little bit of education goes a long way. The rigorous path a biosimilar takes compared to a generic. Shared decision-making is essential. We really need patients to engage, or that nocebo effect becomes a very large cause of phone calls and messages. And really stressing this notion of no clinically meaningful difference. This isn't just analytical. There are clinical trials showing the equivalency. Once patients hear that, the angst goes down significantly, that it's not the same as generic. And then I'll end with just the AAN position statement relative to biosimilars. There is a position statement out there. And importantly, they say changing therapy from reference-branded FDA-approved biologic therapeutics to a biosimilar is appropriate if there exists comparative effectiveness data demonstrating similar treatment responses. So we have official professional society guidelines telling us it's perfectly okay to do this, as long as the data, like an antelope study or any of the rituximab studies or what we'll probably see with ocrelizumab, exist and can be referenced. So we're not going to have much choice in this, is the honest answer. When we get told we're making a switch, it's very difficult because of the mountain of evidence to argue against it. So we have to be vigilant, we have to watch our patients, and we have to recognize but the evidence is overwhelmingly good when it comes to biosimilars and very different than the generic experience. So key takeaways are listed here. Hopefully this has been helpful and gets you prepared for some of the conversations you're going to have. And as advertised, I am happy to take any questions that you may have. So thank you. This one. I hit the right button. Please. Oh.
I just wanted to see it. Okay. Okay, question right here.
Short, sorry. I had a question in regards, I've heard rumors that, United Healthcare as of July 1st is forcing all of their Tysabri patients to biosimilar. And my question is where with these biosimilars, do they have a REMS program that they're following these patients with and checking their, verifying their JCV and that?
Yeah, great question. So I've heard same rumors. I don't know what's true and what's not. I've heard it about a variety of insurance groups. And part of it's regional. Some regional versions are moving in different directions. So from a regulatory perspective, anything that's required of the reference product is required of the biosimilar. So a REMS program, a tracking program, a certification program, it's not going to be branded the way the reference product one is branded, but there has to be an approach to tracking patients and assessing risk relative, in the case of natalizumab, PML.
Meaning a touch program of similar nature.
I don't want to infringe on any copyrights, so I'll just, yes, some REMS program, maybe it'll be a wave instead of a touch, or I don't know what the acronym's going to be.
So with respect to the Tysabri generic and the REMS program that's been created for it, what's happened is with Tysabri, when they determine whether somebody has developed PML, that's done internally. So they have a physician, presumably at Biogen, that uses the criteria that Biogen developed to call it PML. That's problematic because the criteria that they've employed is very restrictive. You have to have either pathologic evidence or you have to have evidence of JC virus in the spinal fluid and radiographic evidence of PML, both, in order to call it PML. So they are missing cases of PML with Tysabri. With the generic, however, they're now employing two separate means of criteria. They have an independent PML adjudication committee. I know about it because I'm the chair of the committee. And we use the Biogen criteria, but we also use the criteria that were established in 2013 and published in Neurology, where there is a probable category. There's a definite category where, there's histopathologic evidence and you have viral evidence clinically and clinical features consistent with it, or a probable and possible category. And what we're finding is there's a discordance. So it may be that you're going to see higher rates of PML with the generic. I don't know that. That is really unfair because of the criteria that are
Different. Thank you.
So let me ask both of you, what about the new assay that the generic has to utilize? You want to comment on that?
So the JC virus antibody assay that was developed by Leonid Gorelick and Ken Simon at Biogen is licensed by Biogen. So they didn't want anybody else to have that. So that had to be recreated. And the best evidence is that so that assay, by the way, doesn't really tell you whether you've ever been infected by JC virus or not. All it tells you is that the antibody level doesn't meet a certain threshold to call you positive. There are many people that are antibody negative that actually have JC virus in their body. And we know that because we did studies years ago at the University of Kentucky where we looked at cadavers and sampled 40 tissues and then sent the blood up to Biogen for them to do the assay. And even in people that were seronegative by their criteria, they still had JC virus detectable in low copy numbers in their kidneys. When they had higher titers, you could find it in very high titers in their kidney, very high copy numbers, and you could also find it in other tissues. The assay that has been developed that is being used for the generic is actually more sensitive. So you're going to find JC virus antibody in higher numbers of people.
Do you want to add anything to that?
No, it's just that it's a reminder, hopefully it seeped in for everyone's consciousness, this notion that tests are not absolute. So this notion that somebody who's antibody negative is shedding JC virus in the urine, which has been documented, so for even not cadavers, but in live R patients, tests are to be used as a tool, but they are not the absolute in terms of assessing who has an infection or who's at risk for what. So as we switch from test to test, we have to learn the operating characteristics of each of the tests.
And one last question, which I'll give you. If you were the regulatory authority, would you have treated glutamer acetate as a biologic rather than as a drug?
If I were the regulatory authority, when that day comes, it'll be such a fun day. So I would have. Glutamer acetate, there was a big discussion when it came out of should it go down the biologics path. And it was caught in the middle because it's not a large molecule, it's not grown in a cell, it checked all the boxes for a generic, except there's not a single molecule to compare. So they went down the generics path. I look at it, and this is just as a clinician and from a clinical trials perspective, I think the apples to apples would have been more the biologics path. So that's an N of one opinion, but yeah.
Maybe two.
Okay.
But thank you very much,
All right,
Ben.
thank you.
Scan that code, please. Okay. Next, we're going to have an update on anti-CD20. I'm going to give a brief introduction, and then Professor Berger is going to give you much more in the way of details of what's happening with anti-CD20. And you heard a little bit already of where we are with it in terms of people using it and such. And so this is supported from an education grant from TG Therapeutics. And here's my section. So what do we know about current treatment needs? Well, we have relapsing MS, we have over 20 choices, and we've done really well with treating relapsing multiple sclerosis. We still have the discussion of how to choose, and then we have to say, well, what are the data? So there are very few completed head-to-head studies of what we call high-efficacy therapy. There are 2 underway, which may give us, well, no, they won't give us that. We'll never see head-to-head, like, say, natalizumab versus ocrelizumab and such. But then there's this tension in the community, which has shifted some towards starting everyone with high-efficacy therapy. But what are the data? And the data are lots of observational studies, but no really good data. There are 2 studies underway, which hopefully will answer that question, at least on how to start. We don't know yet all of the long-term issues of using anti-CD20, and now that we're into 8, almost 9 years, we are starting to see some interesting rare, or at least uncommon, side effects. But there's going to be a question as well as how long can you keep people on anti-CD20? And so long-term issues are it. The best dosing regimen is something to think about. And then can we stop? And so Dr. Berger's colleague, Professor Amit Baror, is looking at this in a study to see what happens to individuals when you stop anti-CD20 after 2 years, and what comes back in terms of the timing of the B cell return and the nature of the B cells that return, with the hope that they may be more regulatory rather than pathogenic. So that will be kind of interesting to look at. And so what are the values? Well, we have excellent efficacy, and we've had multiple randomized clinical trials which validate that with each of the agents that's out there. We have administration choices. We have 2 of the agents that are Q6 months, plus we have the off-label rituximab, which is intravenous as well, every 6 months, versus monthly subcutaneous. And coming down the road are some variations of that. There's also a Q6 month subcutaneous. And one of the things, although we have excellent efficacy on relapse rate reduction and relapse-associated disability, the effect on progression independent of relapse activity, that is the gradual progression that occurs in individuals when they've entered a secondary progressive disease course, is not as effective. It has an effect in reducing it, but not nearly to the extent rather that we see with reduction in relapse activity. And then for progressive disease, we have little data. We have ocrelizumab, which is approved for treatment of primary progressive MS. We have some data from fenebrutinib, which was a non-inferiority study, which we could talk with as to whether that was impressive or not. And we have tolebrutinib data in non-relapsing secondary progressive MS, which was successful, but the FDA thus far has been concerned about liver toxicity and has not approved it. Interestingly enough, the EMA, the European authority, has approved tolebrutinib for non-relapsing secondary progressive MS. The FDA decision is under appeal. We'll see what happens with that. There is concern about treating active disease and ignoring the underlying progression, which is the true value of our understanding of the concept of progression independent of relapse activity, which really has been brought out by the fact that we've turned off relapses, but that if you look hard enough, you can see in some patients, not all patients, this slow, gradual progression. So the challenges that we're looking at are, A, long-term data. How long will they continue to be effective? And what are the consequences of long-term data? We need to get a better understanding of what would be the best dosing regimen. It may not be a best dosing, but rather an individual preference amongst patients. And that's sort of how we do it in our center. We say, look, we treat the anti-CD20s in many ways as equivalent in terms of efficacy, that the main differences that we see are in terms of how you want to get it dosed. Whether it's every 6 months, how fast you want it every 6 months, because a quicker infusion is a better infusion for us because there's lots of infused drugs, and it makes it easier for us to get people into the infusion center if we can do shorter infusions. We have the safety mitigation over the long term. We have to look at issues of sequencing the drugs and cycling, and then develop more, and maybe Joe will discuss this a bit, distinguishing between the different anti-CD20s that we have. And you already heard from Dr. Greenberg that there will be generics entering the market as well. And so I thank you for your attention. I'm going to call up Joe Berger, and then we'll take questions afterwards. So thank you.
So I'm going to start with this slide, because Fred started out telling you there are now more than 20 drugs that we have available. Mind you, it's only been in the last 3 decades, and both Fred and I, maybe the only 2 in the room, are old enough to remember a time when we had nothing available for our patients. So treating MS, and Amy, Amy's certainly old enough to remember that. Certainly things have come a long way, and what I've circled here are those that are B-cell therapies, and they've really taken over the market, as I'm going to show you in a second. So where did the story with B-cell therapies arise? It arose in 2008 in this paper published by Hauser and colleagues. And what they did was they said, well, maybe B cells do contribute in some way to the genesis of multiple sclerosis. Why don't we try rituximab? And they compared rituximab to placebo, and they gave 1,000 milligrams of rituximab at just twice at 2-week intervals, and then looked at patients over 48 weeks. And what they were studying was one thing in particular, and that was looking at activity on the MRI. And what they saw, as is depicted on this slide, was this rather dramatic decline in active disease as reflected in the MRI. And it persisted for the 48 weeks, and they said, wow, that's amazing. So there's got to be something to this B-cell story. In 2017, we have ocrelizumab released to the market. At that time, it's 10% of the market share. This comes from claims database. Currently, 62% of our drug use for multiple sclerosis is B-cell therapy, and that is climbing on an annual basis. So it was a change in the paradigm. This was really a paradigm shift because up until this time, up until 2008, we thought this was a T-cell disease, not a B-cell disease. And why did we think that it was a T-cell disease? That gets back to the studies that were done in 1935 by Rivers and Schwenker, and they created an animal model called experimental allergic encephalomyelitis. And what they did is they used CNS antigens, here depicted in a mouse, but they did it in a monkey, and they injected it with adjuvant that enhances the immune response, and they demonstrated that within a couple of weeks, these animals developed an inflammatory disease of their central nervous system that was very similar to multiple sclerosis. It was named experimental allergic encephalomyelitis, and that became the model for relapsing, remitting multiple sclerosis. And in fact, before drugs come to market, one of the animal models that they're studied in is experimental allergic encephalomyelitis. Sometimes it's predictive that the drug is going to work, but sometimes it doesn't help us at all. So what are the animal models that we use? Experimental autoimmune encephalomyelitis, which is a disease that is mediated by autoreactive CD4 T lymphocytes directed against myelin. Obviously, what role do B-cell therapies have in modulating that? There are virus-induced demyelination, the Tyler's virus and Semmeliki virus model, and there's toxin-induced demyelination, the cuprizone model, and there's a lysolecithin model. But unfortunately, none of these animal models faithfully recapitulate multiple sclerosis. So using them really doesn't tell us whether a drug is going to be effective or not. It's a clue, but it doesn't tell us. And one of the other things that you need to know is there have been therapies developed against T cells that have been tried. You'd think that, geez, if it's a T-cell disease, T-cell therapies are going to work, and they haven't. To date, they really haven't worked. So there's been monoclonal anti-CD4 antibodies that have been tried. Small study, but it failed to reduce MRI activity. There's been a T-cell vaccination trial. It's a 2B trial of 150 patients. It showed no clinical benefit to, again, looking at MRIs. Altered peptide ligands exacerbated disease activity. Ustekinumab, which is an anti-IL-12, IL-23, also related to T cells, works extremely well in EAE, this animal model, but had no effect on clinical or radiographic relapses. And of course, TNF-alpha blockade makes the disease worse, so we avoid it. What did the investigators say when they published that article in the New England Journal? So this is what was said. They said, in contrast to earlier concepts of disease, suggesting that pathogenic T cells are sufficient for full expression of multiple sclerosis, it is now evident that autoimmune B cells and the humoral immune mechanisms also play a key role. Now, one of the things you then have to do is say, well, how's it playing a role? Where is it playing a role? And there are many things that B cells do. And I'm going to say something that's a bit iconoclastic, because when you read about the drugs that we use in multiple sclerosis, we have these nice, neat theories as to how they work. I am going to tell you, with the exception of natalizumab, I think it's all hand-waving. My belief is, while we may be on the right path, we don't have a full comprehension of how each of the drugs that we use in MS truly works. And we may be very surprised to learn that the way they work is very different than what we had envisioned. But having said that, let me return to what I was telling you. So what are the potential roles that B-cell therapies have in the pathogenesis? Well, B cells participate in the innate immunity. Maybe it's some role there. They certainly play a role in antigen presentation. They have a regulatory function on T cells. They produce antibodies, so maybe there's some role for it in that sense. And they activate astrocytes and microglia. So it may be a combination of these things that is occurring. And the B-cell therapies we use are anti-CD20s currently. There may be newer anti-CD therapies that are against a wider variety of B cells, but for the moment, the ones that are FDA approved, as I'll talk about in a second, are directed against the CD20. They affect pre-B cells, mature naive B cells, mature activated B cells, and memory B cells, which one finds in the bone marrow, the blood, lymphoid organs, and spinal fluid. There are B cells that it has no effect on, and that is important, particularly the plasma cells, the plasmablast stem cells, and pro-B cells. So let's take a look at the 4 drugs that we are using now for multiple sclerosis. Even though rituximab is not FDA approved, as you heard from both Ben and Fred, we do use rituximab. There are people that use it throughout the nation. I've certainly used it for multiple sclerosis. And there are clinics in the United States where you have to use it. Kaiser Permanente, my understanding is, sort of mandates that you give rituximab. Certainly the Swedes are very fond of giving rituximab because it's much cheaper than the other drugs that are available. So rituximab is a chimeric drug. It has a 65% human sequence. It's given as 1,000 milligrams. We, as neurologists, give our rituximab very differently than the rheumatologist. The rheumatologist actually weigh their patients and determine how much to give based on body weight. The frequency is every 24 weeks, and it is not approved by the FDA for multiple sclerosis. Ocrelizumab is a humanized, glycosylated form of IgG directed against the anti-CD20. It's got 90% human sequence. It's given either as 600 milligrams intravenously or 920 milligrams subcutaneously every 6 months. The infusions take 2 to 4 hours, and the subcutaneous version takes 10 minutes or so. Ofatumumab is fully humanized, so therefore the sequence is 100% human. It's subcutaneous 20 milligrams given monthly, and it is self-administered. And ublituximab is glycoengineered. It's a chimeric molecule like rituximab. It's 65% human sequence. The dose is 450 milligrams every 24 weeks, and it can be administered within an hour. They are directed against different epitopes on the B cell. So here you see in the pink what ofatumumab is directed against, in the blue what ublituximab is directed against, and in the green what rituximab and ocrelizumab are directed against. Not that it necessarily matters. However, the structure of the molecule does matter, and one of the things that's been done with ublituximab is glycoengineering. There are these glycosylated molecules on the Fc component of the IgG that are removed, And this makes for more effective binding to NK cells. And the relevance of that is that we kill B cells when we attach this anti-CD20 to them in two fashions. One fashion is called complement-dependent cytotoxicity, or CDC. The other is called antibody-dependent cellular cytotoxicity, or ADCC. When you do it through complement, what happens is there's an activation of the complement pathways with an elaboration of C3A and C5A. These are molecules that give rise to the infusion reactions that we see when we administer these drugs. So it causes the release of monoamines such as histamine, and as a consequence, you can develop anaphylactic reactions, as maybe some of you have seen when you've administered these drugs to your patients. On the other hand, if you kill the cell through NK cells, there's very little of that occurs, and there's direct cell lysis. To show you the difference between the ADCC of these various anti-CD20 molecules, this is a study that was done in vitro, so it's not a human study. But what's demonstrated is that in much lower doses, ublituximab drives the reaction more to antibody-dependent cellular cytotoxicity than the other drugs that require higher orders of magnitude of their drug in order to create the ADCC. So what is the effect of the anti-CD20 monoclonal antibodies? We know that within hours of administration, there's a reduction of B cells. And we know that because there were studies done with ublituximab that showed that 96% of peripheral B cells are depleted within 24 hours of administration. Now, it may be true of the other drugs as well. Those studies, to the best of my knowledge, hadn't been done in that short time frame. But the presumption is that most of them act pretty similarly in that a rapid reduction of B cells. Addition, there's a small percentage of T cells that are reduced. There are a small number of T cells that express CD20. I once asked my colleague, Dr. Baror, who was mentioned earlier, whether those may play a significant role in MS, and that what we're seeing is with the anti-CD20s is an effect on these T cells that are expressing CD20 as opposed to the B cell effect. He didn't have an answer for that. He thought it was an interesting thought. There are certain subsets that are differentially affected. And one of the things that I show you on the right-hand side of this slide is that there's also a reduction in T helper cells that occurs. So you'll see this reduction in T regulatory cells and Th1 and Th17 cells when you administer these anti-CD20s. The effect that once you give these drugs is there for months, certainly with the intravenous infusions. So drugs like ocrelizumab and ublituximab, you administer them, and as was demonstrated in the initial study in 2008, 48 weeks later, you still had an effect. So you have this long-lasting effect after you've provided the drug. The repopulation of the B cells starts in the bone marrow and spleen. We use anti-CD20 counts to assess whether or not we're affecting these cells. And why do we use CD19? And the reason we use CD19 is because the administration of the anti-CD20 monoclonal antibodies can interfere with the test that's given. So we don't test anti-CD20s, we test anti-CD19s. And we consider that the drug, when CD19 levels reach 1 or 2% of lymphocyte counts, that we really want the response to be below that level. So how were these studies that confirm the efficacy of these drugs designed? They were all designed pretty similarly. You had patients between the ages of 18 and 55 who had EDS scores below 0 to 5.5, on the order of 0 to 5.5. And they had to have a relapse in the last year or two in the preceding 2 years or gadolinium-enhancing lesions in the preceding year. And the primary endpoint, as with all our studies for MS, relapsing remitting MS, was to look at annualized relapse rate. That was the primary endpoint. There were secondary endpoints looking at gadolinium-enhancing lesions and number of new T2 hyperintense lesions. And there was a select secondary endpoint of confirmed disability progression and a variety of tertiary endpoints. And then the patients were studied periodically. The initial studies for ocrelizumab were entitled OPERA-1 and OPERA-2. So you did 2 studies to confirm that indeed these things were really working. I mean, if there was a discordance between the studies, the FDA would call it into question. The studies were done between 2011 and 2013. Large numbers of patients, over 800 patients in both arms of this study. They received 600 milligrams of ocrelizumab every 24 weeks, and there was an active comparator, which was interferon beta-1b. The duration of the study was 96 weeks, and the primary endpoint, as I mentioned, was the annualized relapse rate. And what they found was a very significant reduction in the annualized relapse rate by about 50% in both arms of the trial. And they also found a striking reduction in disease activity reflected in the MRI. As you see, so the top of the right-hand slide shows what the annualized relapse rate were in both groups. And the bottom of the slide shows you the decrease in lesion burden over time. So that was very successful. They also did another study called Oratorio that was done concomitantly, and this was a randomized double-blind controlled trial of 732 patients with primary progressive disease. And they studied these patients over 120 weeks, and what they found that there was a reduction of confirmed disability progression by 24% in the ocrelizumab group compared with the placebo group. So that led to our belief that it is likely having some effect on progression. It is not a dramatic effect, as you can see in the slide, but there appears to be some salutary effect, and it is probably not unique to ocrelizumab, that the other anti-CD20s likely have a similar effect. For ofatumumab, the studies were called Askepolos-1 and Askepolos-2, designed similarly. However, the comparator drug in this instance was actually a bit better. It was teriflunomide, and the duration of the trial was 30 months. The primary endpoint, again, was annualized relapse rate, and they found that there was a similar reduction in the annualized relapse rate when compared to the active comparator as there was with ocrelizumab against interferon. They also found that the 3-month confirmed disability worsening was significantly reduced with ofatumumab, as you see in the bottom of the slide on the right, and that the mean number of gadolinium-enhancing lesions were very, very significantly reduced, 97% in one of the studies, 94% in the other. So again, highly effective therapy. And then we had ublituximab, which was the most recent of the anti-CD20s introduced to market. Similar design to the trial. Again, the active comparator was teriflunomide. The duration was 96 weeks. And what they found was, again, a dramatic reduction in the annualized relapse rate when compared to teriflunomide, with almost complete suppression of MRI activity with respect to gadolinium-enhancing lesions. So one of the things that we should be concerned about is NIDA-3. Actually, we probably have NIDA-4 and NIDA-5 to worry about, but the data that's available is looking at NIDA-3, and for those of you unfamiliar with NIDA-3, it's no evidence of disease activity in 3 separate spheres. One is clinical relapse activity, the other's confirmed progression of the disease disability, and the third is MRI activity. So, I mean, this is what we're concerned about. We don't want our patients to get any worse. And if you look at the variety of studies that have addressed whether these anti-CD20s have been effective, what you see is somewhere between 50 and 85% of patients achieve NEDA 3 with these drugs, which is far better than virtually any of the other drugs that we have available to us. And one of the things I want to draw your attention to is under ocrelizumab, you'll see the casting trial done by Patrick Vermesh. And one of the things that they did was they re-baseline the patient. And this is important, and I'm sure you're familiar with the concept of rebaselining. With any drug that we give, it doesn't work like that, except for maybe natalizumab. I mean, it takes a while for it to work, so rebaselining the patient at 6 months' time and using that as the time forward helps you, because you may see disease activity in that initial 6 months as the drug hasn't had enough time to do its thing. And when they rebaseline the patients with ocrelizumab, they found almost 75% of them achieved NEDA 3. And I'm going to show you that if you look at the ultimate 1 and 2 trials under ublituximab, without rebaselining, so starting at time 0, they found a 45% NEDA 3 rate. But if you rebaseline the patient and assess them up to 96 weeks later, so you looked at 24 to 96 weeks only, 82% of patients achieved NEDA 3. So this is very, very helpful in terms of addressing whether you're suppressing the disease or not. What about the safety information? Well, the contraindications for using these drugs are active hepatitis B. We know this from rituximab being used in patients with lymphoma. That's where the whole story of reactivation of hepatitis B came to the fore. We also are concerned of life-threatening infusion reactions with drug exposure. If you've had that before, you certainly don't want to use the drug again. And the things that we're concerned about are the infusion reactions, which are in many respects more of an annoyance than anything else, but can be life-threatening. Reactivation of hepatitis B, infections, diminished immunoglobulins, neutropenia, liver injury, PML, and then the efficacy of vaccinations. So the recommendation is complete live or live attenuated vaccines 4 weeks before you give these drugs, and complete non-live vaccines 2 weeks before the first drug. And I'm going to talk about each and every one of these. So infusion-related reactions occur in about 30% or more of patients with their first infusion, regardless of which drug we're talking about. But it decreases in frequency with repeated dosing. With ocrelizumab, we see that about 4% of them will have a grade 3, but it's quite rare to see a grade 4 reaction. With certain of these drugs, the infusion reactions are very low on repeated dosing. And so with ublituximab, because of that glycoengineering and the fact that we are not activating the complement system, repeated infusions see this progressive decline in infusion reactions where it quite infrequently seen. The respiratory infections are the most common infections we see with the use of anti-CD20s. Urinary tract infections are number 2. The highest rates are seen with rituximab, and there's a 1.7-fold increase relative to interferon and glatiramer acetate. Now, this is a study that I found in JAMA Neurology from 2020, where they looked at infections in MS patients, but they didn't look at all the anti-CD20s. They only looked at rituximab, where the risk of infection is highest. One of the things that I want to draw your attention to is, if you look at the pink at the top of the slide, that's the general population. That's the rate of infection over a 6-year period. And even with drugs like the interferons and glatiramer acetate, we see a higher rate of infections in the population than in the general population at large. So that shouldn't alarm you. But it does look like with the anti-CD20, at least with rituximab, the risk is somewhat higher. Some of the complications include hypogammaglobulinemia, lymphopenia, both of which may increase risk of infection, PML, which remains fairly rare. So There was concern about PML raised about the anti-CD20s for many years, but in large measure, we were using rituximab in patients that had a predisposing cause to PML. They had underlying leukemia or lymphoma, and even in the absence of rituximab, those people were at risk for developing progressive multifocal leukoencephalopathy. The same is true of people with connective tissue disorders. So we've been using these anti-CD20s in patients with various forms of polyangiitis, rheumatoid arthritis. These are also diseases that, of and by themselves, there's a risk of PML. However, for MS, there is no increased risk of PML. So when you see PML appearing in the MS patient, that's alarming. And that, by the way, is true of ulcerative colitis and Crohn's disease. Never previously been reported with those diseases. So it turns out that the risk is several orders of magnitude below the risk with natalizumab. We're talking on the order of about maybe 1 in 20 to 1 in 25,000. Very infrequently reported, but observed. Hepatitis B activation can be seen. Of course, we test patients before we start it. The decreased vaccine response I talked about. And patients can develop neutropenia. The estimated incidence is about 0.5, 0.6 per 100 patient years. It occurs usually between 4 weeks and 6 months out, although I've seen it longer, further out than that. Some patients had neutrophil counts of 0, which can be quite alarming. And about 60% of those individuals that developed neutropenia also developed infection, and as many as a half required hospitalization. But a third are asymptomatic. I will tell you that I've had a few in the, before I retired. Actually, I still see a couple patients, one of whom that I see developed neutropenia in the face of generic rituximab. The reason I use generic rituximab on this patient is he lives overseas, and I just had started him on rituximab because other drugs weren't available to him at the time. And he developed neutropenia, and I put him back on the drug, and he's fine. So sometimes you could restart it, but a third of the patients, it recurs in. Hypogammaglobulinemia develops in at least 10 to 12% with long-term use, and the highest risk, again, is with rituximab. Ocrelizumab is associated with decreased IgG of about 0.38 grams per liter per year, so it's a progressive decline seen with it. Ofatumumab doesn't seem to have the same effect on IgG levels. They appear to be more stable. And of the immunoglobulins, IgM appears to be the most commonly affected, more so than IgG. IgAs tend to remain stable with all of them. And it is an independent risk factor for infections, as I mentioned earlier. Now, in terms of the effects on of the various anti-CD20s on the immunoglobulins, what I can tell you is that generally there's this decline, but it doesn't get to the lowest limit of normal. In other words, a patient will start up here, they'll decline, but it doesn't make it past the threshold of the lower limit of normal, at least as has been demonstrated in the 5-year data with ublituximab for both IgM and for IgG. That's not to say they don't have a reduction, but is the reduction one that you need to be concerned about? There are predictors of serious infection. I mentioned hypogammaglobulinemia, lymphopenia, neutropenia. These are the others: over 50, being white, being a female, having comorbidities, having progressive disease, having lower IgG and IgA levels below the baseline, and having prior DMT use. Those seem to be the risk factors. What about anti-CD20s in pregnancy and breastfeeding? I will tell you that it was the young woman who would come in my office when we learned about the value of rituximab that really got me interested in using rituximab. And I started using it before ocrelizumab in that population. I thought it was brilliant to do so. And the reason is you give the drug if you tell the patient to wait. Now, the half-life of these anti-CD20s is 18 to 32 days. So if they wait 3 months, The drug's gone. That's 5 half-lives. So there's no risk to the developing fetus. But additionally, the placenta serves as a barrier. And we've learned from people like Riley Beauvais that even when you administer this drug during the course of pregnancy, which is often done accidentally, it doesn't really have a significant effect on the developing fetus. There's no teratogenicity that's been reported with it. So it's advantageous. It's safe to administer these drugs 3 to 6 months before conception. It does lower maternal relapse rates because the drug has this effect that lasts for a long period of time. And that's great. Again, no evidence of teratogenicity, and even exposure in the 2nd and 3rd trimester, which may result in transient neonatal B-cell depletion. The key word here is that it's transient. The B-cells come back in the infants. And with respect to breastfeeding, there's very little that gets across into the breast milk, and there's been no effect on anti-CD20 levels, on anti-CD19 levels, the ones we measure. Now, one of the things that Fred alluded to earlier was, well, we're not going to see head-to-head comparisons of these anti-CD20s. And I don't think we ever will in the MS population. On the other hand, the oncologists have some data on this. And this comes from a study in which rituximab was compared to obinutuzumab. Obinutuzumab is a glycoengineered form of rituximab. And they were interested in developing a drug. Genentech was interested in developing a drug that could be used in individuals that had what was called rituximab-resistant disease. Either it was rituximab-resistant CLL or lymphoma. And they created this drug called obinutuzumab to increase the affinity of the antibody to natural killer cells and drive the killing of the B cells by the ADCC as opposed to the CDCC. So they did a head-to-head trial of these two drugs, which were essentially the same, one being glycoengineered, the other not. And what they found was that survival rates in CLL and follicular lymphoma were actually better with the glycoengineered version. But there was no advantage for another form of tumor called diffuse large B-cell lymphoma. And while the spectrum of toxicities was the same, the adverse events were slightly higher with obinutuzumab. What do we screen for in individuals that were starting on anti-CD20? CBC for sure, liver function studies, hepatitis B, depending on your patient, maybe HIV, quantiferon gold, VZV, IgG suggested. And we do a quantitative serum immunoglobulin. We used to repeat it at 6-month intervals, but that's a waste of money. And I'm going to bring you back to the beginning of the talk, which was before the early 1990s, we had no treatment. 1948, at the Association for Research in Nervous and Mental Diseases, there was a conversation between Robert Foster Kennedy and Houston Merritt that was recorded. Robert Foster Kennedy, both of these people, by the way, renowned neurologists with diseases named after them. Kennedy said, one should no more tell our patients that they have multiple sclerosis than we should tell them they have inoperable cancer. When I have to make the diagnosis of multiple sclerosis, I make it to the relations, not to the patient, because once the name is heard, it is vested with lamentable results. And Houston Merritt stood up at this conference and said, I agree with Dr. Foster Kennedy, that the diagnosis should never be told to the patient unless it is absolutely essential to that patient for the arrangement of their life. We've come a long way, haven't we, baby? And I'll leave it at that and be happy to take any questions. Yeah, low IgG. All right, so I have a difference of opinion than my colleagues from Canada, who I see in the audience. In individuals that have infection, certainly it makes sense if they're hypogammaglobulinemic to give them immunoglobulin. But in the absence of infection, I don't think it really makes sense to do that. And we haven't been doing that. But I think, Mark, I think I heard you once say that in your individuals, if they're hypogammaglobulinemic, even in the absence of infection, you treat them.
If it's persistent, then, and especially if it's an older patient, then I will. But in Canada, IVIG is free. It's a blood product, and therefore, there's no cost to the patient.
So many reasons to move.
One more.
Second question is, what is the repletion time in neutropenia?
I don't really know the answer to that. I presume it's in the course of a few weeks or months, a couple months at most. I could tell you the patients that I've seen with the neutropenia, when I've repeated within 3 months, their neutrophil counts have returned. But I don't know what the studies actually show. Maybe somebody else does.
All right, thank you, Joe.
Sure.
Okay, for our last talk of this session, we're going to talk about CD40's role in treatment of multiple sclerosis. This activity is supported by an independent education grant from Sanofi. And so you heard Joe Berger talk about the importance of B cells. I'm going to take you back to T cells and what is an exciting and new pathophysiology for us to look at. So here are some of the evolving concepts in multiple sclerosis, and that is, we've talked about T-cell mediated disease, and Joe did a really nice job of showing you how this evolved. You should know that he didn't mention this, but the human form of EAE was actually discovered by Pasteur accidentally when he was immunizing people for rabies, because it was done, the rabies were grown in rabbit brain. So anyway, T-cell mediated comes from EAE, but it also comes from analysis of tissues, and we've learned that this is primarily subsets of T cells, being Th1 and Th17, which are helper T cells. They're CD4 positive helper T cells, and under normal circumstances, they help you fight off infections. In autoimmunity, they help themselves to one of your tissues, and so that's the pathogenic part. And so then we have the idea of CNS resident innate cells. We've gotten very interested in microglia as well as astrocytes and what's going on within the central nervous system, which is part of the innate immune system. And then moving forward, we're looking to see how we can put this all together. And so let's go through that. So MS is a disease with multiple biologies. There's much that we know about it. There's much that we don't know about it. Part of the limitation is our limited access to the tissue. So dermatologists, rheumatologists, and GI people have ready access to the target tissue. And we don't, because we tend to not want to biopsy our patients. But we have two aspects of this. One is the acute neuroinflammation that is associated with the relapse and with the gadolinium-enhancing lesion. And then we have the more chronic neuroinflammation that characterizes what we think is progressive disease that involves inflammation within the central nervous system. So the acute neuroinflammation we think is part of the adaptive immune system, okay, which involves T cells and, as you heard, B cells crossing the blood-brain barrier, enhancement there, and attacking tissue within the central nervous system. The innate part comes from more chronic things, and we've seen this with some of the MRI findings of Pearl's phase rim lesions, slowly expanding lesions, chronic lesions within the central nervous system that we think involve microglia to some degree, and maybe a major degree, and has become an important target that we think about. And then we have this chronic neuroinflammation, which involves these players that you can see, which eventually microglia, astrocytes, plasma cells that are available, but we also have the infiltrating T cells and B cells that are present there. And so this is an important target for us, which we're just starting to address. We've addressed it a bit with the BTK inhibitors, and I told you earlier that at least one of them had a successful trial in non-relapsing secondary progressive MS, and we think that's because at least some of the BTK inhibitors can get into the central nervous system in reasonable pharmacologic numbers. And so we have chronic compartmentalization as part of the progression of MS. And part of this is inflammatory, part of it is neurodegenerative, and the link between the two is not entirely clear. There is a pathway where one could see chronic inflammation causing persistent damage, but there's not a one-to-one link yet that says all of the damage that occurs that's leading to gradual progressive disease is inflammatory, and some of it may be independent neurodegeneration, but still the possibility that it's being kicked off by this inflammation within the central nervous system, and if we could turn that off, then we can halt, hopefully, the idea of progressive disease, which is really our major therapeutic goal right now in MS, is to deal with the chronic inflammation and the progressive disease that's there. And so there are a lot of players within the parenchyma that are involved in that. And then this is a relatively new idea that's come up, and that's the idea of central nervous system border tissues regulating the chronic neuroinflammation. And the border tissues involve the meninges, and they involve the choroid plexus, and things that are present within the central nervous system, and how they may be impacting the underlying tissue. And so this is, again, an idea of things that are coming into the central nervous system, either in the meninges, in the spinal fluid, or coming out from the choroid plexus. And this is an area of active investigation at the present time. The idea that there are gradients that suggest soluble factors coming from the meninges, from the choroid plexus, that are infiltrating into the brain parenchyma and affecting inflammation there and affecting underlying tissue damage. And so a new molecule, the new mechanism is frexalimab, which is a second-generation anti-CD40 ligand monoclonal antibody. And I'm going to show you, this is the picture of the monoclonal antibody. It's also FC engineered to overcome what were seen as thromboembolic events with the first generation of this drug. And so it's been changed a little to do that. And this is a mechanism that I don't think was a Joe's slide of failed T-cell studies in the past, but it involves co-stimulation, inhibition of co-stimulation. So I hope this is clear. In the middle panel, when you have a T-cell, the T-cell recognizes antigen that's presented by an antigen, and I hope this isn't too much neuroimmunology for you, but it's a new mechanism, so I do want to review it a little bit. So T cells recognize antigen complexed with HLA, okay, and that's what sets them off. But in addition, there are co-stimulation molecules that also have to be engaged. CD40 ligand and CD40 are one of the co-stimulation. There are others. This is the opposite of checkpoint inhibition, because that's an anti-co-stimulation that the body has itself. So here, if you block the co-stimulation, you're inhibiting the ability of the T cell to be activated. Activated T cells are part of the cascade of events that causes the damage in multiple sclerosis. So co-stimulation is important. In the past, co-stimulation had not been a very successful approach, but now, in fact, we see evidence that it is. What's even more important is that this co-stimulation effect, CD40 ligand, is not only involved with adaptive immunity, but also in the innate immune things. And so here on the left, you see how a B cell is activated with, and a T cell interacts, involves co-stimulation there, CD40 ligand. But there's also activation of macrophages and microglia. Macrophages and microglia are important aspects within the central nervous system for the innate immune response that we think is driving at least a major part of the chronic inflammation that occurs in multiple sclerosis. So also CD40 ligand is a key regulator of lymphoid neogenesis and the formation of meningeal lymphoid follicles. So this has been something that's been discovered in MS, that these follicles are there, and there's thought that they may be playing a role in causing the damage underneath it to the cortex of the brain. So that's the immunology, and now we're going to take a look at what happens with this. And so there's some biomarkers of both T cell and B cell activity that decline, and this is results from a phase 2 trial, and I'll show you the data from the phase 2 trial, but just to show you that blocking the CD40 ligand binding has effects both on T cell activity and B cell activity. And the same thing here could be seen with decreased innate immune cell markers. In this case, it's CCL19 and IL-12 go down. And so there's potential for multiple ways that this impacts the immune system to cause a therapeutic effect. And this one I'm not going to deal with. Myeloid cell, again, macrophages and microglia are reduced over time. So let's take a look at what were the data for this. So this was a phase 2 trial of CD40 ligand monoclonal antibody to frexalimab. And in this study, we looked at both frexalimab intravenously and subcutaneously versus placebo in a fairly typical study for phase 2 where you're reducing the number of gadolinium-enhancing events or acute events. And you saw this, Joe Berger showed you very nicely how this worked in studies of anti-CD20. But this was the design of the study over time. And what we see is a dramatic effect on the gadolinium-enhancing lesions or the focal inflammation here in these groups. And so you understand this. What happened here is patients were treated for 12 weeks with either frexalimab or placebo intravenously, frexalimab or placebo subcutaneously. And then after 12 weeks, everyone's rolled on to active drug, either intravenously or subcutaneously, given every 4 weeks. And so what you see here is the effects. Top left is the rate of GAD-positive lesions at baseline versus week 12 in the placebo. And then once they roll over to active drug, you see a dramatic decrease. Let's see which would be better here. Nope, I can't reach that one. So a dramatic effect here. This is frexalimab from start to finish, and you see there's also a marked decrease after baseline to this group here going out. And then you see the same thing here. So a very dramatic effect in phase 2. This phase 2 success leads to phase 3, which we'll discuss. The annualized relapse rate in those patients followed over time remained quite low, at least out through 2.5 years. And EDSS score, which is hard to assess over short periods of time, in these phase 2 studies, but nevertheless remained stable. So you didn't see any trending away from a therapeutic effect. NFL levels declined over 2.5 years, again indicate reduction in neuroaxonal damage and really a reduction in inflammation, because that's one of the things that we've seen with NFL in most of our studies. And lymphocyte counts, importantly, remained stable over 2.5 years. So in this phase 2, the tolerability was good. There was a high study completion rate of 97%. The retention rate in the open label trial was also good, 79%. The major complication was COVID-19, because that's when this study was being carried out. One also saw rare cases of liver changes, pulmonary embolism. That was considered unrelated. I'm not sure how that was determined, but that's of interest. But all of this together suggested that it was worth going into a phase 3 program. And these are the most common treatment emergent events: COVID-19, which we don't think was associated; nasopharyngitis, which is the commonest adverse event in every MS clinical trial; headache and back pain. The serious events are listed there, one each. So there was no clear pattern of other serious adverse events in these studies. So the clinical trial program, so there's the Frexalt phase 3 trial of frexalimab in relapsing multiple sclerosis. This is underway now. There's 2 trials, Frexalt 1 and Frexalt 2, which is comparing this drug to teriflunomide. And you saw that this was a comparator both for ofatumumab and ublituximab. And actually was a comparator also for the BTK studies that we've seen to date, which is kind of interesting because in 2 of the 3 relapsing remitting BTK studies we've seen, 2 of them were not superior to teriflunomide, which may mean that people are using a better grade of teriflunomide than in the past. In any event, these are going to go against that. They're using the IV Q4-week portion, 2-year study, looking at relapse rate, and then as the primary outcome, and of course the secondary outcome, will be time to confirm disability, composite confirmed disability worsening. Composite confirmed disability worsening means a worsening of the EDSS, the typical EDS worsening of 1 point and such, or half a point if you're up at higher EDSS levels. The composite also adds the 9-hole peg test and the time 25-foot walk, so it's a broader assessment, and we think maybe more important. And then the usual MRI metrics, number of T2 lesions, GAD lesions, brain volume, and things of that sort. And of course, they'll be testing for safety. So this is a relapsing remitting population with a typical number of relapses in the prior 2 years and the prior 1 year. And so those 2 studies are underway. And then FreeViva is a phase 3 trial of frexalimab in non-relapsing secondary progressive MS. So unfortunately, a prior group at the FDA got interested in MS as a non-relapsing disease rather than a non-active disease. And the difference is that non-relapsing only looks at relapses, whereas non-active also looks at new MRI activity, either gadolinium enhancing lesions or new or clearly enlarging T2 lesions. So this study was set up during that time period, and so it's still looking at non-relapsing, similar to the population that was tested with tolebrutinib. And we talked about that it has a positive sun. So previous diagnosis, secondary progressive MS, no relapses in the prior 2 years. And so that's a definition of non-relapsing secondary progressive MS. And then followed over time. And the primary outcome here is the increase only in EDSS, not the composite EDSS, but EDSS alone, or, and this is an or a 20% increase from baseline in 9-hole peg test or time 25-foot walk. So this is an interesting composite because you can do either or and when you do the composites here. And means you have to hit both. And or means you can either do it off of the EDSS or do it off of the 20% change. So it makes it a little easier to make your outcome, but it's a valid outcome measure. And then they'll look at the usual secondary things, looking at the full combined composite and change in brain volume. And what's interesting is that brain volume has become rather problematic as a marker of progressive MS. So there was thought that during some of the phase 2 studies that change in whole brain volume would be a good predictor of clinical success in secondary progressive MS, but this hasn't held up in phase 3. And so there's a degree of confusion in our community of what to do about brain volume, but it is a bit of a concern. And then there's Frexite, which is an IV sub-Q bridging study to see if you can switch from IV to sub-Q over time, which would be more convenient. And so that one's looking at sub-Q all along versus IV for a period of time, then switching to sub-Q. So frexalimab has demonstrated sustained reduction in disease activity over 3 years, low numbers of gadolinium-enhancing lesions, again, in the usual 3-month study, but also in the extension. The clinical endpoints remain stable over 3 years, and 86% of the patients remain relapse-free with stable EDSS scores. It was well tolerated, no significant emerging safety signals, and lymphocyte counts were stable. And the phase 3 trials are underway, and I don't know the date when they will be completed, but it will be sometime in the not-too-distant future. I'm actually not involved in this study. So I will bring you back to this slide here because this is a new mechanism. And so we have actually working on 2 different new mechanisms now that are coming into the clinics. One's going to be BTK, which you'll hear more about later, but the other is this co-activation blockade. So this one is completely unique. We expect, and our hope is that it involves both innate and adaptive immunity. And even though it's a monoclonal antibody and it's not going to get into the central nervous system to great degrees, we think that the altered T cells that it does produce will get into the central nervous system because T cells can get across the blood-brain barrier and do what they do. So that's the hope, and that's why it's being looked at in both relapsed and remitting disease and in progressive disease. And with that, these are my colleagues at Mount Sinai. I thank you all for your attention. And just to remind you here, scan this code to get your CME credit, and I'll stop and take any questions. And if you sent a question in on the thing there, Tim, could you hand me that? And it's not. So, questions? Okay, if not, that ends the first part of our program. We're going to take a break for how long, Nicole? What's that?
10 minutes.
5 or 10 minutes? 5 or 10 minutes. We'll take a break, and then we'll start the second part.
1, 2. Check, 1, 2. Test, 1, 2. Check, check. Nothing, nothing.
This is patient care for advanced practice providers. This is a course of forms, the advanced curriculum of multiple sclerosis that we've been teaching in other venues for quite a few years. This one is brought to you by and accredited by the CMSC. Let's see, where's my clicker here? And then, okay, so if you scan this, you could take the pretest, and it gets you registered as well. Again, you may have gotten this, write this one out ahead of time, I think, also, but I'll give you time to scan that. Okay, and this is our agenda. We have some didactic lectures, and then we'll take a break, a few more didactic lectures, and then we're going to have a panel discussion of various aspects that are practical and relevant to the care of MS patients. And this advanced curriculum is supported by an educational grant from Genentech, for which we are grateful. And this is how you get your credit statement. And this is the CMSC's credit statement. And these are the faculty disclosures in easily readable print. And as before, scan this QR code to ask questions of the faculty, but in addition, I'll take questions from the floor, which tends to work just fine in this size room. And so to kick off, we're going to have a discussion on MS pathophysiology, epidemiology, et cetera, from Scott Newsom, professor of neurology from Johns Hopkins University. Scott, welcome.
All right, well, thank you very much. I was told I have 2 hours to go through the pathophys of MS, because the reality is it would really take that much amount of time. We've learned a tremendous amount about the putative pathophysiology, and I say putative because every few years we learn more about how intricate the immune system is, and not just normal immunity, but when the immune system goes rogue, as I like to speak. So I only have really about 20 minutes, so there will be some slides that I go through very quickly. But hopefully you'll have the slides for future to reference. And so we'll just talk very briefly about MS overview, and then what are some of the trickle-downs effect that we're seeing clinically. And then we'll do a deep dive into the putative pathophysiology. We'll talk about some of the immune cell players, and then I'm not going to go into disease-modifying therapies because one of my colleagues on the faculty roster will be doing that, but I just want to set the stage for that. So I think it's important to think about history, where diseases were first described, even if it was prior to the advent of MRI and some of the sophisticated tools that we have. And I don't think we can ever go a talk, at least on pathophysiology and MS, without mentioning Charcot. And you can see these nice little illustrations that were hand-drawn here of what he saw histopathologically around areas that were normal and in areas that were damaged. And so, for example, to the right of the slide, you can see within a plaque, within a lesion, as we call it now, the destruction that can happen. And you can see how there's like room within that area, and that's because there was widespread damage, including axonal injury. As far as I know, in the 1950s is really when multiple sclerosis as a diagnosis was first seen in the English literature. So a number of years later, Bruce Trapp did some eloquent work using a confocal microscope, and you can see the images here. Now this is going back to the 90s, where he showed in a very nice way almost what was seen by Charcot, right? So you can see here axonal damage, these little bulbs, which are the bulbous ovoid axons that are transected, and then the green with the arrows that are pointing at it, that's demyelination. So really interesting, right? So Charcot drawing it out and then seeing it more eloquently many years later. And why I'm showing this is just to emphasize that early on, axonal injury occurs, and it's direct result from the immune system in various ways. Now, this is just another illustration just to show that MS is not just a white matter disease, and we know now it involves gray matter, cortical, and even substructures. And this is a nice publication that gives you sort of a visual representation of the different regions, including the visual pathway, which now many of you probably know in the 2024 McDonald criteria that we can make a diagnosis utilizing the visual pathway, anterior visual pathway specifically. So why do we care? Well, you know, we're lucky nowadays that we have highly efficacious drugs that are preventing, I think, some of the disability that we see in MS, but it's an exceedingly common condition, especially here in the United States and North America in general. It still remains, I would say, the most common cause of non-traumatic disability in people between the ages of, you know, well, this keeps changing, but up to 60. And we know from natural history studies that the majority of people who do not get treated with immunotherapy, they eventually develop disability. I'm not going to go through this slide in detail, but this is just a couple, I would say, key references that I found very interesting. And some of these studies are done prior to the advent of therapies, but just emphasizing the fact that we need to treat our patients with immunotherapy and then also treat with non-pharmacological interventions, which we are seeing benefits, including immunological benefits. So here is a busy table, but you'll hear throughout this conference that we're looking at MS a little bit differently now. We're looking at relapsing and progressive biology almost going as a continuum. And so I think there's plenty of data, even older data, that speaks to this, that, you know, progression starts from even the start. And so it's very important to recognize that. And why is it important? Well, with the advent of maybe emerging therapies, some of these emerging therapies are directly targeting the progressive biology. So this is going to be incredibly important because even with the best therapies we have available today that shut down what I would deem as all macroscopic disease activity, whether it's relapses, new lesions on MRI, there are people that continue to progress. And that may be aspects of the immune system, like the innate immune system, that we're just not getting to with our current therapies. And so there are a few different publications out there that have sort of a picture like this. I will say the one area on this figure that we know least about, at least in my mind, is the age-related progression, right? That's that sort of bottom piece of this figure in blue, which you can see it actually starts way back. So as we're in this room together, we're all aging together. Trajectory of aging may be a little bit different depending on what's going on. So this study is a recent study that I feel hits home progression starting very early. So this was a study done by Turn colleagues, which really showed after having your first clinical event, following people over time, there are people that take different trajectories with respect to their disability. And what I would recommend is reading this paper because it has a lot of good information. But the highlights are for individuals that were deemed to have this progression independent of relapse activity, there were over 20 times greater risk to go on to develop disability than those that had more relapsing biology. So really important to take note of that. And it happened within a short period of time. So there looks like there may be a window of opportunity to really help our patients. And so here, again, I'm not doing a immune therapy or disease-modifying therapy talk, but as I go through the rest of this presentation, Just think about this picture and the different immune cells, both peripheral and central, that are, leading to MS pathophysiology. And it's really, in my view, this cascade of effects that's occurring, whether you're a peripheral or central person, there's opportunity to, target and to help hopefully impact that cascade of events, which then will help our patients. So, this is a very complex interplay between the adaptive immune system, which we talk about B and T cells, the innate immune system, which I think most of you have heard, microglia, right? These are a hot topic, but also astrocytes. So there's a lot of players that contribute to MS pathophysiology. And I will say, at least from my perspective, we're at sort of the infancy of really understanding what about mitochondrial strain, oxidative stress, and how does that impact pathophysiology and the interplay of the other parts of the immune system. And then, of course, remyelination. I know a lot of people are excited about this. There's a boatload of treatment trials that are trying to identify different ways to remyelinate. But we're learning remyelination may not be possible in some individuals, depending on their age, depending on how many axons they may have within the central nervous system. So primary etiology of MS is still unknown, but I think what we've come to realize, I like this Venn diagram because it shows, in my view, like a perfect storm situation. You have maybe someone who's genetically predisposed and has been exposed to something in their environment. EBV, which I'll talk about in a minute, has come to the forefront. But other environmental triggers in combination with genetic predisposition that causes the immune system to misbehave and not be happy. There will probably be several, I think, sessions talking about individually some of the environmental triggers, so I encourage you to attend those. So EBV, by show of hands, how many of you have heard about EBV and its relationship to MS? Love it. So I don't have to go through this in detail, right? EBV is incredibly important to set the stage for someone to go along the path and deviate towards MS. But it's not the only thing, right? We know for sure that if we take blood markers of exposure to EBV, 90% of the population will have some elements of exposure or evidence of exposure, where MS, if you do check someone's blood, and this was done in a very large study that came out of Harvard, that essentially 100% of people had markers of EBV exposure at some point. And if you do the sort of odds ratio and looking at what does that mean, we're talking more many-fold risk of going on to develop MS if you are exposed to EBV. And it doesn't have to be asymptomatic EBV, like mononucleosis, just showing exposure, than any other environmental trigger. We talk about vitamin D deficiency, 2 to 3-fold greater risk going on to develop MS. This was over 20-fold risk if you had evidence of EBV exposure. So really, really important. No other virus, to my knowledge, or bacteria have been shown up to this point with that association. And it has been studied. Now you come to the question, well, how is this? Like, why does the immune system, after being exposed to EBV, go towards an MS pathophysiology? And I will say I'm not an immunologist, so that's should put that on my disclosures, so I don't do bench work. But from doing a lot of reading and trying to get an understanding of what's happening, there are a couple different ways that people's immune system can get sparked. And one is molecular mimicry, and the other is what is almost bystander, activation. And I would say for MS, it's probably likely a combination of these things. Now, it could make a difference when you're thinking about developing a treatment for EBV. If someone already has MS, that doesn't mean that you can't develop a treatment to maybe make it less severe, or maybe progression will be less. If you have someone where you think, well, before even getting exposed to EBV, right? Like in pediatrics, if there was a vaccine available in someone who is genetically predispositioned, maybe their mom or dad has MS, this would be an awesome opportunity to try to prevent MS in the future. So nonetheless, immune tolerance has to be done within each of our immune systems. And this is a busy slide, but just to say that when there's failure of this self-tolerance, that's when autoimmunity occurs. And that's autoimmunity across the board. And I like this little cartoon on the side. I think about this when I think about autoimmunity. You have, the immune cells up top where they're going to attack one of their own, right? And this is what happens. The immune system gets misdirected. So a lot of immune cell players. I'm just going to touch on a couple, but we have, the lymphocytes, the B and T cells, which are part of the adaptive immune system. And then I already mentioned microglia, but in astrocytes, and there are a few other players. But I do recommend, this is just one reference out of many, to read a little bit about the immunology. And so if we think about the relapsing biology in MS, we think about, okay, there's peripheral activation that has to occur. So within the lymph organs, blood, and then those angry activated immune cells travel into the central nervous system. However, that's not it. So there are different proteins and inflammatory markers that have to, in a sense, reactivate some of these immune cells. There's a lot of crosstalk that's occurring between the adaptive immune cells and the innate immune cells, the BT cells, in microglia as one example. And here's sort of a nice picture, not that A, B, C, D, it goes in line of this. A lot of these things are happening simultaneously, but just to give you a representation of how complicated the immune system is, especially in disease. But there is a lot of crosstalk, again, between cells, whether it's B cells talking to T cells, dendritic cells talking to microglia, and beyond. And each of these players can impact the nervous system in different ways in our patients. And again, just circling back to what I mentioned about therapies, there are opportunities directly and indirectly to impact the cascade that's occurring on this slide. And so, I'm going to quiz you with all the interleukins and cytokines at the end of this. But no, this I always have to go back to. T cells are still critically important in autoimmunity, even though, B cells have come to the forefront, microglia have come to the forefront, but T cells you need in order to help keep things homeostatic within the immune system, but also as different inflammatory markers activate certain T cells, that those end up causing sometimes direct damage to the nervous system, whether it's axons as an example, or activating other immune cells, including microglia. So you can see on this slide, depending on what inflammatory milieu that is present, that will deviate or differentiate a T cell into whether it's Th1, Th2, and beyond. So this is probably too small for you to see, but this is an old publication that I pulled out that I really got the message that, okay, cytotoxic T cells can participate in direct injury in multiple sclerosis. You can see sort of in the periphery on top, and then going all the way down into the central nervous system on the bottom, where a cytotoxic T cell that's autoreactive, that is a bad player at this moment in time, is actually directly targeting the myelin-producing cells. And so these on their own can do some of the dirty work. Now, B cells, how many of you have heard of a B cell, right? You wouldn't be here if you hadn't. So B cells are incredibly important. We know there are therapies out there that we use that target B cells directly and work incredibly well. But I do want to remind people that B cells, it's not just about, B cells are the only important player. I think personally that the B cells' most potent impact on MS is actually because over time, T cells do not get activated. And so you can see sort of in the middle of this slide, sort of the B cells being one of the main antigen-presenting cells, that you knock enough B cells out over time, many of the T cells won't get activated, and then there won't be that downstream effect from T cells. Obviously, B cells cause antibody production, which can do their own damage. And then I think one thing we're learning more about is B cells producing certain proteins and cytokines that activate directly our innate immune system, macrophages, microglia. And why is that important? Again, thinking about, are there potential roles for emerging therapies to target some of these, I guess, areas within the cascade that our current therapies are not. So progressive biology in MS, as I mentioned, progression seems to start from the beginning. So I think in an ideal world, short of a cure, in my view, in coming years, I see MS as a combination or a cocktail of interventions. So you have therapies that focus predominantly on that relapsing biology, and then you have therapies that can focus more on the progressive biology. And you can see in this little picture here that the little round area on the left of the slide, that's the chronic active lesions, and around that are the microglia. And you can see leptomeningeal disease, which is onto the right side, where you have B cells, T cells, and other cells. And so there has to be therapies that can work on the central nervous system side. And Fred had mentioned, and I think it was in your last talk, about most of our therapies do not cross the blood-brain barrier, or at least the large monoclonal antibodies. And so we need ways to get into the central nervous system so we can impact this process. And as I mentioned already, microglia, astrocytes, these are a couple references that I would highly encourage you to read. There are good players and bad players, and it depends on what the immune cell is being exposed to. And so just as one example on the microglia, you can see the red usually is bad, right? So red, not good. So this is an angry microglia that was exposed to pro-inflammatory cytokines and T cells. And then on the right is green, good. And microglia are actually really important beyond just keeping things chill in the central nervous system. But if there's myelin debris, it actually cleans up myelin debris and helps enhance endogenous remyelination. So they're really important, but we want them to be green and not red. Parametric rim lesions, this is a sort of a hot imaging topic. As many of you know, or if you don't know, you will know in a little bit. Pearls has made it into the 2024 diagnostic criteria, and so this is becoming a really important imaging finding. And also this imaging finding is essentially that microglia that's lipid-laden, or they are lipid-laden, but also iron ingested microglia, and that's why you can end up seeing that rim around. And there are some studies suggesting that the more you have, the more disability someone may have in the future. And that might be an area of intervention with certain therapies, which I'm sure you'll hear about. So I'm almost done, but non-classical antigen-presenting cells, this is something that I think came out not too long ago. Peter Calabrese at Hopkins and others, I think, showed this. And oligodendrocytes and OPCs in the right milieu can become active and actually become bad. And it's fascinating to me because you have, the myelin-producing cells that are supposed to be good, and now they turn a switch and they can actually become inflammatory and cause problems and actually activate other immune cells to enhance the cascade that we see in MS. So here is that picture again. When the immunotherapy talk is given, just think about, okay, the immunology that we went through very quickly. What are some of the targets, both direct and indirect targets, that are being used today, and where are we going in the future? Because again, I think MS is going to be sort of the cocktail of interventions that's going to be required to help, I think, the progressive biology especially. So primary etiology of MS, we don't know, but it's a perfect storm situation. It seems like someone has to be genetically predisposed, have environmental factors like EBV to act as a kindling effect to push someone towards MS. We know now it's not just about the adaptive immune system, but the innate immune system is heavily involved. And actually the innate immune system may be more heavily involved in progression and the progressive biology. We need to learn more about this though, in order to develop more targeted treatments. And, you know, I do feel like we have a couple on the horizon, but we will see what clinical trials tells us, right? That's sort of the ultimate decider. Okay, I know that was fast and a lot. That's it. Yeah, no, I think, yeah, the controversy between inside out, outside in, I don't think that's been settled yet. And that's where I think developing treatment strategies that target both is the way to go. But I agree with much of what you said.
Okay, other questions? Okay, thank you, Scott. Next, we're going to hear on the evolving topic of MS diagnosis and misdiagnosis from Dr. Obadat from the Medical College of Wisconsin.
Okay, thank you so much, Dr. Lublin, and thank you for the invitation for Academic CME. And thank you, Dr. Newsom, for making my life easier too, also for talking about the pathophysiology of MS. I'll be touching on the new updated criteria for multiple sclerosis, or what we call the 2024 revisions. And we'll try to kind of put them in context to the clinical practice, of course, but also highlight what has been changed and what is new. And then a little bit touch on possibly how can they help us with maybe reducing misdiagnosis. Some may argue maybe the other way, but we're going to touch base on both views during this presentation. So this is kind of what you see here as the objectives, and we'll discuss any questions you may have after the actual session. So the 2024 revisions of the McDonald criteria. This is the paper that came out in Lancet Neurology, published online 2024, in print 2025. But what you can see here is that there are some key updates, and we're going to really talk about each of these updates in detail in this presentation. But as a summary, optic nerve was added as the 5th anatomical site, and we're going to talk about it more. new biomarkers. We're going to be looking more at imaging. You just heard about the paramagnetic rim lesions or iron rim lesions, what you can call them. But those are actually made their way to the criteria. And we're going to talk about them and implications of that. Then we talk about unified relapsing and progressive MS as a one disease continuum. And you've seen this also in the presentation where we look at MS as a one disease versus looking at relapsing versus progressive. So that's kind of coming into the criteria and kind of talking about diagnosing MS as one entity. And then there's expanded pediatric guidance. That's very much an unmet need, but we're seeing more and more medications are going to be tested in pediatric population and hopefully get approved for our children who have multiple sclerosis. So I go back in time for my time as a medical student many years ago. And we had the professor come into the clinical rounds. I was a fifth-year medical student. I'm from Jordan. So we had a 6-year medical school curriculum. And neurology in the 5th year. And then this professor comes in, and I still have contact with him till today. But then he comes in and sit there and say, multiple sclerosis diagnosis, dissemination in space, dissemination in time. That's what's the summary. That's really what he gave us. But we're going to look a little bit about this revision that just came in. And is this changing? Is this concept that we have always, is it changing? Or is it just being added on? So that concept will still hold, but maybe there are a few things that you may not need to fulfill both of them at the same time. The goal is to diagnose MS earlier so we can treat people earlier with disease-modifying therapy. That's one of the goals, or maybe with life-altering strategies like nutrition, exercise, and other things related to lifestyle. So rationale, why do we need a new revision? And the panel kind of met and decided that, yes, there is time now for a new revision. There is enough accumulating evidence that we need to incorporate into a new revision of the criteria. But also, there is evidence, plenty of it, that came after the 2017 publication that actually told us that maybe if we add the optic nerve, we may enhance sensitivity without compromising specificity. There were multiple papers that were published, and I think the panel kind of took this into account with the 2024 revision. But then patients without a typical clinical attack, but similar MRI, those we call as radiologically isolated syndrome patients, those RIS patients have been in the literature and more knowledge is coming about them. And maybe people arguing maybe they actually do have multiple sclerosis, at least the biology of it. So the panel kind of wanted to see if it's time to allow for an MS diagnosis in people without a typical clinical syndrome. And then dissemination in time and space may not always be needed if you think that there is a strong evidence that this person has the biology of MS and you need to intervene and treat them early so you prevent accumulation of disability. The other thing is that we talked about the MS disease continuum, the Lancet paper again, that kind of showed the MS as a disease continuum. And this paper kind of paved the way for this criteria to talk about maybe we need to look at MS as one disease and maybe trying to de-emphasize a little bit of the progressive and relapsing kind of components, but that's also open for debate. But this is what the revision kind of took on. The other important thing, and this touches also on the misdiagnosis of MS, people with vascular disease risk factors, maybe smokers, high blood pressure, diabetes, people with cardiovascular disease, and those with migraine, and those older than 50. This is the population that we may be misdiagnosing with MS easily. So the new revision meant to try to tell us to be cautious with this group of people. So we have to really look for supportive evidence that this is MS. This is where the biological biomarkers like the central vein sign we're going to talk about, or this paramagnetic rim lesion comes into play. And this is where oligoclonal bands become really important, or what we call something called kappa-free light chain we're going to talk about. So this is where you actually need to add more evidence for you to diagnose MS. So this is what the revision really wants you to do, is to try to decrease this chance for misdiagnosis. So 2 fundamental principles for diagnosis of MS. MS is a global disease. So someone in the United States who develop MS and someone maybe in Jordan who develop MS, or someone in Asia develop MS, or someone in South America develop MS, right? It's a global disease. So we have to find criteria that could apply for everyone, and we do not want to remove the essence of diagnosing MS with dissemination in time and space. So we keep this for places where there's low resources and they cannot have access to the advanced imaging. But we don't want to stop advancing the field because we want it to be globally applicable. So there is this balance that really we wanted to reach. So the essential role for paraclinical tests, which are meant to improve our sensitivity and without compromising specificity, and maybe actually improve specificity in some cases. So we'll start with one at a time. So we're going to talk about the optic nerve. And as I mentioned before, there's more and more accumulating evidence that if you add the optic nerve to the diagnostic criteria of 2017, you improve sensitivity without compromising specificity. Postmortem analysis confirm optic nerve involvement in the majority of MS patients. In fact, even in my practice recently, the patient had no history whatsoever of optic neuritis, no history clinical, and we sent for an OCT and had the thinning on both optic nerves that consistent with demyelination. Had never had any visual symptoms. So that is something we see in people living with multiple sclerosis. So you can establish optic nerve involvement via orbital MRI, via OCT or optical coherence tomography, or visual evoked potentials. And how many people do VEPs in their practice? Anyone? One? Yeah, very few. Actually, we do it too in our practice, but I've learned that it is not being done routinely. It's very hard to find people who are doing it. So maybe the criteria is going to push us a little bit to do more of VEPs. So that's one of the things we could actually benefit from new revision of criteria. Also, studies shown that including optic nerve increased sensitivity, and we talked about this, and optic nerve involvement should always have no better explanation. So if someone had a ischemic optic nerve, this is something that you have to be aware of. But we have to have no better explanation. So these are the anatomical lesions. Now, locations, we have 5 instead of 4. So the classic periventricular, cortical, juxtacortical, and this came from the 2017 revision because it was juxtacortical before, but they added cortical. And then infratentorial, spinal cord, and now we have the optic nerve. And remember, you can confirm involvement by OCT, VEP, or orbital MRI. Then comes to looking at the criteria, there is modification to dissemination in space with the new revision. So classically, you have to have 2 or more areas involved to say there is dissemination in space. That still holds. It's not changed. But then in people with progressive MS, you only need dissemination in space by having 2 lesions in spinal cord, which was alluded into even in the 2017 criteria for primary progressive MS. But now if you have 2 spinal cord lesions with a progressive disease course, that is sufficient for dissemination in space. Now, if you have typical lesions in 3 or 4 areas, that improve your specificity to nearly 100%. So that's very important. If you have someone with cortical, juxtacortical, periventricular, infratentorial, and spinal cord, the chances of this person have MS are very, very, very high. So specificity is near 100%. So that helps. Now, higher lesion number and count can correlate with the relapse risk. And that's something that mentioned in the criteria here, which actually is important because people with more lesions may be at higher risk of relapses and having more activity in the future. Now, one major change here is if you have someone with only one anatomical area involved, and that's pretty new for this criteria. If only, let's say, periventricular or juxtacortical involved. Now, you have to actually find a way to find a dissemination in time. So you actually have to follow them and see maybe if another lesion pops up, even in this same area. But also you want to see if they have oligoclonal bands. And this is here where you could add the central vein sign and maybe paramagnetic rim lesion to support diagnosis. But now you can actually make a diagnosis of MS even with one anatomical area. So that's one new thing with the 2024 revision. This is a central vein sign, and you've heard about it. It comes from the anatomy. It comes from the studies on pathology of MS, where the lesions form around venules. And it does increase specificity, but there is a key paper that was published by a large multi-center group. And they found that 6 or more central vein sign is almost equivalent sensitivity to oligoclonal bands, specificity to oligoclonal bands. So now if you have someone with lower than 10 lesions total, the majority of them should have central vein sign to be qualifying as demyelinating. So that's important, but this is something that where interpretation can make it into the criteria, right? So you want to make sure that if you have, if you're using central vein sign to help your diagnosis, that those are real central vein sign. So you have to have support from radiology. You have to have advanced imaging that's able to pick those on the correct sequences. SWI here will be, and then some post-processing may be needed. But it's important that if you're going to use them and apply them, that you are actually applying the correct tools and applying the correct findings. Because the last thing you want to do is to just call it and then call central vein sign on lesion that they really don't have central vein sign just because there's a vein that is close to the lesion. So that's important to avoid. This is where some pitfalls may happen if you don't have the actual technology. So this is why those to be used in certain circumstances where you're unable to confirm the dissemination in time and space as the usual way with the regular biomarkers like oligoclonal bands. So this is important addition. The other one that you've heard about is a paramagnetic rim lesions. And those need even more technical advancement to be able to see them. So how many of you seen a paramagnetic rim lesion in your patients? I've seen one of them. One patient, okay, one few. Very few, right? How many got a report saying your patient has a paramagnetic rim lesion in the radiology report? I think mostly probably at advanced academic centers you may get this, but not in usual regular academic center like mine. We don't get that. So it's important to think about this because while this is very highly specific for multiple sclerosis, if you see one paramagnetic rim lesion, the chances that this is MS is pretty strong. There are other diseases that you could see them like NMO very rarely and Sjogren syndrome, you can see very rare paramagnetic rim lesions. But other than those, you don't see it with stroke, you don't see it with migraine, you don't see it with the common mimickers of MS. So it's important for us that this would be a really good sign to use if we have the right technology. This is why we're going to see that this may actually reduce misdiagnosis, but also potentially could increase misdiagnosis if someone is overcalling them or not calling them accurately. So this is why you need advanced and specialized techniques for this. They're not mandatory for diagnosis, but they can help you in certain times where you're unsure of the diagnosis, but you see a paramagnetic rim lesion. That could help you with the diagnosis of MS. Kappa-free light chain. Have you heard of kappa-free light chain before? Yeah, there are many papers that came out and compared it to oligoclonal bands and looked at it as a potential easier biomarker than oligoclonal bands. Because as you know, oligoclonal bands are subjective. They're actually time-consuming for someone to go in and count those bands. So it's prone for a human error too. Now, the kappa-free light chain is more quantitative, is more automated, and it's cheaper too to do, right? So it can be more accessible if we're doing it in the right way, in the right patient. So now this is being included in the new revision as a substitute, or you could alternate between it or oligoclonal bands. So you can say you can have oligoclonal bands, unique or restricted oligoclonal bands, or you can have kappa-free light chain index increase. So you can use either. Now what we've been doing in our center is we're sending both also to see if they actually congruent or not. So that's something that you're going to be seeing in future studies where people are going to be comparing the sensitivity of oligoclonal bands to kappa-free light chain in real practice. So, but if you have access to one versus the other, you can do either of them. So that's important that this is now allowed. So they're interchangeable with oligoclonal bands. Advantages, quantifiable and rater independent. Okay? Now this, radiologically isolated syndrome, you've heard about it. There is a growing evidence of it. There are multiple groups that have done this. Dr. Darin Okuda has been one of the leaders in this field too, among many others. But this RIS concept is where a patient comes to you because their MRI showed that they have MS. They got an MRI for migraine, they were involved in a car accident, and then they got an MRI, and then their reports say this is consistent with multiple sclerosis. So you get a patient, comes to you in a clinic, and now you have to say, what is your risk of developing clinical MS? This is what we used to do. We used to look at age, is there spinal cord involvement, is there oligochondral bands, and some of the patients we would treat actually with disease-modifying therapies. And there are a few studies that shown that treating RIS may reduce the chance of clinically converting into multiple sclerosis or clinically definite MS. However, the new revision of the criteria allows you to diagnose multiple sclerosis without a typical clinical syndrome if you have typical multiple sclerosis lesions on MRI that were observed. Importantly, that here you have to fulfill dissemination in space and dissemination in time. So it's more strict here than if someone has a clinical syndrome. So if someone has only one area on MRI without a clinical syndrome, you cannot still make the diagnosis of MS per the 2024 revision. But if someone has a clinical syndrome and in one area, you can with supportive evidence of other biomarkers. But with RIS, you have to prove both dissemination, space, and time, which will make sure that we're not overdiagnosing this entity. The one argument that here you do not have to have spinal cord involvement for RIS anymore to diagnose MS. In the OCUDA and others criteria for RIS, you have to actually find spinal cord lesions to justify this is a truly RIS, or was one of the main things to look for. Here, only with the brain, 2 lesions, 2 areas, you could actually diagnose RIS. So it's an important addition that allows for earlier diagnosis of MS. This is kind of summary here with you need to really positive CSF, positive select central vein sign to be able to diagnose. So it's a little more strict than if someone comes in with the clinical syndrome of MS. Finally here, looking at pediatric MS, and we're going to talk about progressive MS a little bit, but pediatric MS, the new criteria emphasize the importance of testing for MOG antibodies in kids with multiple sclerosis to make sure that you're not overdiagnosing MS. The other important things is the actual criteria applies for adult and pediatric the same way. So there is no separate criteria for pediatric, separate criteria for adults, so the same criteria apply for both. ADEM presentations were also talked about, require a second attack or new T2 lesion 90 days or more after the index attack to diagnose multiple sclerosis. Again, it's important, it's very clinically practical that we have to look into. And the pediatric population is expected to get more medications. Ocrelizumab was just approved, but they're more in the pipeline too coming for this population. And not much changes on the primary progressive MS compared to 2017, except that we look at MS as one disease here versus, progressive versus relapsing, the update was. But still, you can diagnose with the 2 spinal cord lesions and a 12-month objective progression that you can see with the patient telling you the story of their progression. Same diagnostic criteria now apply for PPMS and RMS, which is important. And then finally, in those with 50 and older, very important that we look for evidence for inflammation because you don't want to overdiagnose MS here. So enhancing lesions, CSF oligochondral bands, or kappa-free light change increase likelihood of multiple sclerosis, and then really strongly recommending additional testing, including looking for spinal cord lesions, again, CSF band, or looking for central vein sign. And this is the final part, is just this one slide. We talked about misdiagnosis, but remember there's implementation challenge. If you implement this criteria the wrong way, the risk of misdiagnosis can go up. If you implement the criteria the correct way, actually you could reduce misdiagnosis by looking at these biomarkers in imaging. So this revision could actually move the field forward by allowing radiologists to look more into paramagnetic rim lesions, central vein sign, and maybe automated AI, helped MRI in clinical practice. So this is an important thing that could move the field forward. If you look in the literature, you're going to see some concerns remain about RIS patients and whether truly we should diagnose them with MS or not. There's a debate there, although the revision say, if you find those evidence of biology, what is looking on MRI and CSF, you need to diagnose them with MS, so maybe you can treat them earlier. but the treatment still a shared decision between the patient and the treating clinician. And remember, it's unlikely to replace the classic dissemination in time and space. These revisions are just to add on in cases where you don't have the actual luxury of seeing the dissemination in time and space to help your diagnosis. So in summary, adding optic nerve, still with dissemination in space is there, but is not mandatory. Dissemination in time is still helpful, but is not mandatory again. Central vein sign can diagnose MS in specific situation. Paramagnetic rim lesion, remember kappa-free light, interchangeable with electron bands, and then RIS and non-specific presentation can now be diagnosed as multiple sclerosis. Pediatric and adults, same criteria. Progressive and relapsing MS, same criteria. And then people with 50 or older or vascular risk factors or migraine, be very careful. Ask yourself twice and 3 times and 4 times before you diagnose them with MS. Make sure you have sufficient evidence to diagnose them with MS. And thank you so much.
Okay, questions, Dr. Badat? That was a lot, but really well done. Thank you.
Thank you.
Question, right? Okay.
Based on the 2024 criteria, and correct me, Dr. Lublin, I think it does meet the criteria. You have two dissemination in space, and then you have the CSF restricted bands, which is dissemination in time. So they fit the criteria. Yeah. Yes? Still have to rule out mimickers, of course, NMO, MOG, and everything else, but and Sjogren and all of the other stuff. But if you apply the criteria, they meet it.
Diagnosing doesn't necessarily mean treating.
Exactly, yeah. So you could watch this patient, right? Who knows? Yeah.
Other questions? Thank you very much.
Thank you.
Okay, so we're going to move from diagnosis to clinical course of multiple sclerosis, which is moving along. So first, be very clear. There are only 2 ways you get worse in multiple sclerosis. Clinically, it's residual from a relapse, or it's progressive disease. And then we have to ask, is worsening from a relapse different than gradual progression? Well, not so clear. So it's very clear when you have a relapse. That's the reason why it's the primary outcome measure in all of these phase 3 clinical trials, whereas progression is a little hard to see. But then there's this issue of solitary sclerosis that the Mayo Clinic people have described with a single lesion leading to progressive disease. Pathologically, they're similar. There's demyelination, there's neuronal and axonal loss. And immunologically, we've talked about the difference between adaptive immune system and innate immune system. They're probably both involved in different ways, and so it's a simplification to do that. So the clinical phenotypes that we've been employing forever, we codified these in 1996, but they've been present long before that, were relapsing MS, secondary progressive MS, which was a consequence of relapsing disease that then developed gradual progression, and primary progressive MS. And these were the relapsing forms of MS, and what happens with a relapse, you may or may not recover afterwards, and if you don't recover, that's raw, right? Relapse-associated worsening. Now, the determination of raw depends on how hard you look. and whether you look things like OCT and VEPs and cognitive changes and such like that, but about 50% of attacks leave some sort of residual disability, which is why it's so important to stop relapses. For progressive forms of the disease, you have secondary progressive MS, which occurs in some patients some point after relapsing remitting disease. And it's worsening that's independent of the relapses. Sometimes it's hard to tell that apart, and it also takes time to be able to be sure that someone is actually gradually progressing. And again, this depends on how hard you look. Primary progressive disease is progressive from onset. It's in some ways easier to diagnose, but it's also the form of diagnosis that's most often incorrect in both directions. That is, thinking something else is primary progressive MS, and missing primary progressive MS, thinking it's something else. So here are some of the models that we have used this is from a 2013 adaptation, and this has served us well. In 2013, we added on these definitions for active disease, and the reason we did that was we wanted to put some dynamic analysis on someone when they came to see you in clinic or in consultation. And that is, what was their disease doing at the point at which you saw them? And whether it was relapsing remitting or progressive disease, were they active? Active meaning had a relapse or new MRI activity. And what was critical here, that it had to be framed in time, because otherwise it had no meaning. And so you could pick whatever time frame you want. Over the past 6 months, over the past year, they were active, not active. And that could be done either by clinical or by imaging. And the same thing for progressive disease, not so much, they could be active or not active, but are they progressing? So progression is not inexorable. And I would tell you, since this is a little bit controversial, it's not inevitable. And I'll come back to that. But it can start very early, as was mentioned earlier by Scott. And this is one of the things that's brought around this concept of PIRA, the idea that in some patients, progression's very early and we just weren't recognizing it. So progressive disease is characterized clinically. There is no imaging correlate to progressive disease. There are imaging correlates to worsen disease, like pearls, right? They are an indicator that someone's going to do worse. It doesn't tell you that it's necessarily progressive disease, but they're going to do worse. And so someone with progressive disease could be active and progressing, active but not progressing, not active and progressing, or not active and not progressing, meaning they're stable. And this happens to a percentage of individuals. One of the problems with the current labeling of drugs was that the prior FDA group, who did two things that have caused us trouble. This is the first, I'll get to the second. By defining secondary progressive patients with activity and saying they could be treated with our usual relapsing remitting drugs. But they didn't define what they meant by active secondary progressive MS. And if you don't time frame it, then every secondary progressive MS is active. Now this is good for you, because when you want to prescribe one of the relapsing remitting drugs for your secondary progressive patients, you can honestly put down that they had active secondary progressive MS, because you can't get to secondary progressive MS unless you had something. You had to have a relapse and such. So that they made easier, just incorrect. So the other is this idea of disability worsening across MS phenotypes. And so you will see varying percentages of patients with PIRA. And the reason why you'll see varying percentages is two. One is it depends on the population that's studied. So if you study a nascent population, which is what I'm showing here on the bottom left, which is from clinical trials, it's actually a very low percentage. If you study patients who have been treated with an anti-CD20, then the greatest percentage of the worsening you're going to see is PIRA, not raw. The other thing is PIRA is definitional, whether it's confirmed or sustained, how hard you look, how long you wait after a relapse before you call it PIRA. So this is some work we took from a database that the folks at Novartis took 27,000 to 35,000 patients from clinical trials and observational trials, and they gave them to Oxford University's Big Data Institute for analyses. And so we analyzed here. And what you see in the bottom left is from phase 3 clinical trials in relapsing remitting MS, you can see the percentage of individuals in red who actually had a relapse. And then the very bottom there, the sort of brown and green are the patients with RAW and PIRA. And the percentages are about the same. And it's a low percentage because confirmed disability worsening using EDSS is not a frequent occurrence in our usual phase 3 trials. Now, when you get to secondary progressive MS and primary progressive MS, that green bar becomes more dominant. That's the progression independent of relapse activity because that's what makes progressive disease. And so this is thinking of MS as one disease. And so you can go from RIS to clinically isolated syndrome to relapsing remitting to secondary progressive, but you also can go from radiologic isolated syndrome to primary progressive MS. And the percentage that does that is the same as the percentage of primary progressive MS in the community. So it kind of unifies that course. And this is the idea of one MS. That's starting out as either MRI onset, which would be RIS, attack onset, which would be relapsing remitting, or progressive onset would be primary progressive disease. Now, as we think about this further, we're actually looking to the left. So to the far right is clinical MS. Now we're starting to have things about the prodrome, the MS prodrome, which we're doing studies on, and that's what patients with MS look like before they declare their MS. And it's non-specific type symptoms, increased hospitalization, increased doctor visits, increased infections, increased psychiatric issues and such. But even before that, there's a biological onset to the disease. And then to the far left are individuals who are at susceptibility and at risk. And you heard earlier that this is a combined genetic and environmental disorder. And so that's what's setting the stage. The EBV is an interesting part of the environment. I'm old enough to have been through dozens of other viruses that have declared themselves as the cause of MS, so I'm not quite as excited about EBV. I'll take Scott Newsom's view that it may be necessary, but not sufficient. But more to come on that. Like I said, there's a long list of viruses. And then there's my colleague Stephen Krieger, who looks at progressive disease as an unmasking of prior focal activity as the brain loses capacity and loses reserve, which happens every time you get a lesion. And so this we already talked about. So we talked about RAW and we talked about PIRA. And those are the two ways you get worse. Distinguishing them can be somewhat difficult, and it may not matter, except that we're probably better at dealing with RAW than we are with PIRA. And so that gives us the challenge of trying to treat this progressive disorder, which, as I said, in many patients starts very early. So recognizing it is difficult, but may be important, especially as we develop treatments, hopefully treatments that could treat both relapsing and progressive aspects of the disease. Now, my friends in Europe have tried to come up with a harmonized definition of PIRA, and it is a definition. And all of PIRA is definitional. It's not biological. And again, it depends on how hard you look, whether you look to see whether it's confirmed or it's sustained. Confirmed means that you have a change, say in EDSS, that you see after 3 months or 6 months. Sustained means that that EDSS never improves, and that's not always the case. And so that's irreversible PIRA, that's sustained. But there is a reversible PIRA, people who have worsening over several months and then improved. And it's not clear to me what that's due to. It could be due to repair mechanisms, which would be interesting biology. It could be due to EDSS error, and that's common and that's worrisome. And it may be related to some of these other things like pearls and slowly expanding lesions, which can resolve, although the resolution period is longer than what we're seeing for reversible PIRA. Oh, I didn't mean to do that. Okay. And so this I talked about a bit. I showed you this part here. So what we're trying to do now is come up with this one MS concept and think about MS as a dynamic approach which maybe have phases and states. So relapsing phase, a progressive phase, a stable phase, repair phase, inflammatory, whether it be adaptive, innate, or maybe both, and maybe a degenerative phase. And so when we looked at this Novartis database, we noticed several things. One is disability accumulation was affected by prior relapses. So a relapse in the year prior to a disability event increased the risk of disability by 31 to 48%. PIRA may start early and becomes the dominant driver when you get to progressive MS. The principal risk factors for further disability accumulation are prior disability and older age, and neither one of those helps us very much. And this is some work done by my colleague, Dr. Satya Narayan, who again looks at things definitionally. And so she's looking at PIRA and PIA, progression independent of activity. So the very top part of these bars is PIA, which is a smaller number than PIRA, just never say. And the reason I show this is because these three bars show different definitions of a relapse. So the one to the left with a very strict definition that we used in CombiRx. The middle one was a less stringent definition. And the one to the far right was part of the clinical trial where the investigator said it's a relapse, even if they didn't meet the criteria for relapse, like a change in EDSS. And every time you do that, you double and then triple the relapse rate, depending on your definition. And this is why comparing across trials, comparing PIRA definitions and such, becomes so difficult, because the more stringent your definition of the relapse, the more likely you're to find PIRA, and just the opposite. The less stringent your definition of a relapse, then you'll have less PIRA. And so the question is, how long does worsening after a relapse take to resolve? And this comes from Marcus Koch's work from Canada, looking at the CombiRx, showing you that it could take up to a year for people to completely stop worsening from a relapse. And so that has an effect. And then this idea, is all MS progressive? My view is no. And so I have patients with hair grayer than mine, older, hard to believe, than me, who look just great. Now, could they have lost a couple of IQ points along the way? Well, maybe, because I can't assess that. But they're not progressive disease. And I think this is an important concept, that the difference between MS and every other neurodegenerative disease is that MS is variable, not necessarily predictable, as opposed to all the other neurodegenerative diseases, which are predictably bad. And so we talked about this a bit. So the new visualization of these courses, again, is over time. We have a paper in Nature Reviews, which it's been accepted, but now we have to wait for the editor to screw it up, outlining how we're going to bridge where we are now to where we want to be. Where we want to be is a biological definition. So when we did these clinical course definitions, they're what you see in the consultation suite when you're looking at a patient. They're going to match one of those clinical courses, okay? But that's not biology, that's just clinical. And what we want to do is develop a biological definition of the disease, because then we can more directly treat people and more appropriately treat them for their underlying biology, whether it be T-cell mediated, B-cell mediated, neither of those directed innate immunity and such. And so that's one of the things we're thinking about as we move along. And so this, thinking of MS as a spectrum. And I was very pleased with myself that I actually found this slide and made it myself. I'm not very good at making slides, as you could tell from the other ones, but I liked this one. And so there have been some concepts produced. I'll talk a little bit more about the one on the top left there. I talked about the bottom right, that's Stephen Krieger's. I don't talk about it. So the top one is an AI-driven study looking at states of MS. So they took that Novartis data set and they threw it into an AI black box and said, tell us what's different. Give us groups of patients. And the black box put out those, 8 little circles there. The blue was mild, early evolving disease. The red was more severe disease. And numbers 4 and 5 4 was MRI activity, 5 was a relapse. And what the AI said was to get from milder disease to more severe disease, you had to go through an inflammatory phase. So we hope to see this confirmed in other ways, but it's really very important saying that you have to turn off relapse activity. And if you don't, then you're going to be left with more progressive disease. So it's kind of an exciting result, and it comes up with these additional states that individuals can be in. So that's where we're going, and we're not ready for a biological definition yet. We've got papers on that. We have this transition paper which is going to say how we can better define what a patient looks like in time in multimodal ways to give a dynamic framework for the patient as you see them in your clinic on this particular day, until we can get to the understanding of MS we need for a biological definition. Now, I promised you something about the new diagnostic codes. This is an unmitigated disaster. So this comes from the FDA, asked the CDC to change the codes. The MS Society got wind of this, not that they were notified, but they got wind, and we hurried up and we had phone calls with the CDC, and they promised us they wouldn't do it, and then they did. And so you're left with these different codes. So this is a gift to the insurance industry, not that they needed another gift. And it serves no good purpose. So for breaking down MS into active, non-active, because it's the same mistake. It doesn't define what they are. So it's whatever you want to call it. But for your purposes, be very careful, because the insurance companies are going to be looking at what you call it. And if you just call it MS, which is one of the codes here, not even the unspecified, there was one, just G35, you're going to get dinged on the next prescription. So you have to actually match it to what you're treating. And I think it served no good purpose, and it's unfortunate, but it's just another thing you need to be careful of when you do that. So with that, I thank you all for your attention. So, questions? I don't know if there was any. If you put it in the thing there, I can't see it anyway. So, happy to take any. Hearing none, we'll maybe get caught up a little bit. So next up is going to be Carrie Hirsch, who's going to give us, from the Cleveland Clinic, give us a therapeutic update.
Well, hello. How are you all doing? I think I'm keeping you from your break. But don't worry, I'm only going to be giving a 2-hour talk on disease. No. I have, however, been given the lofty goal of presenting a therapeutic update for MS in just 30 minutes. So you're going to see that the way that I designed the presentation is that I provided slides that have very specific information on real-world disease-modifying therapy approaches that you can take home and use in your clinical practice, while I will be speaking on overarching themes and goals around DMT considerations. So you guys are with me? You ready? All right, here we go. So the overall goal of my talk is to gain a better understanding of the different disease-modifying therapies, or DMTs, that are available, that have various efficacy, safety, and mechanistic profiles, and review the different approaches that we have on starting and switching DMTs, and when to recognize it's appropriate to switch therapies. And finally, I hope that by the end of our talk, we'll learn how to individualize these approaches across different patients, incorporating comorbidities, risks, lifestyle, and reproductive considerations. So in MS, neuronal damage begins early, even before symptoms develop. And this contributes to long-term disease burden and loss of brain reserve. And DMTs matter. because they aim to reduce the overall disease and disability accumulation, but moving beyond just relapse control towards NIDA-based goals. So by targeting clinical, imaging, and emerging biomarker measures, DMTs help address the full spectrum of disease burden earlier and more comprehensively. So in MS, the key message is simple: time is brain. So sorry, stroke people who are in the audience, we're going to be taking your concept. Time is brain in MS, and we want to optimize therapy before irreversible disability accumulates and preserve the overall neurologic reserve by treating early and effectively. So that means starting high-efficacy treatment when appropriate and not waiting long for DMT switch if there is evidence of breakthrough disease, while also remembering that therapies take time to reach their full effect and differ across the different classes of DMTs. And the goal is to act early and adjust proactively to prevent ongoing disease activity and long-term disease progression. So MS DMTs can be grouped into low, moderate, high efficacy therapy, and then immune reconstitution therapies that are based on their overall impact on disease activity. So low and moderate therapies provide modest relapse reduction, while high efficacy agents offer stronger and more rapid disease control. And immune reconstitution therapies uniquely aim for durable benefit through temporary treatment and long-term immune resetting. So MS DMTs target different points along the immune cascade to reduce CNS inflammation. So our platform therapies such as interferons and glutaminyl acetate modulate immune activity, while agents like S1P receptor modulators and natalizumab limit lymphocyte trafficking into the central nervous system. Fumarates activate the NRF2 pathway that promotes antioxidative and anti-inflammatory effects. And more potent therapies like our anti-CD20 agents, including ocrelizumab, ofatumumab, and ublituximab, deplete B cells to reduce inflammatory drive. And then immune reconstitution therapies like alemtuzumab and cladribine go further by resetting immune populations for more durable disease control. So selecting an MS-DMT requires an individualized approach that considers patient, disease, treatment, and system-related factors. And treatment discussions often include balancing efficacy with safety and tolerability, while also taking into account a patient's lifestyle, their treatment preferences, their family planning goals, access to services and insurance coverage, because in 2026 that still matters, and ability to adhere to the monitoring and administration requirements. And because treatment decisions are no longer a one-size-fits-all approach, shared decision-making remains essential to ensure therapy selection aligns with the person's goals, their values, and then, of course, their long-term care priorities. And so there are two disease-modifying therapy start strategies that are used in clinical practice. We have early high efficacy and the more traditional escalation approach. And so with the escalation approach, patients start with a lower efficacy treatment that generally has a lower risk of serious adverse effects. And this requires close clinical and MRI monitoring and escalation if there is evidence of breakthrough disease. And in contrast, early high efficacy treatment involves starting more potent therapies up front, which is often associated with better outcomes in younger patients and those with more highly active disease, but it comes with a higher risk of more serious side effects and greater safety monitoring burden. And again, there's no one-size-fits-all. There's no a single approach that works for everybody. And treatment decisions should be individualized based on their disease severity, their risk profile, their disability trajectory, patient-specific factors, and shared decision-making. And Deliver MS and Treat MS, which are two ongoing, large, pragmatic clinical trials, will hopefully help clarify the comparative benefits of these two DMT start strategies. And so when deciding between the two initial approaches, there are some factors that you can consider to guide your conversations with your patients. And again, feel free to take pictures because these are going to be more specific information that's on the slide. So the early use of high-efficacy therapies is generally favored in patients who have more highly active MS features. So this includes high MS lesion burden, particularly in the posterior fossa and the spinal cord, where we tend to have more disability accumulation risks, multifocal presentation, frequent relapses, or incomplete recovery from those relapses. And additional risk indicators include early radiologic activity, such as new T2 or GAD-enhancing lesions within the first year, and early disease progression, cognitive dysfunction, or brain and spinal cord atrophy. And certain demographic and prognostic factors, such as male sex, Black race, and Latinx ethnicity, are also associated with more aggressive disease and may further support the considerations for these patients. So the early use of I already went through that. A more conservative escalation approach can be considered for patients with lower risk MS features where disease activity, it tends to be milder and the prognosis is more favorable. And so this can include lower lesion burden overall, absence of posterior fossa or spinal cord involvement, and good recovery from their initial relapses. And you can also consider escalation approaches in patients with infrequent relapses, minimal MRI activity at the onset, and no early signs of disease progression. And patient-specific factors also play an important role, including preference for therapies with a strong safety profile, as well as comorbidities that may limit the safety of high-efficacy treatments. And in these cases, starting with lower to moderate efficacy therapies with close clinical and MRI monitoring is often reasonable, but we should consider escalation for evidence of breakthrough disease when they're coming into our clinic. And so in real-world MS care, switching between DMTs is very common, and it's typically driven by things like breakthrough disease, including clinical relapses or new MRI activity. And switching may be appropriate because of tolerability or safety concerns that impact adherence, or evolving patient circumstances because they're planning pregnancy. They have new comorbidities that come up as people tend to age that can challenge the safety of certain therapies, or changes in insurance, or treatment access. So these are all real things that we see in our clinical practice. And progression independent of relapse activity, which has previously been discussed, or worsening disability in the absence of relapses or clear inflammatory activity, is also an important signal to reassess the current treatment regimen. But this often really needs to be done in a shared decision-making format with the patient and their care partners, particularly since the therapeutic options specifically targeting PIRA remain limited that is available currently. And a key caution when switching disease-modifying therapies is the risk of rebound disease, specifically after discontinuing certain therapies, particularly natalizumab and fingolimod. Okay, so this slide, take a picture. So this is going to be more specific information, but this is a general go-to guide on how to switch from certain disease-modifying therapies where there are maybe some key factors that can be considered to do this effectively and safely. So key safety considerations when we're switching DMT center on minimizing things like rebound disease, infection risk, and treatment overlap complications. And the highest risk for rebound occurs after discontinuing medications such as natalizumab and fingolimod, and making prompt initiation of the next effective therapy with close clinical and MRI surveillance is essential during this transition period. And additional considerations include monitoring lymphocyte recovery to guide safe timing of these subsequent immunosuppressive therapies. We also have to coordinate vaccine strategies, ideally prior to or during periods of immune reconstitution, and maintaining vigilance for infection risk, particularly when transitioning between B-cell therapies. And MRI monitoring is especially important in the first 3 to 6 months after switching high-risk therapies to detect subclinical disease reactivation early. And in practice, switching strategy depends on the clinical context. So escalation because of breakthrough disease, it generally warrants minimal or no treatment gap with rapid transition to a high efficacy agent. But de-escalation or safety-driven switches may allow for a brief washout, but should still avoid unnecessary delays. So it's trying to find that balance between the two. And across scenarios, the overarching principle is to individualize timing based on prior drug pharmacodynamics and immune effects, while also prioritizing the continuous disease control and avoiding periods of untreated inflammatory activity. So there is a very sensitive balance when we are trying to do this in our practice. But there is an art to it as well as a science. So monitoring for disease activity in MS combines routine MRI and clinical assessments to track both inflammatory and progressive changes. And brain MRI is obtained at the baseline prior to starting DMT, and then can be repeated 3 to 6 months later, depending on the patient's level of disease activity, to establish a new radiographic baseline. And then this can be followed by annual imaging in clinical practice for stable patients. And then if for patients who are more highly active, then we can say maybe we evaluate them a little bit more closely with MRIs that are maybe 3 to 6 months later, again, depending upon how clinically active they are. And this is followed by annual imaging in clinically stable patients. During the monitoring phase, cervical and thoracic spine MRIs are generally reserved for suspected clinical activity or disease progression. And then for patients where we may worry about them a little bit more, such as our patients who are JC virus positive who are on natalizumab, we may want to exercise more frequent MRIs every 3 to 6 months. And then in some situations, stable patients without disease activity for over 5 years, we may be able to extend that surveillance a little bit if safety allows, and of course per our own clinical judgment on whether or not we can do that. And then clinical monitoring includes relapse assessment at each visit. And then of course we can use standardized measures of disease activity and disease progression. And we can use the expanded disability status scale score, the timed 25-foot walk, the symbol digit modality test, the processing speed test otherwise, and then of course our patient-reported outcomes, depending upon how much time we have in our clinic. And then adjunctive tools like our serum neurofilament light chain, our Octave MS disease activity score, and retinal OCT also provide additional sensitivity for measuring disease activity and disease progression, again, depending upon what we have available in our clinics. So let's go ahead and we're going to switch gears a little bit and focus in a little bit more on B-cell depleting therapy approaches and considerations. And the use of anti-CD20 therapies is really common in MS care, both as an initial DMT and also as a switch therapy. And real-world selection for our B-cell therapy prioritizes patients with more highly active disease, concerning disease burden and risk of early progression, or breakthrough activity on platform or more moderate efficacy DMTs. And they can also be considered for individuals with active secondary progressive MS per the label. And for ocrelizumab, given its FDA indication, we can use it in primary progressive MS, but with the caveat that patients who are younger, like less than 55 years of age, or those with radiographic activity based on the pivotal oratorio clinical trial data, these are the patients who are likely going to respond more effectively to this type of therapy. And B-cell therapies are also a good choice for patients who want infrequent dosing, and that's a preference for them. And it's also commonly considered in JC virus antibody positive patients where natalizumab is less desirable because of their PML risk. So lots of different indications for where B-cell therapies can be appropriate. But relative contraindications, or I guess words of caution, include patients who have active hepatitis B infection, those who have pretty significant hypogammaglobulinemia or low IgG levels at baseline, they are at risk of having serious infections or currently have active serious infections. And then patients who need live attenuated vaccines, or elderly and frail patients medically where infection risk may outweigh the overall benefit. So there are special considerations for these individuals. And another place where there are special considerations are our patients who are considering pregnancy planning, and I have more information on that in a separate slide. Okay, get your cameras out. There's a lot of information on that slide, but I intended for there to be a lot of information here. So again, you can bring this information back to your clinical practices. So the long-term safety management of B-cell depleting therapies in MS focuses on a balancing act. It's balancing high efficacy with risks of infusion or injection-related reactions, infections that are mostly upper respiratory and urinary tract infections, and immune suppression. And so the infusion reactions are most common early, and they're generally manageable with pre-medication and monitoring. It's really not so much of a big deal in our clinical practice. But serious infections remain an important ongoing concern, particularly with advancing age and in patients who develop hypogammaglobulinemia with prolonged treatment. This is quite common amongst our patients who are on long-term therapy. And this warrants IgG monitoring and potential treatment adjustment. Overall PML risk is rare in anti-CD20 users and has predominantly been reported as a carryover effect from natalizumab, but it does remain a key consideration, especially as our patients remain on treatment longer and they may have ongoing risks for other opportunistic infections. So vaccine responses are attenuated during therapy. This is important for us to remember because we're always talking to our patients about vaccines. There is a reduced but not absent influenza and COVID-19 vaccine response, but we can tell our patients that we can time those vaccines around their injections or infusions for a better response. And a recent single-center study reported a potential association between prolonged ocrelizumab exposure and late-onset interstitial lung disease, particularly organizing pneumonia that occurs after long-term treatment of about 10 years and greater. And these patients presented with respiratory symptoms that often required a corticosteroid therapy. But the importance of these emerging data are to showcase that as our patients continue these B-cell therapies long-term, there may be some additional risk factors that emerge that we really need to be careful about the safety of long-term use. And in a recent article that was published in 2023, the authors proposed a practical monitoring and management approach for secondary hypogammaglobulinemia in patients on B-cell therapies. And this emphasizes routine IgG surveillance and interpretation in the context of clinical infections rather than absolute thresholds alone. And patients with mild hypogammaglobulinemia and no infections may continue therapy with close IgG and infection monitoring, but those with declining IgG and emerging or recurrent infections should prompt mitigation strategies such as extended interval dosing or disease-modifying therapy switch altogether. And in cases of clinically significant hypogammaglobulinemia, particularly with recurrent or serious infections, more substantial intervention may be required, including a switch in their DMT or IVIG that's reserved for patients who have persistent infection burden or severe HGG as either an adjunctive support tool or bridging therapy to maintain immune protection while they're on the therapy. So let's talk a little bit more about our disease-modifying therapy approaches in specific situations and populations. All right, get your cameras out. So this slide provides detailed recommendations on DMT strategies around pregnancy. And this is based on available evidence. Since you all will have, I think you have a more detailed conversation on women's health and pregnancy planning a little bit later in the program, I'm just going to focus this on DMT approaches. So MS disproportionately affects women of childbearing potential, and this makes our conversations around pregnancy planning a key component of our care, even from day one when they're coming in to see us in clinic. And this requires a balancing act of relapse prevention with fetal safety and the risk of disease rebound after withdrawal of certain DMTs. And pregnancy is generally protective against relapses, but postpartum inflammatory activity is common. So proactive family planning, individualizing disease-modifying therapy selection, and a very clear postpartum treatment strategy are really important. So management is guided by the patient's baseline disease activity, their prior treatment, and of course, their own preferences. And there are variable recommendations for continuing DMT, washout, and timing. And this ranges from pregnancy-compatible agents such as interferons and glutamate acetate, to fumarates that can be stopped at conception, and to therapies requiring a more structured washout or risk mitigation, such as our S1P receptor modulators because of risk of rebound, teriflunomide because of teratogenicity effects, and cladribine and alemtuzumab that have defined conception delays. And high-efficacy DMT options, such as natalizumab, with extended interval dosing strategies during pregnancy, and B-cell therapies are increasingly used in highly active disease to maintain that disease control throughout their pregnancy, with special attention to the fetal exposure and then the neonatal effects. So overall, care requires individualized risk-benefit decision-making across the spectrum, across preconception, across pregnancy and postpartum periods, with close monitoring, and then of course early resumption of therapy postpartum to reduce that overall reoccurrence of activity risk. So switching gears to DMT approaches for our aging patients. We'll review the data here on why and how to de-escalate and discontinue MS therapies, although it's very important to know that there is no clear consensus on this, so this is based on what we know and the information is emerging. This is especially important because the aging MS population is becoming increasingly prevalent as our advances in care simply allow our patients to live longer. And there's currently no consensus, as I had mentioned, on how to do this, but the question, again, is so important, not only because our patients are aging, because we're starting our patients on early high-efficacy therapies. They're on therapy longer, and they may require adjustments because of those long-term safety concerns that emerge later on, such as the infections, the hypogammaglobulinemia, and other comorbidities. And this is also relevant with the observation that efficacy difference between the higher and lower efficacy treatments can narrow with age, and it can narrow with disease evolution, as we have seen in other talks earlier today. And we should keep in mind that the observational data suggests that de-escalation from natalizumab to moderate efficacy therapies may be associated with increased disease activity compared with a lateral switching approach to another high efficacy agent, and therefore it might require a bridging therapy. But the evidence remains limited for de-escalation from B-cell therapies and is currently being evaluated in other ongoing clinical studies. And one of these is the COAST clinical trial that is evaluating the efficacy and safety of switching from anti-CD20 agents to ozanimod. So more data are going to be emerging rather quickly. And the importance of evaluating de-escalation strategies from B-cell therapies in particular is based off of prior observational studies. So in a couple of single-center studies at the University of Colorado, investigators demonstrated that there was over a 3-fold increased risk of infections in patients on B-cell therapies who had a low IgG level of less than 500, even after adjusting for other demographic and clinical factors. And they also showed that high-efficacy DMTs, including rituximab and natalizumab, that here is represented by the blue line, were more efficacious than moderate efficacy oral DMTs, here represented as the red lines, including dimethyl fumarate and fingolimod. And so as MS patients aged, the likelihood of disease activity decreased, and the difference between the high efficacy and moderate efficacy DMTs became more negligible, especially as the patients approached 50 years of age. So here we summarize those basic considerations for de-escalating and discontinuing DMTs using the available evidence from clinical trials, observational studies, and also what we just see in clinical practice. And again, these are not consensus-driven. But some basic tenets are that DMT de-escalation may be considered in patients with long-term stability. They have low disease activity, particularly in the setting of aging. So for example, patients who are maybe getting to be 55 years and older, and those with more comorbidities. And de-escalation can also be considered in patients who prefer a safer drug or need to reduce infection risk. And in practice, de-escalation after high-efficacy therapy should be approached cautiously, allowing for some immune reconstitution in certain circumstances if there are safety risks, with careful consideration of bridging strategies. And disease reactivation can occur. And this can occur particularly within 6 to 12 months of making this switch, so monitoring is going to be really important here. And I would say that close MRI surveillance at about 6 months and then every 6 to 12 months for at least a couple of years can be some MRI detection tools of whether or not there is some reactivation of disease coming forward. So for discontinuation, we can consider this approach in older patients. And again, there's no age restrictions here, but we have been seeing maybe along the lines of about 60 years of age, if they have evidence of having prolonged stability, inactive disease, and minimal disability progression. And we should keep in mind the data from clinical trials and observational studies are being used to guide these approaches, and set ages are not standardized at this time. So we need to keep in mind that the age cutoff for certain clinical trials, such as the DISCO-MS clinical trial that was evaluating patients who were continuing versus discontinuing DMT, was maybe a little bit on the younger side of 55 years old. And so this is just giving us information of when might be an appropriate time to have those discontinuation DMT conversations with our patients. So as an example, younger patients, maybe less than 50 years of age, those who very clearly have inflammatory disease such as new T2 and GAD-enhancing lesions, should not discontinue DMT unless there are specific extenuating circumstances like safety considerations. So overall, de-escalation and discontinuing strategies require shared decision-making. It requires clear counseling with our patients on the risk of reactivation and a predefined plan for prompt reinitiation if disease activity reoccurs. So I think I'm getting a little bit close on time, so I'm going to skip over our clinical case vignette, but if anyone has questions on how to use these approaches in clinical practice, I am more than happy to go through those examples with you all. So let's put everything into summary. Treatment in relapsing MS should be started early with two main strategies for DMT selection. We have our early, highly effective approach, or a traditional escalation approach, that's guided by many different factors. This includes patient factors, disease factors, treatment factors, and of course, our system and access factors. And the overarching goal is preservation of that neurologic reserve with a low threshold to switch therapies in the setting of breakthrough disease, reinforcing the principle that time is brain. And there's no longer a one-size-fits-all approach in MS. Treatment decisions are increasingly being individualized to balance efficacy, safety, and tolerability for each patient. And emerging evidence does support a more aggressive strategy, including early use of high-efficacy therapies in patients with more highly active disease, or if they have risk factors for early disability and timely escalation of treatment before irreversible disability accrual occurs. And so there's growing evidence that supports the safe use of select DMTs during pregnancy planning. And this is alongside an increasing emphasis on individualized treatment strategies, including a thoughtful de-escalation or discontinuation step in appropriately selected patients. And so with that, I thank you. I think I was able to keep us at 30 minutes, hopefully. But there you go. DMT approaches in 30 minutes or less.
Thank you, Kevin. That's an enormous amount to cover, and it was great. So, questions? While you're thinking, I'm wondering if escalation is a misnomer, because it implies that you're going to be changing. And in fact, lots of people do just great
That's
on the therapy you start them with.
That is very fair. And I think that the name itself is a misnomer because in the definition, it states we escalate if we need to, if there is evidence of breakthrough disease or clinical MRI breakthrough disease on that therapy. But yes, someone could start on a lower efficacy therapy and may never need to escalate to something else.
Right, so there's responders to every therapy we've ever
Correct.
done, otherwise it wouldn't be approved. Our ability to identify that was not necessarily a great mark.
Thanks for the whirlwind tour. Patient is stable on anti-CD20 several years, no relapses, stable MRI, but they're getting worse. What are you changing to?
That's a very loaded question. Those patients for me are by far the most complex and challenging because as mentioned before, our currently available disease-modifying therapies are not really all that great at targeting more of our progressive disease drivers of progression that tends to be more in the central nervous system. And our available FDA-approved therapies primarily work outside of the central nervous system. When available, so we're very fortunate at the Cleveland Clinic where we do have clinical trials that are open for patients with either secondary progressive or primary progressive disease. So if they are eligible for participation in a clinical trial where we're able to evaluate their efficacy, safety, and overall response to these emerging therapies, which Dr. Friedman, I know you're going to be giving a talk on, that may be a good place for them. We potentially have some therapies that are currently being evaluated for FDA approval for progressive disease in the BTKI arm. So we'll see if that is going to be available for these patients. But we may be able to switch their mechanism of action because there are some patients who are better disease responders to certain DMTs as opposed to others. Maybe this will help slow down some of their disease progression. But I really am having open and honest conversations with patients that some of this is a flip of a coin.
Other questions? Yeah.
That's a great question. So,
Because I don't think people heard it.
I think that you were asking, sir, about the use of biomarkers in my practice, such as serum neurofilament light chain, to make treatment decision-making. So at the Cleveland Clinic, we're a little bit behind in using something called the Octave MS Disease Activity score. I think there's going to be some talks on this throughout the course of the consortium meeting. But you know, this is emerging right now, and I think we need to be careful with some of the more commercially available SNFL because not all of them are standardized across their normalization, across age, across sex, and across BMI, which can also be important. So I think we have to be careful about how we're using them currently. But there may be some evidence that some of these emerging biomarkers, such as Octave MSDA, can give me some indication of whether or not someone is having inflammatory activity if they don't have access to an MRI, it's too costly, or they just had one. So it can be informative.
Other questions? Okay, we're going to take our delayed break, but only for 10 minutes tops, and then we'll come back and complete the rest of our day. Carrie, thank you so much.
Thank you.
What?
It's a good idea to come home and see
Yeah.
what she would.
Backwards, forward. She's going to load it up for you.
Yeah.
Or you can use this.
Oh, okay, good. Good, good.
I don't want to touch it because you probably have it set up.
Yes, and I actually have to just swap out one. I don't know why I would have brought this up there, sorry.
I do this myself.
What's wrong?
I just have to swap one side
Okay.
deck out really quick if I can.
We're going to get started, guys.
Okay, I'll do it after you.
Everybody, back at your seats. Good, good, good. Okay, let's get going because we're running behind.
Is that
But
better?
we've had some excellent talks. And we're going to continue that for the rest of the afternoon. Is he set?
There we go.
Okay. All right, grab your coffee and come on back. All right, moving along, we have Professor Mark Friedman from the University of Ottawa, who's going to discuss emerging therapies.
Thank you very much, Fred.
Actually, for those of you who have been here for most of the afternoon, it's going to be a rehash of a couple of things, but I'm going to try to focus in on a family of drugs, the BTK inhibitors, touch on a new molecule, frexalimab, which you did hear a little bit earlier if you were listening to Fred. Talk a little bit about an emerging therapy that I found very interesting, and then talk about CAR T cells, but that you're going to hear a lot more about that in other sessions throughout the consortium. So Bruton's tyrosine kinase is a pretty universal enzyme that is important for a number of cells, and in terms of what's important to MS, it can affect the B cells, so it's important for B cell maturation. And you've heard a lot about the immunology. I'm not going to go through this, but through the maturation of the B cells and where B cells might play a role in the pathophysiology of MS, I think Scott talked about right at the beginning of the afternoon. But it also has another role, and it's expressed on cells of the innate immune system, especially the microglia. And we've heard that the microglia are very important to perhaps the progressive nature of the disease, contributing to that low-grade inflammation that is compartmentalized. And so you have now a small molecule which can do two things that are relevant: affect the B cells, which we know can contribute a lot to the inflammatory part of MS, but also to affect the microglia, which may be important to the progressive nature of the MS. And it does get into the brain, therefore we might be able to kill two birds with one stone using the Bruton's tyrosine kinase. Where does this play a role in terms of the B cell functions? You can see here on this slide, hopefully my little pointer works here, where the anti-CD20s and the anti-CD19s can affect through the maturation of the B cell. BTK is important almost throughout the same spectrum of B cell development and lineage. And in that case, it can reduce the formation of these inflammatory B cells. What it does to the innate cells or the macrophage microglia is to change their profile from these pro-inflammatory, which may be contributing to that so-called smoldering disease, to more of an anti-inflammatory, where those cells will no longer be responsible for some of that ongoing damage. So that's the theory. There are many BTK inhibitors in the family. Most of these are in advanced development in phase 3 trials. Evobrutinib, you've heard, has failed in its initial trials to take on teriflunomide and improve upon its anti-inflammatory effects. Tolebrutinib has finished its studies. Fenebrutinib has finished its studies, and I'll just touch on some of the data because a lot of it's not published but recently presented. Remibrutinib is still very much in study currently, both in relapsing and secondary progressive MS, and orelabrutinib is actually just starting its program. So these two have completed, and I've got some data to share with you on both. Some of this data has already been published. The Gemini studies, we're looking at tolebrutinib versus teriflunomide. Most of these studies are looking at ARR, or relapse rate reduction, as a primary outcome. I'm just going to go quickly through some of the baseline characteristics, but point out that these were very active patients. And they're somewhat similar in the sense that more than a third of these already had enhancing lesions at baseline. So they are truly an inflammatory form of MS. And the bottom line is that teriflunomide has done much better than expected in almost all the studies that it's been used in recently, with very, very good ARRs, not particularly better with the tolebrutinib, and if you look at the pooled ratio, there's really no difference. So it failed on its primary outcome of relapse rate reduction. However, it did show a significant reduction in confirmed disability worsening over 6 months. Keep that in mind when I tell you about some of the other studies in a moment. Interestingly, it had no effect on reducing MRI lesions. In fact, if anything, it looked like, for whatever reason, there were more lesions in the tolobrutinib arm than the teriflunomide arm. But when you look at new or enlarging T2 lesions, there really was no difference. In walks fenebrutinib, similar to studies in relapsing remitting MS called FENHANCE 1 and 2, and the very similar type of trial design. Again, look at the activity in the baseline population, very similar to what we saw in the Gemini population, with more than a third of these patients having enhancing lesions at baseline. They needed the same type of relapse inclusion criteria coming in, but unlike tolobrutinib, here is a very strong effect at reducing relapses relative to teriflunomide, which is already running quite good. So when you actually look at these numbers, you're having one attack every 10 years with teriflunomide, and you're changing that to maybe one attack every 15 years to the patient. So you got to start asking yourself the question, is that really a relevant reduction or not, 50% or not? If you take 50% away from almost nothing, you still have almost nothing. So what's important here? Well, there is also the mimicked reduction in inflammatory lesions, what you would expect in any drug this far that has ever reduced relapses. It also reduces MRI activity. However, when you look at confirmed disability progression in these studies, there was no treatment effect whatsoever. All of those confidence intervals cross 1. So there was no significant effect on reduction in worsening. And there's this extra little thing that came up, which is interesting, but still not terribly explainable. There were a lot of deaths on the fenebrutinib arm, with only 1 death in the, actually with no deaths in the teriflunomide arm. So there's some safety signal here that needs to be worked out. Now, a very interesting sub-study that was looked at just very recently by Maria Pia Sormani, who's a statistician very much involved in clinical studies, asked the question, and maybe we can come back and have Fred comment about this because he talked about it earlier, and what is driving the disease progression? Is it something associated with relapse, or is there something distinct? And when you look at the association, and this is a statistical effect, okay, so it's a statistical type of equation. These are all the studies on this line that compared themselves to teriflunomide and asked the question, is there a relationship between the effect on reducing relapses and MRI activities versus reducing disease progression? And in every single one of these studies, evobrutinib, ponesimod, which is an S1P receptor agonist, ofatumumab, anti-CD20, ublituximab, anti-CD20, the effect on disability was reflective of the magnitude of effect on relapses in MRI, but not tolobrutinib. There was a very much a distinct effect of tolobrutinib on disease progression that seemed very much independent of any effect on the inflammatory metrics, which are relapse and MRI. So in walks another study of tolobrutinib on non-relapsing secondary progressive MS, wherein the baseline population, about just a little more than 10%, had Gatto-enhancing lesions. So this is truly a non-inflammatory form. And when you look at that, is there any effect on disease progression? There was a very clear effect at delaying disease progression. So is this particular BTK inhibitor doing something that the other BTK inhibitors are not? That's an important question. And in this Hercules study, there was a reduction in T2 lesions for whatever that means. When you look at primary progressive MS, and there's a lot of question as to the population that was in this primary progressive study, because for those of us who were involved, I can tell you we were recruiting for this Perseus study at the same time
we were recruiting for the Fentrepid study that I'll tell you about in a moment. In the Perseus, it was primary progressive patients randomized to placebo versus tolobrutinib, whereas in the Fentrepid studies, it was fenebrutinib now randomized in patients with primary progressive MS to either fenebrutinib or to ocrelizumab. So it was very hard to take patients into Perseus because they had to decline ocrelizumab, which was the only drug for primary progressive. So in that sense, very different type of population. End result was no difference. So there was no difference between placebo and tolobrutinib in this primary progressive population. More to come about that. It didn't meet its primary endpoint, but there's a lot of secondary analyses going on which may support still an effect on what is creating the disease progression. When you look at fenebrutinib versus ocrelizumab, the other study I told you about, similar trial design, but this time ocrelizumab versus double dummy study, so they didn't know patients what they were getting. Again, no difference. So it was non-inferior to ocrelizumab. And whenever you have a non-inferior study, your result is always that the drug being tested, in this case fenebrutinib, is as good as ocrelizumab or is as bad as ocrelizumab, but you don't know because there's no natural history arm here, which is the placebo. So more to come about these two studies, which are somewhat revealing. Okay, so fenebrutinib didn't do any better than ocrelizumab, but there is some question as to whether these BTK inhibitors differ from one another. Evobrutinib didn't work at all. We have tolobrutinib, which has an effect on progression independent of relapse activity, and we have fenebrutinib, which has an effect on relapses but doesn't affect on disease progression. You heard about frexalimab, and because Fred almost showed the same studies, I'm going to just go through these very quickly. It's a different pathway. It's an adhesion molecule that was tested years ago. I think Biogen brought the first agent out, but because of its way it was engineered, it ended up causing bleeding due to its effect on platelets. So it was re-engineered. Now that problem is gone. So it's got some idea, the theory behind it, and this is again similar to the BTK inhibitors, is that it has an effect both on B cells and on possibly the innate cells, the microglia. And in a phase 2 trial, which is always a test of whether or not it's going to reduce inflammatory lesions, it was very clear that the effect on B cells manifested as a reduction in gadolinium-enhancing lesions over time without compromising immunoglobulin function. And you heard earlier today that that can be a problem, especially in the older age group, where immunoglobulins go down and increase the risk of infections. The lymphocyte counts remain stable over this period of time, so it's a different mechanism of action. You don't have to deplete the cells. You interfere with their activation, and therefore you don't necessarily compromise the patient. So at least this phase 2 study has gone into extension for safety purposes. It looks like it has some promise. They are both in phase 3 studies. Fred talked about that already, and we'll probably hear more about this in a couple of years. Now, recently presented at the ACTRIMS was a phase 2 study using a completely different molecule that many of you haven't heard about. At least I didn't hear about it before I knew about this Moonstone study, obexelimab. So what's so neat about obexelimab is that this is an engineered molecule which links CD19 on the surface, that's a surface on the B cell, as you know, but on an Fc gamma receptor to the base of the obexelimab. So it's a humanized monoclonal antibody, sort of bivalently coming together for CD19 and Fc gamma RB, which is an inhibitory molecule. So it turns off cells. It's kind of like mimicking the signal that the B cell would get when it binds to different types of antigen-antibody complexes. So they did this study in relapsing, remitting, and active secondary progressive MS. It's not a huge study. It was a 2-to-1 randomization. You can see 78 patients in obexelimab versus 38, I just gave away the answer. In the placebo, the phase 2, always looking at enhancing lesions as an outcome measure. And sure enough, look at the reduction. So this is akin to what we saw when the first anti-CD20 antibodies came out. You got 90+% reduction in new enhancing lesions. So near complete suppression of new GAD. What's really neat about this? Okay, oh, and also new enlarging lesions, so just exactly what you would expect, but very few side effects, and more importantly, no change in the B cell count and no change in immunoglobulin levels whatsoever. So this is a very, sorry, this is just going crazy. So this is a new molecule obviously going into phase 3 studies, which takes advantage of the mechanism of reducing the B cell introduction of antigen, however it drives the disease, turning off the new inflammatory activity, but no compromise to the patient. And it would be very interesting to see what happens in the long term with this drug, because it could easily exceed any of the anti-CD20s, which have been fraught with problems owing to the fact that they have to deplete the B cell population. Here, you don't have to deplete the population. You've affected them in a certain way that leads to the advantage that we're seeing with anti-CD20. And finally, in the last few minutes, I'll just introduce CAR-T. If you want to know more about that, there's a session on Thursday that I'm running, a whole symposium on cell-based therapies, and CAR-T will be amongst them. Very, very sexy type of new therapy that is being used for treatment of mostly hematological malignancies, but now moving towards all different types of cancers, because the technology allows us to take the T cell, the important T cell, and its receptor, and cleave this receptor and put on it whatever we want. So the patient's own cells, or you can manufacture these cells. So there's companies now that are making them that are MHC compatible to whatever patient. But you can make these cells that now you can put on a receptor. And in this case, the first construct was against CD19. So it would target B cells. Now, what makes these cells very interesting is, as we've been trying to get things into the central nervous system, because it's so difficult to do, you can get small molecules in, but our body has had the ability to move things into the central nervous system for, that's the way we were born, and cells have a way of trafficking into the brain, and T cells in particular have no problem getting into the brain if they have to. So you're using the T cell construct to now penetrate and seek out whatever we stick onto the surface of these CAR T cells, and in this case, if they recognize CD19, maybe they'll be able to get at those cells that are stuck behind the blood-brain barrier that we think might be doing something to the disease, especially progression. And if you believe in the follicle story about these follicles on the surface of the brain being involved in that disease progression, then there's a way of targeting them. And all, just like the way I showed you the BTK effects on the B cell lineage, you can see here all the cells that might be involved and the target of these CAR T cells right up to the plasmablast. And the idea is that these are cells that now can penetrate the central nervous system, and they can maybe get at these resident B cell follicles that are in there. So not surprisingly, with all the different companies that are making these CAR T cells, there are a whole list of studies shown here on the left that are registered on clinicaltrials.gov that are mostly phase 1 safety studies, because it's so hard and tedious to treat these patients. I know that we're trying to get the study off the ground in Ottawa, and you need a bed in hospital for 4 to 6 weeks for each patient because of the potential of problems once these cells gain entry to the central nervous system. Once again, the Chinese have got us scooped on all of this. How they've been able to move ahead so quickly is unclear, but the group in Wuhan have done a number of patients already with multiple sclerosis. We heard about one from Gavin Giovannoni's group at Bart's. He talked about it at Ectrums. This was one of the patients that went to Wuhan to actually get the treatment, and they've just recently published this in the Cell Journal. So here are 5 progressive patients that are being treated with a bivalent B-cell MA anti-CD19, so BCMA is B-cell maturation antigen, so it's another molecule on the surface of the B-cell. They have a bivalent now construct on the surface of their CAR T. And when they looked at these 5 progressive patients, you can see very, very not so easily on the graphs on the right, but their EDSSs looked like they actually improved. And they also looked at some other factors within the CNS, and they saw some microglial activation or markers of activated microglia going down. So maybe they are affecting the whole process that is driving progressive disease. These are very early studies, and in their particular circumstance, they didn't talk about high ICANN. ICANN is the problem that you get, immune cell-associated neurotoxicity that you can get with these CAR Ts. So they're not trivial, but nevertheless, if they can change the course of progressive disease, they would be fascinating. So in conclusion, we've got a whole family of BTK inhibitors that are being tested now. Some of these are looking like they may do different things. Maybe they're going to get at the compartmentalized inflammation that's driving progression. Frexalimab now in phase 3 studies using an co-stimulatory molecule that may have unique properties over what we currently have. Obexelimab targeting the B cells without having to deplete them or reduce immunoglobulins, so maybe safer in the long run. And finally, the sexy CAR T group that we'll probably hear a lot more about in coming years. That's just a touch. I just grabbed these. If you look at clinicaltrials.gov, there's a whole slew of other things being looked at in MS in both phase 1 and phase 2 trials. So enjoy the rest of the meeting, and I'll take any questions you might have. Thanks.
Thank you, Mark. Okay, questions? Really? Okay, Anthony.
Oh. So there were a lot of deaths in the fenebrutinib study. They were all causes. There was no seemingly theme, but it was already posted all over the FDA website that they were concerned about all those deaths. Because there weren't, well, teriflunomide is pretty well tolerated. And whenever you see a disparity like that between treatment arm and one arm and the other, you've got to ask some questions.
Any understanding of the mechanism of action of obexelimab, that it doesn't reduce the B cells?
Well, you don't have to deplete the B cell. So it's a bivalent target, so it's working through the Fc gamma receptor, which are inhibitory receptors. So every cell has Scott talked about the immune system earlier. It is very complex, but in the sense, there's always a yin and a yang, there's a go and a no-go, and so it's hitting a bit on some of these receptors that are no-go. So they can turn off a cell. Otherwise, we would just chew ourselves up all the time for immune reactions. So these are one of the inhibitory receptors that work on B cells. And by targeting CD19, you identify the cell. By targeting the inhibitory receptor, you turn it off. So you're able to turn off the B cell and whatever it is producing that perhaps is contributing to the inflammation. Maybe, we don't know why you knock down the B cells. It's certainly not because you're reducing immunoglobulins. It's probably the interaction it has with the T cell. So in the end, obexelimab is probably hampering those pro-inflammatory B cells from interacting with the T cell, and the end result was a 95% reduction in inflammatory lesions, which is pretty well what we saw with all the anti-CD20s, but you didn't have to kill the B cell, and you didn't have to reduce immunoglobulins.
Nice. Other questions? All right, thank you, Mark. Next, we have Colleen Harris from the University of Calgary. Mitigating adverse events. Welcome, Colleen.
So thank you. I'm going to go, this presentation should be quite straightforward, and we should be able to gain a bit of time back because Dr. Hirsch took care of some of it for me in her very comprehensive, excellent talk. So my topic is mitigating adverse events with MS disease-modifying therapies. I'll just go through these in the division of early treatments, oral medication, and higher efficacy treatments. This diagram is familiar to many of us in the room. It just sort of shows you how many different medications have been brought to market since 1993. My history with MS dates back to pre-1993. And I have been able to see the introduction of all these medications into the treatment landscape of MS and notice improvement right from the beginning. Very lucky to have the higher efficacies now, even seeing more benefit to our patient population. Again, this has been touched upon in previous talks. Our drugs can be sort of divided into their unique action, all contributing to controlling but not curing this disease. Our ultimate gold standard aim is to definitely alter the natural course of the disease and delay disability progression. I was around when there wasn't drugs that altered the natural course of the disease, so this has very much been a very rewarding career that I've had to see such changes. So our early treatments, very quickly I'll touch upon the interferons and glatiramer acetate. These drugs were very well tolerated overall. However, they were expensive and they required lab monitoring and self-ejection. So that was the complicating factors there. This was when MS nurses really got on the map because you needed an MS nurse to teach the patient about it and to work on issues like adherence, monitoring side effects. The interferons, as I said, were quite well tolerated. There were flu-like symptoms in the beginning, mitigated with acetaminophen and NSAIDs and dose escalation and injection site reactions, which were common in the early days because we didn't have as many of the sophisticated auto-injectors we have now. Lab abnormalities in the beginning, liver enzyme elevations had to be monitored, and we sometimes had to adjust doses. Glatiramer acetate, again, an injectable medication, different class of drug, very long safety record. It was very rough on the skin, caused inflammation and lipoatrophy. Adherence was challenging because it was a daily injection, and yet there was nothing else, so our patients persevered with this medication until something better came along. Along came the oral therapies. We all assumed these would be super easy to take care of from a clinician perspective, and it would be much easier for the patient. The very first one to hit the market was fingolimod in 2010. It was an oral daily drug, but because of its off-target effects, in other organs of the body because it is an S1P receptor modulator, and there's lots of SP1 receptors throughout the body. And so this off-target effect, particularly on the heart and the retina, required a lot of vigilant monitoring. So there goes our thoughts of easy. We had to, because of the cardiac effects or the risk of those, we had to do first dose observation on patients starting this medication just for their first dose to ensure there wasn't significant heart block. We also, because of the symptoms, the worry about the retina and the worry about macular edema, we had to ensure ophthalmologic exam before and 3 to 6 months after the first dose of this medication. Lymphopenia, which is expected, but we monitored closely to make sure it wasn't too low, and elevated liver enzymes were also part of some of the reported side effects early on. So it was, and other side effects as well. So it wasn't easy peasy just because you popped a pill. It still required significant surveillance. And generally this was done by nurses in the clinic. Teriflunomide has been talked about today. It's a drug that we had studied for years before it went on the market. We were involved in clinical trials. We kept wondering when it would come to market. Well, it did, and we used it. It required monthly liver enzymes initially because it had a black box warning, and teratogenesis for women and men. I don't think it was a manageable issue, but not during when a woman and they were contemplating starting a family. So it wasn't highly recommended for moms wanting to become pregnant for sure. There was a way to get rid of it with cholestyramine, get it out of the system prior to or considering a pregnancy, or if a pregnancy resulted. The normal side effects for oral medication initially were evident, but generally it was fairly well tolerated that way. A little bit of hair thinning, which was upsetting to some, but it usually reversed itself. This drug, because of its history with rheumatoid arthritis, we had to monitor the patient, make sure they were free of TB prior to initiating this medication. So this is when we started our pre-screening for TB, which carried on for other medications for populations at risk. Dimethyl fumarate, we only had one fumarate in Canada. I think you guys had 2 or 3. It was a drug that was oral. We thought it would be better and easier to give than the Gilenya or fingolimod, but it wasn't. Initially, people had a lot of GI discomfort and diarrhea, which was quite troublesome in the early days. We were really afraid that this might not be a good option for patients. However, with perseverance and education on good dietary patterns, this side effect was mitigated quite nicely. Flushing, it was a persistent side effect, upsetting to some. Others, it didn't bother so much. This could be mitigated by ASA, 325 milligrams, just before, 30 minutes prior. It was helpful, but many just learned to adjust to this side effect. Of course, we had to monitor again. Lymphocytopenia was evident in some people, and because, and it was persistent in some, and if it was, we often switched to another medication. There was record of PML, although a very low estimate of existing right now, and it was, those cases were usually associated with prolonged moderate to severe lymphopenia. We would, in my practice, would discontinue this medication if the lymphopenia was persistent, and we certainly use this cautiously in an older population. Cladribine, which is an oral cancer drug used in hairy cell leukemia. It was a very easy drug to get into patients because it offered a really nice dose schedule. They had a 2-month, 1-week course in year 1, and that was repeated in year 2, and then they were done. This was attractive to some people. Lymphopenia was there, but less than a grade 4. We did caution patients with this, going on this medication, about risk of infection, particularly if there were small children in the family. The infection risk in the studies was similar to that in placebo, except for herpes zoster, and so we medicated for that prophylactically. Higher risk malignancies were reported, and this is when we started counseling patients regularly to adhere to their regular wellness cancer checkups for breast, cervical, skin, because this could be a potential adverse effect. So the first, the higher efficacy treatments, the first one actually was natalizumab that came to market in 2004, a very effective medication. It had to be withdrawn. We've talked about that today as well. And then re-entered again in June 2006 after a very good risk mitigation program based on anti-JC virus detection. And it's been used, I think, quite safely since that time for high-risk patients with very active disease. We've gone to extended interval dosing so that we can continue to use this drug. It is very effective. Other adverse effects from that include, of course, iris, if they develop PML, and very mild infusion reactions. The next 2 drugs I'm just going to mention, neither one of them are used extensively, but alemtuzumab was a medication that was very good at T and B lymphocyte depletion, and sort of resetting those really bad presentations of MS. It was administered as a daily 4-hour infusion over 2 treatment courses, 12 months apart, and then again they were done. It had a whole list of adverse events, including infusion reactions. I think this is where our recipe for bad infusion reactions originated, to get our patients through these infusions. It always required steroids, antihistamines, and maybe even antipyretics. There is a whole host of anti-immunity that this medication caused. The most common one was thyroid autoimmune disease, both hypo as well as goiter. We had to use the standard thyroid disease treatment, and we had to have hotlines to our endocrinologists who helped us through this. Thrombocytopenia, again, we had to have a hotline to our hematologists because this did occur. We had to teach the patients about the signs and symptoms of this side effect, looking for little marks on their skin. And then some kidney autoimmune disease as well. We needed hotlines to nephrologists. So this was to me, a very potent drug, a very difficult drug to manage, but it was very effective. We were using it in very highly active disease until the appearance of rare and fatal heart attacks and strokes. 1 to 3 days post-infusion were reported. I think it sort of lost a lot of interest then because we're talking about quite severe issues that couldn't be helped with our other internal medicine specialists. These were very bad adverse reactions. So it did fall out of favor, but it was a very effective medication, mitoxantrone. It was a chemotherapeutic agent. It had a lifetime limit because of its effects on the heart. Again, it was used before we had B-cell therapies, but did fall out of favor when we had safer options to settle down very active disease. It had all the side effects that other chemotherapeutic drugs have, including hair thinning as well as mouth sores. So it was chemotherapy. And then came the B-cell therapies, made a huge difference to how we managed active disease. Dr. Hirsch talked a lot about potential adverse reactions to this medication, watching for suppression and infections. But these drugs are highly effective. We notice an improvement right away. It was almost like, and they were much, much easier to tolerate than the previous high efficacy infusion medications. They did require, of course, MRIs. If patients reported lots of infections, we might take pause and look at their counts and maybe talk about extended intervals or investigate these infections. Patients are taught to monitor the number of infections that they have in a year when they come in for follow-up. And there may be an increased malignancy risk that was included right from the beginning, maybe a signal for breast cancer or may not be a signal for breast cancer. The bottom line is with these drugs that are working with an immune system, patients should always be on the alert and make sure they attend their yearly follow-ups and monitoring for their cancer checkups, including breast and cervical, as well as skin cancers. Vaccinations. All of a sudden, vaccinations became a very big part of what we do in a clinic in recommending and making sure our patients get vaccinations and get them at the right time, and I'll talk a little bit about that in a minute, because B cells blunt the effect of vaccination. Also, we don't want patients getting vaccinations, live vaccinations, when they're on B cell therapies. The next B cell to come to the market, ofatumumab, which is a subcutaneous formulation of a B cell therapy. It's humanized anti-CD20 as well. It's delivered subcutaneously once a month and appears to promote depletion of B cells in the lymph nodes, or so they tell us. Adverse effects are very similar to ocrelizumab, maybe increased infections, injection site reactions, but this is a 21st century injection with a beautiful auto-injector, and it's only given monthly, and we see much less of the problems that we dealt with those beginning DMTs where we were working with patients and begging them to stay on it, and it was hard to believe they would because of all their skin reactions. Again, headache, and again, the caution of immunization. And ubliximab, which isn't available to us in Canada, another infusible anti-CD20 that has a much shorter infusion time in the infusion room. Similar adverse events as are listed for ocrelizumab and ofatumumab. Herpes virus-associated infection, so antiviral medications may be employed even prophylactically. Again, vaccination is important with this B-cell therapy. So those are the lists of the varying classes of DMTs. There's some very important considerations, especially for our higher efficacy treatments. Immunizations, and the recommendations for immunizations vary from country to country. There are specific guidelines for those who are immunosuppressed for immunization. They're very important to consider. We don't want them causing a significant delay in treatment. Anti-CD20s particularly, they can blunt the immune system's response to vaccination, particularly to the mRNA-based vaccines. This was a huge issue during COVID. Lots of talk on immunizations during that, lots of, I think, bad press during that time. Following COVID, it was very hard in some cases to talk to patients for them to consider immunization prior to these therapies. Like other conditions where treatment can cause persistent immunosuppression, consideration should be given to the following immunization recommendations. We've got to make sure immunity to VCV, and we should draw serology. 2 vaccines are recommended if that's negative. That recommendation originated way back with fingolimod. It's important that there is chickenpox immunity. We screen for TB, everybody, not even high risk, where I come from, and with Mantoux skin testing or quantiferum gold. We definitely screen for hepatitis B and immunize if negative. And we consider immunizations as recommended by CDC, local public health, our own Canadian system of recommendation, and the National MS Society. It is hard. If a patient chooses to go on a B-cell in our clinic without adequate immunization, we don't refuse them, but we do make them, we not sign a waiver, but declare that they have been warned about the repercussions. And we're always on the lookout for major side effects. PML, very well identified with natalizumab, very well considered by the company that marketed natalizumab. It can be, of course, fatal, and we have to teach our patients about how to recognize some of these symptoms, and they need to call the clinics or go to a local emerg ASAP, and they do. I think we do a very good job of teaching them for these unusual presentations with any of the medications. And with our anti-CD20s, we can't forget about, there are serious adverse events reported as well. Enterovirus encephalitis has been described. It includes symptoms like flu-like symptoms, severe neurological changes. Diagnosis, again, is with CSF. Imaging may show T2 hyperintensity in the cord and substantia nigra or be normal. Management treatment could include IVIG, and in some cases antiviral agents. This has a high risk of fatality if untreated. However, early intervention can lead to recovery. It's not as clear on how we can monitor for this, given that the anti-CD20 therapies are certainly don't have a risk medication program like natalizumab did, and they're fairly new. But I think they can increase the risk of any infection, and we should teach our patient to be on the lookout for that and report immediately. Again, asking about infections has been written into our electronic health record. And so I'm going to close that. The B cells are really well tolerated, and sometimes we forget that there could be potential adverse reactions. Patients feel so much better than they have on any other medication. I think even before we put them on a medication, we should make sure that we screen for comorbidity, for compliance, for the ability to articulate what some of these adverse events could be. Vaccination, that we need to ensure patients adhere to laboratory and MRI screening and follow-up. Introduce antibiotic and antiviral prophylaxis much sooner than those that are on these treatments. And we may be looking at dose frequency adjusted. We may be looking at extended interval dosing. That's in its infancy as far as monitoring for that, but I think you'll see more and more of that. It's great that we have these high efficacy treatments. I, for one, who's been in this field for a long time, have noted and seen such great improvement in the outcomes of our patients. So I will leave you there. Thank you.
Sit here again. Let's see if we got some questions. All right, questions for Colleen? So even with the mitigated risks, we're still better off than we were 35 years ago, right?
Most definitely, yeah. If we put off with alemtuzumab, yes, we're still far better.
Okay, thank you, Colleen. Next up is Amy Perrin-Ross from Loyola University Medical Center, who will discuss the very important topic of symptomatic therapies. We throw this in because we pay a lot of attention, as we have today in other courses, to disease-modifying therapies and such. But for patients and their day-to-day lives, this is critically important. So Amy, welcome.
Thanks, Dr. Lublin, and I'd like to thank you for the invitation and Tim and Nicole from Academic CME for getting us started off on a good note. You know, when we were first discussing this session, Dr. Lublin was asking about symptomatic therapies. When I think about therapies, I think about drugs. So I decided to tweak it a little bit and just call it symptom management, because I think that this program is an excellent way to kick off the CMSC meeting. For those of you who are here for the first time, welcome. Please enjoy, absorb it all. But really, the focus of the CMSC is the comprehensive team approach to the management of people with multiple sclerosis. So what are some of the challenges in symptom management? Well, patient and family education is one of them. I think it was Dr. Berger who earlier this morning essentially said I'm older than dirt, because I, like him, have been around since before many of the disease-modifying therapies, and he's absolutely right about that. And yet the challenges in MS symptom management remain, both for the patients as well as for us as healthcare providers. What we want to do is we want to look at necessary information. We want to look at active participation. What we really want to do is focus guiding the patient towards self-efficacy. Ultimately, they're the ones that are at home. They're the ones that are managing their lives day to day. Sure, we as healthcare members can be resources, but we want to guide them toward self-efficacy, including things like coping, adaptation, self-confidence, many of those things. And so when Dr. Lublin asked me to cover symptoms, I thought, oh dear, how many symptoms can I get in a 20-minute talk? So I broke it down into two sections, two visible, two invisible, sort of, and we'll just kind of go from there. Again, much of what I am presenting today and the information that I have on the slides comes from many of my colleagues at the International Organization of MS Nurses and through the CMSE. So I do have to thank them for their information. So the first of the symptoms that we'll talk about is spasticity. Increase in muscular stretch reflexes and muscle tone. I know that there's going to be some good talks throughout the CMSC from our rehab colleagues. I know we've got one of them here, Patty Bobrick, who's going to be part of our panel discussion a little bit later. But we look at spasticity, we talk about tonic spasms, we talk about phasic spasms, and the fact that spasticity may worsen over time even in the absence of new lesions. Now, management really is individualized, and over-treating actually can result in increased weakness and impact other components of motor function. But people talk about tightness or pulling or tugging or aching. Sometimes they actually even refer to it as that MS hug. Often results from demyelination in the descending central nervous system pathways. Again, results in increased resistance to stretch, attenuation of the DTRs, uncontrolled flexor responses and extensor spasms, and ultimately excessive energy expenditure. People's legs, for example, that are incredibly stiff expend a tremendous amount of energy just getting from point A to point B, and may ultimately end up with some pain and discomfort. So some of the things that can increase spasticity, and I probably get 4 or 5 calls a week from my patients saying, I'm having a relapse, I need steroids. And I say, well, now tell me about that. Well, my legs are so stiff, they're spasming all over the place. I say, have you had your urine checked? No. Urine, why would I have my urine checked? Urinary tract infection, one of the most common causes of an increase, particularly in spasticity. Also ask them about things like where they're at, if anywhere, with the menstrual cycle and or perimenopause, things like other infections, restrictive clothing. These days the big wide leg jeans are back, or at least they were 6 months ago when I was out looking, and gone are the skinny jeans, the really, really tight ones. Those tend to be a problem for our patients with MS because those skinny jeans can be incredibly restrictive and may ultimately increase spasticity. So what do we think about managing spasticity? Well, for mild or minimal spasticity, we encourage things like stretching, physical therapy, and hydration, hydration, hydration. I can't say it enough. Mild to moderate spasticity, again, a good program of physical therapy, as I'm sure Patty will talk about, and additional potential medications, baclofen, tizanidine, gabapentin, and maybe some diazepam as second-line therapy. But if they have moderate to severe spasticity that may negatively impact their activities of daily living, we talk about a PM&R consult, we talk about the potential for Botox and maybe intrathecal baclofen. And I will tell you that pretty much every one of my intrathecal baclofen patients have said, why did you let me wait so long? They're afraid of it, they don't want to do it, they think it's intrusive, but once they do it and they can get rid of some of that oral baclofen, they're much, much happier. And again, for picture purposes, if you're interested, just some of the pharmacologic aspects of spasticity management. So move on to tremor. Obviously, cerebellum coordinates movements, and dysfunction caused by MS lesions can be seen in the cerebellar pathways. Dysmetria, tremor, nystagmus, ataxic gait, tremorous speech, titubation. I sometimes have patients who say to me, well, you know, we don't go out to eat much, we don't get out much. And why is that? Well, because my tremor is so bad, people in the restaurant think I'm drunk, or sometimes I spill my food or my drink all over me, or whatever. My speech sounds like I'm drunk. Okay, let's think about what we could do to assess and manage that. Again, rehab, PT, OT, let's talk about some speech therapy for these people. Weight-bearing activities, things like weighting of utensils and assistive devices. For example, plates with high rounded edges so that they can simply scoop something onto a spoon if they have difficulty stabbing it with a fork. Deep brain stimulation is another possibility, not something that we rush into, but it definitely is a possibility. And again, treatment of cerebellar dysfunction, multiple different things here. You know, we tend to try to stay away from things like clonazepam, but in some of our long-standing patients with cerebellar dysfunction, clonazepam is about the only thing that's going to cut it, so that's what we go to. Again, trying to tweak that as much as we can to impact their quality of life. Moving on to bladder dysfunction, well, there's a bunch of different types of bladder dysfunction. About 70 to 80% of patients with MS can experience bladder issues. Symptoms include the inability to store, which is an overactive bladder, or the inability to empty, which is an underactive bladder. And I would like to give full credit to my colleague and friend, Marie Namey, who was formerly with the Cleveland Clinic Mellon Center, who has a line that I use on a very regular basis with my patients. Marie was, when she was practicing, was one who really focused on bladder function. And her famous line was, if you sip, 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 you will pee, pee, pee. Right? Patients like, oh no, I'm going every 20 minutes. Well, it doesn't mean you shouldn't drink. It means if you got a 6-ounce glass of water, drink it in 5 minutes or so. And then you can empty your bladder 30 minutes later instead of running every 10 minutes to the bathroom. So inability to store, we see urinary frequency, we see incontinence, we can see a post-void residual of greater than 100 cc's. Inability to empty, again, urinary urgency as well as hesitancy, sometimes some double voiding. And again, we have to get our patients in the mindset of thinking about what they're doing. Most people just, your bladder's full, you go in the bathroom, you empty your bladder, wash your hands, you come out, you don't think about it. But we need to help our patients with MS understand that sometimes you really need to think about it to avoid, for example, that double voiding, to avoid the incomplete emptying and to try to impact nocturia. And then you could have a combined dysfunction, which is detrusor sphincter dysynergia. Say that 3 times fast with a mouthful of peanuts, right? Here's where we see urinary hesitancy, again, incomplete emptying. Here's where we potentially see some dribbling incontinence, and this is often diagnosed by urodynamic studies. So in the inability to store, limit fluid intake. That doesn't mean don't drink, it means drink conservatively. Do some time drinking followed by some time voiding when you have access to the bathroom. Decrease the use of bladder irritants, things like caffeine, aspartame, alcohol. Potentially Botox injections, pelvic floor exercises per physical therapy. And we look at the anticholinergic and the antimuscarinic, various different medications. And ultimately, in more severe cases, potentially the sacral neuromodulation. And with the inability to empty, we need to think about adequate fluid intake. and structured timed voidings. We may need to resort to things like intermittent catheterization, rarely an indwelling catheter, but there sometimes comes a point in our patients' lives where that's the thing that's going to make the most difference for them. Potentially the pure wick catheter for women, Botox for the bladder, alpha blocking agents, again, to promote the flow of urine. And finally, the fourth of the symptoms to talk about is fatigue. At this hour of the day, some of you may be feeling fatigue, especially some of you who, like me, was up at 3 a.m. to get here this morning. The fatigue that we see with MS is, when I'm talking to my patients, not really like, oh, I haven't slept all night fatigue. This is a subjective lack of physical and or mental energy that's perceived by the individual or the care provider to interfere with the usual or desired activities. It's lassitude, it's cognitive. I remember my daughter coming home from second grade one day, and I was asking her about what we were going to do about her homework, and she said, "Oh, Mom, I can't possibly. My brain is just too fatigued." I'll fix that, my friend. But the fatigue we see with the MS patients is truly that overwhelming sense of lassitude, and it could occur 5 minutes after they wake up from sleeping all night. If it's acute or new onset, it may be related to a relapse. If it's chronic, may last longer than 6 weeks. People often describe it as the most common and the most disabling symptom of MS, experienced by up to 95% of people. Not all people all the time, but 60% of people describe it as their most troubling symptom. And it's reported in all of the cases and the stages and the phenotypes and all of the things that Dr. Loveland covered so eloquently earlier. It's across the board. Now, there is some evidence that lesions in the basal ganglia and the hypothalamus may also play an important role. Again, thinking about the clinical characteristics, constant sense of tiredness, lack of mental energy, feeling of exhaustion, not necessarily related to level of disability or physical disability, but it really can be a significant cause of disability in our patients. When I'm filling out FMLA forms and when I'm filling out disability forms, it is a challenge to talk about the impact of fatigue on cognitive function and what that does to the person's ability to work full-time, part-time, whatever the case might be. It may not be ever fully understood. Thinking about fatigue management, exercise improves fatigue. That's one of the big challenges, trying to help a patient understand that they're so tired they ought to get out and move around more. Okay, let's get our physical therapy and our OT colleagues involved here. Let's address some of the secondary causes. Many of our patients truly do have sleep impairments, sleep apnea, for example. If it's undiagnosed and untreated, that's going to only add to the fatigue. So we need to look at the broad spectrum. Things like cooling techniques that the MSAA and the MSF have helped many of our patients with, stress management, and certainly pharmacotherapy. Everything from amantadine and modafinil to dalfampridine. We tend not to use a lot of the dextroamphetamines just because of the controlled nature and the difficulty in dealing with them. But again, looking for potential secondary causes, sleeping difficulties, anemia, depression, thyroid dysfunction, and so on. And again, just a couple of ideas here about the pharmacologic management. So finally then, again, from my colleagues at the International Organization of MS Nurses, effective symptom management is trifold. It's a multidisciplinary team approach that can maximize outcomes for patients. We need to remember frequent reassessment and adjustment in our evaluation of the symptoms. And not only do we as clinicians need to remember that, we need to tell our patients and teach our patients and family members to constantly reobsess and think about what might be impacting those symptoms. And then remembering that sometimes the non-pharmacologic interventions are just as important and just as necessary as the pharmacologic interventions. So on that note, I'll thank you and take any questions.
Thanks, Amy. Okay, questions?
This one? Okay. Yes, ma'am.
I feel like this is maybe a dumb question, but I see stress management listed a lot, and outside of telling my patients, you should try not to be stressed, which doesn't seem helpful. What kind of practical advice do you give for stress?
Well, it's different in every patient because what causes one person's stress could not bother somebody else. Identify what your stress triggers are. Identify what you need to manage those triggers. Like if your stress trigger is your house is a filthy mess when you come home because the kids have destroyed it after school and the kitchen's all a mess, work with your family to identify those issues and manage those. I mean, sometimes it's just as simple as working with identifying what your stressors are and working with your family to impact those stressors. I mean, we can't all avoid stress ever, right? So let's think about what stresses us the most. We can't have everything stressing us, right? But let's think about what the stress priorities are for us and work on them one at a time.
Other questions? Amy, thank you so much.
Thank you.
Now we're going to hear from Dr. Anthony Feinstein from the University of Toronto, talking about brain health and preserving cognition.
So thank you for the introduction. Very pleased to be here. My topic is going to be on cognitive dysfunction in people with MS and what you can do about it. The aims of the talk to understand the multiple negative ramifications of cognitive dysfunction in the lives of people with MS and to appreciate that for some people we can now treat cognitive difficulties. So how common is cognitive dysfunction? Well, it depends on your disease course. You can see that in clinically isolated syndrome, it affects about 1 in 3 people. The rate goes up to about 44% in relapsing remitting MS. When you get into the progressive forms of the disease, the rate's actually very high, 70, 80, 90% with primary and secondary multiple sclerosis. Overall, impairment rates of about 46%. The EDSS and age are also important determinants of impaired cognition in patients with MS. Even in people with radiologically isolated syndrome, we see a lot of cognitive difficulties. This is a study that I completed with my colleague G10 in Toronto, where we showed that about 1 in 3 people with RIS have global cognitive impairment. And I think it gives a lie to the notion that there's something called benign multiple sclerosis, because even if you've got no neurological symptoms, you may indeed have some significant cognitive compromise. So let me give you 10 key facts about cognition in people with multiple sclerosis. The first is, it's a predictor of what comes next. So this is a study of individuals with clinically isolated syndrome followed for about 3.5 years. Over that time period, about 46% converted to MS, but the conversion rate went up to 64% if they had failed 2 cognitive tests at baseline. And it went up to close to 90% if there were 3 cognitive tests failed at baseline. So cognition is extremely important as a prognostic indicator in people with MS. And in fact, when we look at individuals with relapsing remitting MS, if you've got significant cognitive compromise, it can also predict conversion to secondary progressive MS. You can see the odds ratios there of about 2.29. Cognitive impairment is also associated with an increased risk of mortality, particularly if you've got the progressive forms of multiple sclerosis. And these results hold up when you control for multiple covariates. We now know that you can get a cognitive relapse. So when you look at the data over there, you've got the group in green that's stable over time, and the group in red, you can see that the second time point, worsening happens. And on the y-axis, you've got symbol-digit modality scores, which in fact decline with the relapse. And as the individual recovers from the relapse, so the SDMT scores improve. And this is something that's not only linked to processing speed, it's also with respect to memory. In the top graph, you can see how performance worsens on the California Verbal Learning Test with the relapse and then improves with recovery. And the same thing with physical metrics. With the 25-foot timed walk, it gets slower with the relapse and then it improves. What's intriguing is that you might get an isolated cognitive relapse. And how do we know this? This comes about from serial scanning with MRI and gadolinium enhancement. You can see at the baseline over there, the green dot, follow the individual over time, these individuals over time, and when you see the red arrow, this is where you see GAD enhancement, and at that time point, there is no other neurological symptom. The EDSS doesn't change. But when you look at the SDMT, it goes down. So when you look at the numbers over here, you can see that from baseline to second testing, the SDMT scores decline from 54 to about 46. And then once the blood-brain barrier reconstitutes, once the blood-brain barrier reconstitutes, the SDMT scores improve. And you don't see concomitant change in fatigue or anxiety or depression over time. It's simply a cognitive relapse. What about brain imaging? So this was an interesting study done before the advent of MRI. It was a CT brain scan study done by Stephen Rowe, looking at ventricular size, the third ventricle in particular, and he defined 3 groups: normal third ventricle size, mild atrophy, and moderate to severe atrophy. And he looked at the association with memory with respect to word recall and objects recalled. And you see something really interesting, that as the third ventricle increases in width, so memory starts becoming more impaired. And indeed, with the group that has significant ventricular widening, you can see some very significant memory impairment. Now, this is a CT scan finding. So what's the importance of the third ventricle anatomically? Well, here we have MRI data. Patient A has relapsing remitting MS. Patient B has secondary progressive MS, and you can see how the third ventricle becomes increasingly wider as you move from relapsing remitting to a progressive disease course. And what's on either side of the third ventricle? We've got the thalamus, which has been hand traced. And so as the thalamus shrinks or becomes atrophied, the width of the third ventricle widens. And that becomes a surrogate for cognitive impairment. And you see this shown very nicely in a study by Ralph Benedict, where he looked at multiple MRI metrics, third ventricular width, brain parenchymal fraction, flare, lesion volume, etc. And it really was third ventricle width that turned out to be the most robust correlate of cognitive difficulties with respect to verbal memory, CVLT, visual memory, BVMT, processing speed with the PACEAT, and the symbol-digit modality test. And I think this is a really useful clinical tool. You know, when we get our reports back from neuroradiologists, they don't often comment on atrophy, and often you have to detect atrophy with detailed, computerized analysis of MRI, but looking at the third ventricular width can give you a rough and ready clue as to what's going on from a cognitive perspective. So it's a useful clinical tip. So the 6th point to put across is that cognitive impairment comes with real-world consequences. And we see here something called the Bartel Index that gives us various metrics. If you've got impairment, cognitive impairment, our patients can have greater difficulty with their daily hygiene, shelving food, opening containers, using utensils, using appliances, even making a bed. It can also impair driving. Processing speed, as measured by the SDMT, is a good predictor of a person's performance behind the wheel. If they measure spatial learning problems, this predicts collisions and traffic violations. And divided attention can also come with driving problems. By the way, this is a concern when individuals come for neuropsychological testing. Some patients are worried that if they're impaired, you're going to take away their license. But there has to be a discussion about the risks that come with impairment in relation to driving. Another real consequence is that individuals with cognitive impairment forget to take their medication. And there are many reasons why people don't take their medication. They may be tired of injections, for example. They may be fatigued. They may be depressed. But the biggest reason for poor compliance with medication is people simply forget. And if you've got cognitive impairment, that underlies their forgetfulness. Another real-world consequence of cognitive difficulty relates to employment. And these data are really interesting. If you've got an EDSS score of 0, then you're not going to find unemployment going up in people with MS. If you've got an EDSS score of 6.5, in other words, individuals on the cusp of having to go into a wheelchair, the unemployment rate is very high, around 82%. But when you look at individuals with relatively mild physical disability, an EDSS of less than 3, the unemployment rate is actually quite high, 54%, and that's driven predominantly by cognitive difficulties. We now have some thresholds that tell us when cognitive change becomes clinically significant. The simple digit modality test has emerged as a key cognitive measure in people with MS. And if there's a 4-point change, we know that this is clinically significant with respect to group data. And if there's an 8-point change on the SDMT, it's significant with respect to individual data. So these thresholds are useful, as I'll explain just now when we come to looking at rehabilitation studies. An eighth reason why cognition is important, well, if you've got good cognitive reserve, it predicts a better cognitive future. So how do you get cognitive reserve? By having an enriched lifestyle. If your education was good, if you did a lot of reading, if you engaged in intellectual pursuits, this boosts your cognitive reserve. Music, for example, can boost your cognitive reserve. Playing music, learning an instrument. But we can also boost our cognitive reserve from leisure activities. And I like this quote that came from a well-known Victorian, Samuel Smiles, who said that all work and no play makes Jack a dull boy, but all play and no work makes him something worse. So you want to find this, sweet spot between working hard and playing hard as well, because social activity is going to boost your cognitive reserve. And indeed, you see this very nicely in this graph over here, that as the disease burden increases, the slope of cognitive decline is much milder if you've got good cognitive reserve. It's protective. And so work with your patients in boosting their cognitive reserve by keeping them intellectually active, physically active, and socially active. What about cognition as a marker of function in a real world filled with fast-paced distractions? And this is one of the most famous quotes in all of psychology from William James. Everybody knows what attention is. And he goes on to say it implies withdrawal from some things in order to deal effectively with others. And it's a condition which has real world opposite. It's something that the French called distraction. So, James was saying this back in the 19th century. Think about life today with all its distractions. Email, Twitter or X, Instagram, etc. You're constantly being bombarded with stimuli. What Saul Bellow, the great writer, called the tyranny of distraction. And Bellow said this in the 1960s. You wonder what he would make of life today. So this is the environment that our patients live in. Well, we've shown that when you introduce distraction into cognitive testing, you can start bringing out deficits. So we developed an analog of the symbol digit modality test, a computerized test of processing speed you can see over there. Individuals are presented with a code of numbers 1 to 9, 9 different symbols, a buzzer sounds, and they have to match the symbol with a number as quickly as they can. When they get to the end of the line, it repeats itself 8 times. Embedded in this, we put distractions, a telephone ringing, a car horn sounding. And the bottom line is when people generally come in for neuropsychological testing, we put a sign on the door that says, "Quiet, please, testing in progress." But that's not the real-world environment. So we built in some of these real-world distractors into the testing process. Things like this. and we can control the volume and the duration of the distraction. So what do we see? Well, in healthy controls on the left, you can see that distraction, the solid line, certainly slows down processing speed. Processing speed is on the y-axis in seconds. But when you look at MS patients overall, their processing speed is, of course, much slower, but they are particularly susceptible to distraction. You can see how the two lines widen when you bring distractions into testing. So why is this important? Well, patients come to see us for cognitive testing, and we give them the typical battery in a quiet environment, and they do quite nicely. And we call them back in and we say, we've got good news for you. You've done nicely with your cognitive testing. And they say, but why can't I function in the office? Why am I struggling at work? And one of the reasons might be you're susceptible to distraction. You don't work in quiet environments. So let me just finish on what I think is becoming an increasingly optimistic note, that cognitive dysfunction can be treated. And the best way to do it is through cognitive rehabilitation. So this is a large study that we recently wrapped up, funded by MS Canada, looking at treating cognition in people with progressive MS using cognitive rehabilitation plus aerobic exercise. It was a multi-center study, 6 countries, 11 different centers, Canada, United States, United Kingdom, Italy, Belgium, and Denmark were involved, a big sample size of over 300 people. The primary endpoint was processing speed as measured by the simple digit modality test after 12 weeks of intervention. And just to remind you, so that the slides become a bit scrambled over here, a 4-point change is clinically significant in terms of group data. An 8-point change is significant with individual data. It was a 4-arm treatment trial. Some individuals got both aerobic exercise and cognitive rehab. Others got cognitive rehab and sham exercise. Others got exercise and sham cognitive rehab, and a group got a double sham. And the therapy went on for 12 weeks, 2 sessions a week, about 120 minutes of therapy per week. And the hypothesis was that if you give exercise together with cognitive rehab, you're going to get the best result. But unfortunately, our primary hypothesis was not supported. But what we did find was that patients got better either with exercise or cognitive rehab alone. At 12 weeks, at our primary endpoint, 60% of participants demonstrated a more than 4-point improvement on the SDMT, and 37% had a more than 8-point improvement. And 6 months after we completed the rehabilitation, 46% of individuals still showed a 4-point improvement, and 26% an 8-point improvement. So the bottom line is that cognitive rehab and exercise was effective in improving processing speed in this group of individuals with progressive MS with a mean EDSS of around 6.5. What does neuroimaging data tell us about cognitive rehab, giving us an understanding of the mechanism behind improvement? And this is a nice study done by Ralph Benedict, in which individuals got an MRI at baseline. They then had neuropsychological testing. They went through 12 weeks of cognitive rehab, and then the neuropsychological testing was repeated. And they looked at the simple digit modality test as an index of processing speed. They looked at verbal memory and visual memory. And in fact, the cognitive rehab showed an improvement, in particular with the SDMT and processing speed, but they also looked at their brain metrics at baseline. And when you had increased gray matter volume, your response to the cognitive intervention was improved. In other words, if you've got a better substrate to work with, you're going to get a better rehabilitation result. And you see this very nicely in the graph on the right. This is just some imaging data from our large COGEX study, where Mara Roca showed, she has a lab in Milan, she showed that individuals who got cognitive rehab showed an increase in the gray matter volume in the parietal, temporal, and frontal regions. And this wasn't associated with an improvement in processing speed. There was some spillover. There was an improvement in verbal memory. That was a secondary outcome in our study. Verbal memory improved in association with increased gray matter volume in response to cognitive rehab. And we're now at a point where the Cochrane Review, which is a pretty tough judge of things, is basically saying there's sufficient evidence to support cognitive rehabilitation when it comes to managing memory decline in people with multiple sclerosis. We've convinced the Cochrane group that this is an important therapy. And a study came out recently looking at all the computerized cognitive rehab interventions and basically showing that they're effective with respect to information processing speed and memory. I've given you some of the figures over here, but also for executive functioning. And when you look closely at the Cohen-D, the effect sizes, they're pretty substantial. You know, you've got an effect size of about 1.5 for processing speed and more than 2 when it comes to memory. That's pretty robust for the social sciences. And finally, the clincher is that people with multiple sclerosis like cognitive rehab. They like the computerized programs. They find them effective. It improves the subjective sense of cognition. They like a sense of community that comes from being involved in interventions like this. It improves their knowledge of what cognition is. They engage very well with therapy, and relapse and dropout rates are very low with this kind of intervention. So in conclusion, we know this, that cognition is frequently impaired in people with MS. Processing speed in particular is a sensitive marker. It's easy to detect. Cognition predicts neurological future and mortality. It's a major determinant of real-world functioning. It can predict employment, the ability to drive, and compliance with disease-modifying therapy. Cognitive reserve is a predictor of cognitive resilience. Cognition can deteriorate with relapses, and there is such a thing as an isolated cognitive relapse. Cognition correlates well with biomarkers of MS disease activity. They'd be detected from brain MRI, for example, and even neurofilament light. And in some people, cognitive dysfunction is treatable. So thank you for your attention.
Thank you, Anthony. Questions? Yell.
Sorry? Right. So, you know, there are two broad kinds. One is the part that's given by occupational therapists, particularly they teach cognitive compensatory strategies. The cognitive rehab, the cognitive remediation is much harder to find. But you do have these computerized programs now. The one that we use in our clinic is one called ReaCom. You purchase it through a company in Germany. It costs about $3,000. But they have specific modules for particular deficits. There's a module for processing speed problems, for memory problems, executive problems. And you can learn the program. It's not difficult. We've taught our occupational therapists to administer ReaCom to patients with impairment. And we bring them in for about 5 weeks of rehabilitation, 2 sessions a week. We think probably 5 weeks is sufficient. Each session is about 45 minutes. Most importantly, you can do it remotely. So you can give ReaCom by computer. They don't have to come into the clinic. ReHACOM, ReaCom. Yeah, and it's a German company called Hassomed markets it, and it's out in the public domain. You can purchase the program.
So Anthony, you said it helps in some people. How do you pick them?
Well, I think I showed some data over there. Oops, I showed some data over there that MRI can be very helpful. If you've got better MRI metrics to begin with, you are predisposed towards responding better. So, for example, a Ralph Benedict study showed that if you've got improved gray matter volume at baseline, you're going to do better with cognitive rehab. So that's a clue. We feel that if individuals fail multiple cognitive parameters, they're not going to do that well with cognitive rehabilitation. We tend to limit giving the rehab to individuals that only fail 1 or 2 parameters. So, you know, global impairment is probably not a good indicator for who's going to benefit from it. So between that and your MRI metrics, you can get a fair idea of who might respond.
Okay. Other questions? Anthony, thank you very much. Okay. We're supposed to have a break, which we will, but just for 3 minutes if you need to run out and do anything. Otherwise, we're a little behind time and we have a really nice session planned. So 3-minute break, and then we'll get on to the last session of the day. And I mean 3 minutes. Okay, we're going to get started again. Going the wrong direction, ladies. Okay, so for the end of the day, we're going to do comprehensive care with a couple of experts on comprehensive care. So Clyde Markowitz from the University of Pennsylvania and Amy Perrin-Ross are going to run this session, and they're going to introduce their faculty, who all should come up here now anyway.
Okay.
And this is interdisciplinary comprehensive care, which is key. So, where's Amy? Okay.
Thank you all for staying till the very end. Try and make this as painless as possible. All right, so am I projected yet? Not yet. Let's see what I need to do to make that happen. Well, you know what? There you go. There and there. Okay, good. So let me just start to tell you a little bit about our comprehensive approach. That's what this session's going to be on. Use the microphone there. I got it. No problem. All right. So comprehensive care requires a team approach. There's a lot of complexities with the treatment algorithms and monitoring. These are just an outline to discuss some of the things we're going to mention. Telemedicine, does it work for the MS population? Is it good enough? And is it financially viable for a MS neurologist to exist in today's world? Current challenges with treatments and unmet needs. And what about AI? And I don't know what to say about AI. I'll just tell you that it's a big deal. I'm not going to go through this. You've heard a whole afternoon of issues related to diagnosis. Joe, come on in. Have a seat. In any event, but it's a lifelong process. It's very complex, a lot of heterogeneous clinical features. And, there's a considerable amount of neurodegeneration that occurs, and we have yet to be able to solve that completely. But when a patient gets diagnosed with MS, they may or may not have ever had a doctor interaction. Maybe they had a primary care, a pediatrician. But now you're diagnosed with MS, and you're going to be meeting all of these people in your life, and that may be, the neurologists, nurse practitioners, social workers, physiatrists, nurses, et cetera, and it just keeps expanding out based on your clinical symptomatology. So, in today's world, the keys to be able to deal with comprehensive care is being able to have a team approach, having multiple people interacting with this patient, whether it be the pharmacists, the nurses, the admins, et cetera. And there's a lot of components of things that help manage this patient. That includes patients' families, interacting with the care team because they may feel embarrassed to talk to one person on the team, but okay to talk to this person, and that you kind of develop it as a comprehensive approach. I'm not going to spend too much time discussing this because I think all of these issues will be addressed by multiple speakers after me. But there are a lot of factors that play into what we decide for a patient in terms of treatments and issues related to routes of administration, pregnancy concerns, formulary restrictions on these medications, and all this kind of thing. So I'm just going to mention this because I think this is really the ideal MS center structure, where you're going to have physicians, nurse practitioners, maybe fellows, pharmacist, social worker, a research team. And there's going to be a lot of information that's going to be gathered when the patient comes in to be seen. And that's going to be questionnaires that we would like to have the patients fill out before they come in, go over their medications, screen them for things like depression, fall risk, et cetera. And then when you think about what you need from a physical standpoint, you need the space to be able to accomplish all of the people that you are going to have on your team. You're going to have an intake, and you may have a team of people who are MAs who are going to be checking things like vitals and et cetera. But then you need places to see the patient. You need consultation rooms for all of the other people because you can't have one room. You're going to need multiple rooms to be able to accomplish this. Things like a 25-foot timed walk. In our clinic, we set up a hallway where we put in these green squares that are 25 feet, and this way you can time them and be able to accurately capture that information. Places to do the MSFC. Places to do telemed for the patients who prefer to do telemed visits. Phlebotomist on-site is ideal. Place to review all the imaging studies. Most of the time it's on a computer these days, but having, better monitors to be able to, discuss it with your colleagues, et cetera. And places for the other consultants to come to visit and see patients with you. And that may be the physical therapist, maybe physiatrist, urology, et cetera. So that's all I was going to do. I'm going to bring up the next speaker, which is going to be Patricia Babrick. Is that correct?
Close, Bobrick.
Bobrick, okay, from UC Health in Steamboat Springs, Colorado. One of my favorite places to go ski.
I know, it's amazing.
Yeah. So, and we'll have question and answer after.
Thanks for hanging in there, everybody. I know it's been a long day for you. Don't worry, this last panel discussion is going to be kind of like speed dating. So if you're not interested in a topic or you don't like what I'm saying, I'll be sitting down in 7 to 10 minutes. So any of these topics that we have right now could be a day program. I get the joy of talking about wellness in MS and the multidisciplinary approach to that. To define wellness, and we usually think about wellness as our patients particularly think about the physical aspects of wellness and health. So if they feel ill or they have symptoms, they feel that they're unwell or unhealthy. And I think one of the things that I try to emphasize collectively is that you can have a diagnosis and still be healthy and well, even in with a diagnosis of MS. You can have neurological impairment, but we're maximizing capacity in our wellness strategies. Wellness domains are all of the things: physical, emotional, social, occupational, and intellectual. I would love to talk about all of these in the short time I have, but I'm going to zero in on several of them. Unfortunately, I won't talk about all of them, but we do know the importance of all of them. And I am always, from my entire practice and every time I talk, I'm talking about early intervention, being able to have the patient have strategies given to them have education given to them that they can incorporate into their lifestyles immediately. Many of our patients are coming to me or coming to us as the team and saying, I feel so out of control. I'm taking my medicine, but what else can I do? And this is a lot of what else they can do. I'm calling these the pillars of wellness. They're pillars of wellness. There's more than these, but I want to focus on these. And again, it's just kind of brushing over the surface. Since I'm a physical therapist, of course, I put physical activity and exercise at the top of that list. We just saw from the last lecture that exercise can have a positive outcome on our cognitive ability in our patients. But there's strong benefits across all the wellness domains with exercise. We can have decreased fatigue, decreased depression, decreased pain, improve mobility and function and independence and participation. Exercise has been shown to have an anti-inflammatory effect, and it releases all those wonderful neurotransmitters and chemicals in our brain, which actually help with skill acquisition and embedment of skills. So something like BDNF, brain-derived neurotrophic factor, which is released during exercise, can really help with skill acquisition. So, and that's just one example of many as far as exercise is concerned. In the references that I have listed, if you don't know where to start, and the question is always where can I find a physical therapist or a team of rehab professionals that know about exercise, there's some really good lifestyle and exercise recommendations that were put out by the National MS Society, endorsed by the consortium several years ago. So that's a starting point. Or you call me and I try to find you somebody in your locale. I want to just jump off on Amy's talk about hydration. I'm going to talk just a little bit about nutrition and hydration, but I just want to jump on the bandwagon with hydration. Our muscles don't function well when we're dehydrated. Our bladder is messed up. You're either going to be super concentrated in your urine and it's going to be an irritant. You run the risk of bladder infections then, and we don't have as good cognition when we're dehydrated. So hydration is really high on the list. Our patients often ask us for that MS diet. There's no true MS diet. What we're recommending may be similar to something like the Mediterranean diet, where you're limiting ultra-processed food, eating whole foods, limit saturated and trans fats, added sugars, and alcohol. And I did list here complementary and alternative medicines because our folks are asking about them. So you want to be well versed in what is acceptable and what the research shows as far as things like cannabis and acupuncture and those types of things, which may also augment their overall wellness and health. Sleep is an important pillar of wellness. Sleep is very restorative. If we're not getting good quality sleep, again, altered mood, cognition, physical performance, all of those are declining. Sleep disturbances has a high prevalence in MS patients, up to 50 to 60%, whether it's true insomnia, fragmented sleep, spasticity, which is impairing their sleep. And all of these symptoms and poor sleep patterns are bidirectional. So you get poor sleep because you're having pain or spasticity or nocturia. And then that adds to your daytime fatigue and your daytime cognitive dysfunction. So being able to address those things on a more global level, not just that you're sleeping poorly, let's medicate for that. Maybe it's a matter of we need to manage your spasticity better. Maybe we need to manage your bladder better. Mental health, of course, is a factor in everyone's wellness. Depression and anxiety are 2 to 3 times more prevalent in MS than in the general population. When you are depressed or have poor coping mechanisms or high anxiety, it's demonstrated that you have greater fatigue, reduced physical activity, worse cognition. You can see how all of these symptoms are kind of repetitive of like you're not doing well in this area. It's going to affect these symptoms that you're having. Call it the swirling toilet. You get poor sleep or you don't have good nutrition, you have less physical activity, and your symptoms, your MS symptoms, just continue to kind of spiral. I wanted to talk about self-management because many of our patients come to us and they don't have a primary care physician. They feel that they're having so many appointments that are related to their MS that the neurologist and the neuro team now becomes their primary care, and really kind of emphasize that they need their primary care, they need their annual physicals, they need the dental exams. People with low-grade infections in their mouths can have worse MS symptoms just because of that. Encouraging well women visits. And there were 2 points of lifestyle factors that I wanted to talk about, touch upon, smoking and obesity. We know that individuals who smoke have a greater risk for MS. It may be as high as 50%, and it's dose dependent. The more cigarettes you smoke, the higher risk you have for developing MS. And we do know that secondhand smoke in children also increases their risk for developing MS. Smoking in itself can cause greater disease progression, greater disability accumulation. So that's one of the factors that if we can encourage smoking cessation in our patients, that may have a positive effect on their disease outcomes. Weight management, obesity is associated with increased MS risk, particularly in childhood and adolescence. So somebody going from a BMI that's considered overweight to obese, their risk of developing MS is 41% higher. And people who have a higher BMI and a diagnosis of MS also have greater disease activity and worse functional outcomes. And if you just think from a mobility standpoint, it's harder for people to move when they're overweight. And just to touch on brain health, all of these positive lifestyle changes really are important for our overall brain health, and that physical activity, this cognitive engagement, occupational engagement, social engagement, all of those things add to our brain health along with exercise and decreasing cardiovascular risk. So in this short time, I just wanted to give you some of my final thoughts, that this is a very multifactorial topic, and the outcomes are not only shaped by the disease activity, but modifiable lifestyle factors. And again, I'm always preaching the earlier the better. If you get your whole team, your comprehensive team on board earlier, no one person can handle every issue that may be coming up or every educational piece that's needed. So having a whole team of people to kind of work on all of these different areas is really the most beneficial for the patient and for the team themselves. Targeted lifestyle interventions early on can give us better long-term outcomes along with DMT. It's not in place of, it's complementing the DMT management and other pharmaceutical symptom management. So be proactive, support wellness and brain health in our patients. And I'm going to turn it over to our next speaker.
All right, let's move forward. All right, so we are going to hear from Dr. Ali Bateman from Western University.
Yep.
Okay. There you go.
Thank you again for sticking around. I'm Ali Bateman. I'm a physiatrist from London, Ontario, and I'm really thrilled to talk to you about what I've learned in caring for persons with advanced MS in our multidisciplinary clinic. I have a few maybe lofty objectives for the 7 to 10 minutes, but we'll see what we can do. And I'd be remiss if I didn't mention that a lot of the, especially the statistics that I'm going to talk about, but a lot of the knowledge that I've gathered has come from working in a multidisciplinary clinic, and some of this research has already been published, but we have a program called the Care for Advanced MS program, in which patients receive a dedicated appointment or multiple appointments for symptom management. And I'm really grateful to our neurologists who have included me in this program, as it's made a huge difference for the patients that we care for and for us as providers in terms of how enriching it's been. So persons with advanced MS have a high symptom burden. I don't need to tell any of you that. You all know that MS is a progressive and for now incurable disease. But the burden of symptoms can, and studies show, is probably analogous to the burden of symptoms in persons with metastatic cancer. And we know that many people will not have adequate symptom control. And as a result, CMSC in 2019 issued recommendations that patients should receive comprehensive and palliative care and should receive a dedicated visit in order to address their symptoms. We've done that in London, Ontario, which I affectionately describe as being halfway between Toronto and Detroit. This is the first clinic of its kind in Canada, and we started it back in 2019. And so in its 7th year, we've learned a lot from what we're able to offer individuals in this program. The Care for Advanced MS program, or the CAMS program, aims to improve the quality of life of persons with advanced disability living with MS in our community. And it's a one-stop shop, or at least we like to think of it like that. Patients and their caregivers, or whoever they would like to have come to the appointment, they sit down with 3 physicians, an MS neurologist, a neurologist who specializes in palliative care, and a physiatrist, me, and we talk about what it is that the patients want to address. And I've learned in 7 years here that no 2 patients have the same needs, symptoms, or solutions, but that we often have a lot to offer in these dedicated appointments. In terms of the symptoms patients are most likely to experience, most often patients have altered mobility, neurogenic bowel and bladder, spasticity, pain, and cognitive impairment. Patients have a whole host of symptoms. Some have more than one. Some have just one that they want to talk about. But what we've learned is that there's this sort of suspected high symptom burden is in fact real. We have patients coming to us who all have a primary care provider. They all have an MS specialist. They're often connected with other specialists. And yet, at these appointments, we change management 96% of the time. So we're trying to address at least one symptom at 96% of appointments. And most often that relates to pain, spasticity, bowel symptoms, and bladder symptoms. And how we do that varies from person to person with a highly individualized approach. For a physiatrist, these are pretty bread and butter problems. But if you're looking at that list and thinking, like, I don't want to manage any of those things, or I don't feel that I can, or I'm not good at it, there is some guidance from MS Best, and the Paralyzed Veterans of America MS guidelines are also helpful for each of those topics. And there's some good learning in other sort of analogous conditions or similar conditions, particularly spinal cord injury, for the management of these things. Other recommendations that we've made include increasing publicly funded in-home supports, increasing our referrals to our allied health providers, which I'll talk specifically about. We've made a lot of physician referrals from our program, often to urology for the management of neurogenic bladder, but also to plastic surgery for wound management, plastic and orthopedic surgery for surgical spasticity management, and neurosurgery for intrathecal baclofen considerations. And I really want to emphasize that at 1 in every 5 appointments, we deprescribe medications. So we reduce the dose or we discontinue a medication altogether. And I think there is a huge missed opportunity in persons with advanced MS for us to be reducing the burden of medication and polypharmacy while also improving their symptoms. In terms of what we ask our allied health providers to do, I've highlighted social work, speech and language pathology, occupational therapy, and physiotherapy here, but we refer to all kinds of allied health providers outside of this scope as well. The two things that I'd like to emphasize: if you have access to an occupational therapist that can go into a patient's home, they're probably most likely to be successful at identifying devices, therapies, or strategies that might improve activities of daily living or instrumental activities of daily living and provide a lot of functional independence that way. And for physiotherapy, we often think a lot about physiotherapy in our ambulatory patients, but for patients that require assistance for transfers, physiotherapy can be really invaluable for teaching patients and their caregivers how to safely transfer so that the patient is safe, but also so that the caregiver is safe. And finally, in this clinic, but also in my own physical medicine and rehab practice, I get a lot of referrals for gait optimization, which means a lot of different things to a lot of different people. Gait is really complicated. I love it. I could talk about it all day, but I won't because I've got 7 to 10 minutes. But there are a lot of moving parts, literally and figuratively. If you take nothing else away from this, spasticity is often blamed for gait impairment, rightly or wrongly. But if you try treating somebody's spasticity and you make them weaker, then spasticity might not be the problem. Similarly, patients often describe stiffness when they mean weakness. And weakness is not something that we can medicate, and it's not something that I can inject. And treating weakness requires like a whole different thought process about what you're going to do. And I've highlighted a couple of those things here. So some weakness will have restorative potential or rehabilitation potential, but some won't. And some problems with gait won't be treatable with a specific medication or one intervention, but there are other things that we can do to help patients restore functional independence. And I really want to highlight that things that contribute to gait dysfunction can be polypharmacy. So if we're over-prescribing medications for spasticity in particular, we can be adding to people's weakness. If we're not thinking carefully about what medications we're prescribing, we may also be adding to patients' fatigue unnecessarily, and that can also reduce their ability to walk or transfer safely. And then there are lots of things that add to pain, weakness, and spasticity in a person with MS, like temperature regulation, constipation is a huge one, bladder dysfunction, and if we can treat some of those problems, we may be able to restore some function in patients without specifically addressing the spasticity or the weakness that we think is the problem. And I'll leave that there. I'm happy to answer questions at the end, and I'm going to transfer over to our next speaker.
Okay, next we're going to hear from Kristen Crisco from the University of Toronto about family planning.
Thank you. So thank you for the invitation and thanks for sticking around and including women's health as a topic. So because it's such a short time and a big topic, I wanted to highlight 5 key points that I want you to know after this, and then we'll go through each of them with some supporting evidence. So my first point is do not undertreat women with MS due to potential pregnancy planning. Second, beware of rebound with stopping certain DMTs that we heard about earlier in the day, and this does apply also to pregnancy planning. Do not automatically stop DMT when a woman tries to conceive. Fourthly, most pregnancies in women with MS are not high risk. And finally, women do not need to choose between breastfeeding and starting DMT postpartum since there are several breastfeeding compatible DMTs. So first, not undertreating women with MS due to potential pregnancy planning has been really an important message that I've been trying to raise with an international group. And we wrote a paper in Lancet Neurology in 2023 discussing this, which is a review on family planning, and we're working on new guidelines for family planning in MS through ECTRIMS, so more to follow. But this is a study from France where they compared women in their MS registry to men in terms of which DMTs they were given, and this is outside of the pregnancy period. And you can see in the top row, DMTs were less commonly prescribed to women, about an 8% lower odds of a woman being treated with DMT. And then almost alarmingly, it's even worse for high efficacy DMTs, where it was a 20% lower odd of women being treated with high efficacy DMT. If we look at the individual DMTs, some of the patterns are reasonable, like we see more glutathione and fumarates used in women, which makes sense because these are more pregnancy compatible compared to teriflunomide or S1P modulators, which were less commonly used in women. But they did see that anti-CD20s were actually less commonly used in women, even though this is one of the preferred approaches for family planning. So in this review article, we really wanted to highlight that there are not only risks of continuing treatment, but there's actually risks of stopping treatment too. And so if we think of risks of continuing treatment, of course, we want to think about the infant and the potential effects of treatment exposure in terms of teratogenicity and immune system effects. We also need to think of the mother and potential risk of dual immunosuppression by both pregnancy as well as their DMT. But there are, of course, benefits on disease control to continuing DMT, and then there are risks of stopping treatment. So if we look to the bottom right, there are risks to the mom in terms of disease control, risk of rebound, long-term disability, effects on well-being with that worry about a potential relapse, and there are risks to the infant of stopping the mother's DMT, including the impact of maternal disability or the need for relapse treatment during pregnancy. And then coming to the second point, we need to be aware of rebound with stopping certain DMTs. So we'll bring you back to the issue of fingolimod and natalizumab and any cell trafficking medication, that there can be an increased risk of relapse after stopping the drug. The figure on the left shows the typical relapse pattern around pregnancy. If we look at the line in light green, which is no DMT, pink, low efficacy DMT, and blue, dimethyl fumarate, we see the typical pattern of reduced risk of relapse during pregnancy, especially the 3rd trimester, but at heightened risk postpartum. If we look at the line in orange, which is fingolimod, or teal, which is natalizumab, we see a different pattern where we see in patients discontinuing these medications for pregnancy, very high risk of relapse during pregnancy and especially postpartum. And then on the right is a nice study from Germany where they looked at natalizumab discontinuation for pregnancy and the impact not only on relapse risk, but also on disability. And they found that 10% of patients had clinically meaningful disability that affected ambulation from a pregnancy-related relapse after stopping natalizumab. And fortunately, we now know there are better strategies. And so this is data from the MS-BASE group, which is an international study, and they looked at different strategies. So for instance, in purple is the natalizumab strategy if it's stopped, where there's a really high risk of relapses in pregnancy and postpartum. And we can contrast that to natalizumab active strategy, which is in blue, where we see very few relapses. And so natalizumab, if it's continued through the pregnancy until the 3rd trimester and resumed early postpartum, we're seeing much better disease control. And then newly in this study was data for ocrelizumab, where using anti-CD20 therapy before conception, pausing treatments during pregnancy, and resuming postpartum in most patients typically resulted in really well-controlled disease. And you can barely see the green line because there were no relapses during pregnancy and less than 5% risk postpartum. So that brings us to our third point. Do not automatically stop DMT when a woman tries to conceive. So we have many DMTs that have emerging and increasing data for safety with use around pregnancy, and the recommendations are evolving over time. And so some of the recommendations in our 2023 paper will change with our current work with the ECTRIMS group on a pregnancy guideline, and we don't have those yet. Those are under development. But from our core group discussions and a workshop we've had recently, these are the current likely recommendations. So interferon and glatiramer have a ton of pregnancy safety data and can safely be continued through pregnancy and breastfeeding. And the benefit of continuing those drugs for a patient who are stable on them, if we continue through pregnancy, it may continue to have an impact during that higher risk postpartum period compared to when it's stopped and resumed. There may be a therapeutic lag during that really important window of high risk of relapse. Dimethyl fumarate has data for early exposure in pregnancy that appears quite safe with no increased risk of miscarriage or malformation. And so this allows women who are stable on dimethyl fumarate to continue the medication while they're trying to conceive. But because we only have data for early first trimester exposure, they should discontinue when they become pregnant. Then moving to the monoclonals, so natalizumab we heard earlier, and I just mentioned it cannot be stopped for pregnancy without an alternative strategy. So if a woman does want to continue natalizumab and not transition to something else before pregnancy, they should continue it, generally extending the interval to every 6-week infusions, with the last dose around 30 to 34 weeks gestation, with an early resumption postpartum, usually within 1 to 2 weeks postpartum, trying to kind of target no more than 8 weeks between infusions to reduce that risk of rebound. Importantly, with this third trimester exposure, there will be crossing of the drug across the placenta and fetal exposure, and so the infant needs to be tested for anemia and thrombocytopenia. So here we need to involve the pediatric colleagues and alert OB of this. The anti-CD20s, as I mentioned, are really emerging as a very good strategy for use around pregnancy. We don't have time to go into all of the data, but there's been a lot of data in recent years, and I'll be presenting some data for ocrelizumab later at this meeting. But the current ECTRIMS group is most likely going to recommend no washout, which is a change in practice. So we recommend not infusing during pregnancy, and up until recently we recommended a 1 to 3 month washout from their last infusion before conception. But based on emerging pharmacovigilance data that shows use closer to conception does not seem to have any increased risk of miscarriage or malformation, as well as based on some prospective data showing no neonatal B cell depletion in women who had received ocrelizumab in the 3 months before conception, as well as some of the pharmacodynamics of the drug and pharmacokinetics. Most likely we don't need a washout. And the goal would be to infuse at the time of their menstrual cycle so they're confident that they are not pregnant, and then they can continue trying to conceive. For those that are potentially exposed, so if an infusion is given during pregnancy, inadvertently in the first trimester, or if it's given later on purpose when they know they're pregnant, this is usually avoided, but if that does happen, the infant should be checked for their B cell count to see if they're depleted, especially prior to any live vaccinations, which typically occur around 2 months of age. And if ocrelizumab is given shortly before conception, it's kind of a risk-benefit discussion if you want to have the infant checked for those markers. Ofatumumab has a somewhat shorter half-life, so it is eliminated by about 3 months from the last dose. And we know immunoglobulin G levels don't cross the placenta during the first trimester. And so this can be continued when someone is trying to conceive, typically stopping when the pregnancy is confirmed. And for patients who are tracking their cycles and are motivated, they can time their monthly injections with their menstrual cycle to try to avoid taking a dose in early pregnancy. But if there were a dose in early pregnancy, such as like 4 to 5 weeks of gestation, typically that would not have a negative impact since the drug would be eliminated by the mid-second trimester. Again, if there is potential concern for exposure, the infant can be tested for their B cell count. Next, so most pregnancies in women with MS are not high risk, and this is important because routine obstetrical care can be provided to these patients. They don't typically need additional ultrasounds or tests during pregnancy, so we can reassure our patients regarding this. I think it's important as neurologists that we bring up that mode of delivery is generally guided by obstetrical indications. It should not be altered due to a diagnosis of MS, although there is data to suggest a slightly higher risk of induction, forceps delivery, or C-sections in women with MS. And then epidurals and spinals and any form of anesthesia can be used in women with MS, and it's important for us to also bring that up. Some studies do show a slight increased risk of preterm delivery in small for gestational age, and this requires further investigation, although typically we're not seeing the very small or the very preterm deliveries. It's just a mild difference from the general population. Finally, women do not have to choose between breastfeeding and starting DMT postpartum. So again, there's a ton of data that has emerged for safety of medications, and many are lactation compatible. So the platform injectables are compatible with breastfeeding. Interferon is measured in very small quantities in breast milk. Glatiramer is broken up into its amino acid components, and both are felt to be quite safe. The trouble is these don't work quickly, so they're not ideal to start during that high-risk postpartum period. They may not help with relapse reduction early on, but if someone is stable on these medications, they can simply be continued during that period. For the oral DMTs, we have very limited data for dimethyl fumarate. There are a few cases where very low transfer into breast milk, which is quite reassuring. And there's also a small series from Italy with good outcomes for a small number of infants who are potentially exposed through breastfeeding. And so this medication, this is another potential change that most likely it is likely safe to breastfeed with dimethyl fumarate, and we'll see what comes with the EXTROMS guidelines, but I am counseling patients that this is likely safe. All of the other oral medications are not felt to be safe while breastfeeding and should not be started during lactation. But if we look to the monoclonal antibody therapies, these have excellent safety data for breastfeeding, and there are other monoclonals in many different conditions in GI and rheumatology, as well as in the MS field, that can be safely used while breastfeeding, including natalizumab and all of the anti-CD20 agents, which have low transfer into breast milk, low oral bioavailability, so even if there is a small amount in breast milk, it should not cause issues, and reassuring B-cell data in infants exposed potentially through breastfeeding, so these can all be used while breastfeeding. Typically, we like to wait until the mature milk, which is about 2 weeks postpartum for the anti-CD20s, but natalizumab, as I mentioned, the risk of having a long interval between infusions is too high to wait, and so they should definitely be started earlier to make sure there's no 8-week gap between infusions. So this is the 5 key points I wanted to raise about family planning and MS, and I can refer you to this 2023 paper as well as the forthcoming ECTRIMS guidelines. Thank you.
Okay, and our last speaker will be Amy Perrin-Ross, and she will talk about shared decision-making.
Great. I'm bringing up the rear with a couple of words about shared decision-making, and I think hopefully this will tie together some of our discussion. Shared decision-making occurs when a healthcare provider and a patient work together to make a healthcare decision that's best for the patient. And I think about what you said, Ali, and I think about what Patty said, and I think about my patients who want physical therapy, and they want me to continue to order physical therapy, and they do it until their sessions run out, and then they don't do anything until they decline, and I have to order it all over again. Think about those things. Optimal decision takes into account evidence-based information about the available options, as well as patients' experiences, values, and preferences. 5 steps of shared decision-making, obviously engaging patient and family, caregiver, care partner participation. Explore and compare treatment options. What are your options? If your insurance runs out and you no longer can get physical therapy, can we do this kind of stuff at home? What are your options in terms of DMTs if you're seriously considering getting pregnant in the next 6 months? I think pull all of this together. Assess the patient values and preferences, reach a decision on treatment or management with the patient, and then evaluate and continually reevaluate the patient decision. Comprehensive team approach, patient obviously at the center, We've talked a lot about the team, and I think that's part of what our role as panelists are here this evening, is to finish up and talk about the fact that there are a multitude of factors that contribute to optimal management in MS, overall outcomes, a multitude of things. And so for that, I will thank you and turn it back to Clyde for some discussion.
All right. Well, so we'll just open up some general questions to anybody in the audience who has any specific questions. I will start by asking this question, which is, how do you convince a patient to do lifestyle changes when they come in and you're talking about their diet, smoking, etc., and they tend to be somewhat resistant to making changes? Any good strategies you can suggest?
Well, I think the first thing is that you have to read the room, right? What are they going to be able to hear? And sometimes they can't hear very much. So you have to really modulate what you're able to give them. And then I'll give them information and I'll say, how do you feel about that? And they'll be like, smoking is relaxing to me, eating is, my coping mechanism. And so we might have to leave it lay for a little bit. And then we try to approach it again. So there's never a successful way to shove something down somebody's throat. But you get a great number of people who do have a lot of interest in what additional things that I can do in my life to make a positive change that would have a positive outcome on my MS. Again, it is a team effort here. We all need to be talking the same language. So if they just hear it from me, or they just hear something slightly different from one of their other healthcare professionals, that's going to lose their confidence in what the information is going to provide. So the team talking exactly the same language, providing the same information is also very helpful.
I'll open it up to the rest of the panel to weigh in on this one.
I'll just add that sometimes patients are really motivated when they're able to come up with reasons why something is valuable to them. So, for instance, patients who would benefit from drinking less alcohol, who have a lot of bladder symptoms, linking those two things together for them because alcohol is a bladder irritant can be really helpful. Or patients who are interested in losing weight and haven't identified some of the ways that their lifestyle could be contributing to that, particularly when it comes to alcohol use. That's another way that you can tie it in. And so figuring out what's motivating the patient can also help you get some buy-in. But most of the lifestyle recommendations are easy to say and difficult to do. So also important to acknowledge when patients have tried to make steps towards some of those lifestyle goals or recommendations. Because again, it's easy for me to say you should quit smoking, and it's really hard for an individual to actually do it.
And having concrete resources for them. Like you said, it's easy to say stop smoking, but if you don't support them, if they make a decision to start moving forward on something, you want to make sure that you have some resources for them.
And the resources might be something that you would prescribe, or would you refer them to their primary care doctor for smoking cessation type of things?
It may be all of the above. It may be support groups. It might be primary care. It might be another MS patient that I know who quit and could offer support that way. It just depends on the person.
Okay. Amy?
Clyde, I think here, again, we look at the comprehensive approach. If you've got somebody who's a smoker who lives in a house full of smokers and overeaters who don't actively exercise, my focus on somebody with MS is going to be a drop in the bucket. I need to help work with that patient to identify what their priorities are for their health. They're the patient, they're the ones with MS. What are your priorities for health, and how can I help you work within the circumstances that you're living?
Okay.
And it's ultimately their choice.
Yes, it is. But we have learned over the years the impact of, like cigarette smoking and the progression of the disease, et cetera. So it's our job to communicate that, but maybe do it in a way that the patient can hear, which is not easy. I'll ask another set of questions. This is more related to the pregnancy and breastfeeding conversation. So historically, we have been recommending patients who are on a B-cell depleting therapy to, after they get their infusion, and this is a planned pregnancy conversation, so they're going to start on, they're on treatment and they want to have kids in the near future. So we would say, why don't you get your infusion, wait about a month before you start trying, sometimes 2 months, depending upon their anxiety level about it. And then you can start trying. And that has been fairly successful. And then we talk about their breastfeeding issues, and we usually say, don't breastfeed right away. I mean, you can breastfeed right away, but don't get your treatment right away. Wait about a month or so that the milk gets somewhat mature, that you can then start breastfeeding. I mean, that you can then get your treatment and continue breastfeeding. Any thoughts or approaches that you suggest about that?
Yeah, so like in light of the new data that's come out recently for ocrelizumab, this is where the ECTRIMS group is probably going to say we don't actually need a washout, and I think that's even easier for patients. They actually don't have to skip any months of trying. For some patients who have just started trying to conceive, like they may be okay missing a month or two, but for patients suffering infertility, skipping 2 months, maybe twice a year of trying can actually be substantial for that patient. So I think that will help, but I think it's really about informing them about the data. Obviously, the label is different than the data, and hopefully there'll be future label changes. Like in Canada, it was just approved for use while breastfeeding ocrelizumab. The label has changed, so that actually helps a lot to say now the label actually, validates this advice. But I think it's really informing them, empowering them that they can treat their MS and still try to get pregnant and be on an effective medication. And the nice thing with the anti-CD20s is you get that pause in treatment during pregnancy itself, so they know they're not taking the medication actively while they're actually pregnant, and I think that helps also kind of make them more comfortable. But I think it requires a lot of counseling, and we need to be consistent between team members. That's kind of something that was talked about in the context of other issues. Like if their obstetrician is saying something different, then that's an issue. So I think we need to educate and be aware of like which kind of providers are going to be more actively treating MS and going along with our recommendations. Same with pediatricians. Usually they're not worried about most medications while breastfeeding. Like we're the ones who are shying away from it. So having their buy-in and like that discussion helps.
And do you ever think that there's a need for a distinction for like a different B-cell depleter like Briumvi? When it first came out, there was a concern that you needed to have a pregnancy test done before you would go ahead and infuse patients.
So we don't have that in Canada right now, but I would say it's very similar in terms of the half-life, and we think a lot of the effects are like class effects with the anti-CD20s. So I actually don't really recommend doing a pregnancy test, because if you think about doing it like a blood test, you do their pre-infusion labs like a month before their infusion, they can get pregnant in the interval. So unless you're doing it like right there, it's not going to necessarily be that accurate. They could do a home pregnancy test, but it's not positive until they're 4 weeks pregnant usually. So I think we don't want to impose that on patients. I have some patients who do choose to do a home pregnancy test before their infusion or before their injection, which is fine. But yeah, I would say in summary, there's no reason to think it's different with ublituximab versus ocrelizumab.
Okay. I have one more question to ask. So, patient who's doing physical therapy is really benefiting from it. The insurance company says no more, or the insurance company will limit their ability to get the PT because it's not for maintenance management. Is there any tricks or strategies you have for that?
So whenever anyone comes into the clinic that has any insurance limitations, it's one of the first discussions I have with them. And the reason I don't like to discuss insurance, but I think it's really valuable because, first of all, many patients don't know what their benefit is, and they're shocked when they're out of visits. Second of all, those are their visits to use, and I can make a recommendation to them. This is not traditional orthopedic rehab where you get seen 2 to 3 times a week for 6 weeks and then you're discharged. So if somebody comes in and they have a insurance benefit of 12 visits, I'll say, okay, let's do a couple of visits right up front, get you started on some things, and then let's see you once a week or once every other week so we can stretch those visits out. Both of the larger institutions that I've worked at have a self-payment plan that was very reasonable. So if someone had run out of benefits and they chose to continue, they could self-pay for a discounted price, which was very, very helpful. There used to be grants that we could go for. Those pretty much dried up. But I really try to involve the patient with their decision about how they want to use their benefit with my recommendation about what I think would be best for them. Then we've also coupled with area exercise facilities and tried to identify people in those facilities that have an interest or that we've educated in MS so they can transition to a community-based exercise program.
Okay. I will pass it along to see if our physiatrist has any good ideas.
Yeah, I'll just add, a lot of the benefits of any kind of therapy come from what you do at home. And so putting in the work at home is hard for some patients, but often is where a lot of those benefits come from. And it does allow people typically to spread out their interactions with a formal therapist. There are also some opportunities for engagement in lower cost plans that use physiotherapy assistants or occupational therapy assistants, other rehab assistants, for lower cost interventions between formal therapy appointments, which can also help with cost mitigation. But as I say, a lot of the benefit of therapy in that maintenance phase, if we want to call it that, is what you do at home. And it can be hard to motivate people to do that, but that doesn't mean that it's any less valuable as a therapy tool.
Okay.
I just wanted to add, in Canada, I don't know if you know about like the Sclerosis program, but there's some like online free therapy programs that patients I found are really engaged with and really enjoy, and like it gives them a group of people to meet with and do their exercise. So things like that may help spread into a broader environment.
Yeah, Neurosask is also available to anyone, anywhere. NEURONSASK because it's out of Saskatchewan.
Okay. All right. I think we're going to close this session. Thank you all for hanging out till the end, and I hope you enjoy the rest of your meeting here. So thanks.
Vaccine, they can delay up to 4 months of age, and if they don't get it by 4 months, they can't do it for safety reasons. And then some places have BCG vaccine or early measles vaccine, but if they were depleted, you would want to make sure they come up to normal before giving any live vaccines. But that's where we educate the pediatric provider and then kind of rely on them.
Okay, so thank you very much. Those of you who made it through the end of the day, thank you to our panel here. So many to thank. This was the start of the meeting, so we're thanks to CMSC for organizing this with us. Thanks to academic CME, Dr. Hayes and Nicole and Chelsea for the excellent organization of running this meeting. And thanks to Genentech for supporting it. So everyone have a great meeting. I'm sure it'll be the